Raj, things are all set?
All good, Patrick.
Yeah, things are all set?
Yeah, all good.
Very good.
Can you hear us? Can you hear us, Patrick?
Yeah.
Okay.
But sound is less.
I'm speaking from.
How's my sound? Can you hear?
Yeah, it's okay. Volume you can increase.
Can you hear now?
Volume is less.
Oh, put this damn thing and I'll see whether.
Yeah, fine. Now it's better.
No, that will not do.
I think it's lagging there.
Is it better now?
Yeah, it's better, definitely.
I guess I need to bend a bit. Now can you hear?
Yeah, good. Now it is good.
See, now it is good.
Sir, that is only for speaker.
Yeah, not for him.
That is good now. No, that is because this will only go to the main
Amplify.
amplify, that's all.
Patrick, this is good, right?
Yeah.
Okay, all right, thanks.
All right.
We are good.
Okay, shall I admit all?
Yeah.
But still 10 minutes are there.
Okay, I will, Sandeep, I'll be over there.
Okay, yeah.
I'll be in the lab.
Dr. Laha.
What is this?
I'll be in the lab.
Dr. Laha.
Dr. Laha.
Yeah.
So I'll take the pen.
Yeah.
How do we get this much, man?
Special connection.
Money collector.
That's what I heard. That's money collector.
My father is a fish field. It's just 1.5 hours from my wife's place. This is not the special laddu. They have vada.
I know.
You should eat on that same day.
Then one of you open the YouTube link, second day.
Yeah. It's still there.
I'll take.
This is different laddu. If you go to Kalyanam, like you have to sit there.
Like a special puja.
Then that might be for 1.5 hours. Then they'll give separate 2 big laddus.
Even general laddu is so good.
No, you do a lot of time to do something.
Yeah.
Okay, confirm. Someone confirm.
I'm okay, sir.
It's coming as your name only. There.
Oh, no, I have changed it. Yes. I relogged in, I forgot.
Everything comes.
There will be a lot of people for next question.
Oh, Sandeep, mute us. Oh, from here is okay. Okay.
Will you give an e-certificate for attendance?
Sponsor, I'm going to have to give an e-certificate for attendance. Certificate of participation. Generally, students may need it. Is it?
Morning, Professor Sharma. Can you hear me?
Can you hear me?
Yes, I can. Thank you. Good morning.
Good evening, good morning, whatever it is.
All right. Yeah, good. Good to see you.
Good to see you.
And I'm sorry that you both have to be at these early hours. I don't know whether you're online at home or at your department.
Well, I'm in my office. I had to come here.
Oh boy. Thank you.
So
Thank you.
I wrote to you an email. I woke up at 2:15 and then got ready and did prayers and
Wow.
came to my office at 5
Super.
o'clock.
Super. Good to see you.
Good to see you.
Yep.
So our session will start your 4 o'clock your time, right?
That's right. I think it should happen in another 5 minutes or so.
Yeah.
I already see the organizers being there.
Okay.
So we'll just hold.
Okay, no problem.
No.
Yeah. Good evening, Professor Sharma. Can you hear us?
Yes, I can hear you. Can you hear me?
Yes, and we can see you very clearly. Thank you.
Okay, thank you.
Video, start the video.
First, them. No?
Okay.
So I think for convenience, maybe we can just manage from here. Is that okay?
It's not working. That's what we.
Right now it's muted.
Raj, we can hear you.
No, you have to mute them. Now can you hear us, Dipankar?
Yeah.
Yes, we could.
Sandeep.
Professor Selva has also joined. Maybe you can make him also co-host.
Good morning.
Something happened to me. I'm off the line now. Okay.
Hey, Surendra.
Hey,
Morning.
Selva, how are you?
I'm great. Thank you. How are you doing?
Okay,
morning.
how are you doing?
Hey, Shashagri.
Hi, good morning, Silva. How are you doing?
I'm
Yeah.
good. Thank
Please
you.
make Professor Selva co-host too.
Yeah, it's kind of challenging to have an organized conference organized with so many time zone.
Yes, we do appreciate the fact that you all have to get up early and for this meeting.
Of course, it's a pleasure.
Some of us never sleep, so there's no problem.
Hello, Seshgiri, Selvaraj and Professor Sharma. This is Professor Rangarajan, Chairman.
Hi.
How are you?
Hi,
Hi,
Professor
good
Rangarajan.
Good evening.
Good evening, sir.
Good evening. Thanks for your consent to speak.
Of course, it's a pleasure.
Hello, how are you doing? We go way back to long. He was my
Yeah, it's been a long time. I've seen, yeah, I'm doing great, Raghunajan.
Yeah,
Oh, yeah,
very nice.
it's a great honor to be there.
Yeah.
Thank you very much, and Professor Seshagiri for inviting us to participate in this session.
Our pleasure.
We can start now.
Yeah.
Who's the rapporteur for this?
Yeah, so the big team can start, please.
Am I audible?
Yes, you're audible. Go ahead.
Okay. A very good evening to all of you. Welcome to the third session of the Biochemistry Centennial Celebrations. This session will focus on the reproductive biology and human health. This session will consist of talks by Professor P.B. Sheshagri, Professor Selvaraj Nataraja, and Professor Surendra Sharma. The session will be chaired by Dr. Purusharth Rajaguru. So Dr. Purusharth Rajaguru is an associate professor in the Department of Biochemistry. He joined the department in the year 2013. His research group focuses on the post-transcriptional gene control and specifically understanding about the mRNA fate decisions. So before proceeding for the talks, I would like to inform everyone that if you have any question, you can raise your hand and your questions will be addressed by the chair at the end of the talks. YouTube participants can post their question in the chat box. So now I request Dr. Purusharth Rajaguru to invite the speakers for their talks.
Yeah, thank you, Shweta. I hope I'm audible and visible.
Yes, can hear you. Not
All right.
visible though. Yeah, now, yes.
Okay, all right, thank you. Very good morning, good afternoon, or good evening to all of you, depending on which part of the globe you are plugging in from for this conference. Very warm welcome to all of you for this day 2 of the Biochemistry Department Centenary Conference. We witnessed an exhilarating mix of talks, not only from the invited speakers, but also from our very own students yesterday. And this was followed by some very interactive session at the end of the talk. And despite all this, we managed to stick to the schedule of day 1. I'm really hoping that we'll be able to continue this trend today as well as tomorrow. And we look forward to your cooperation in this direction. We will now dive into the first session of the day, which happens to be the 3rd session of the conference. This will be focusing on reproductive biology and human health. In this session, we have 3 eminent speakers who are representing diverse research environments. The first speaker will be Professor Seshagiri, who is from IISc. This talk will be followed by a talk from Dr. Selvaraj Nataraja, who will present his research experience in the pharma industry. And then the final talk in these invited talk series in the afternoon will be given by Professor Surendra Sharma, who will join us from Brown University. So the last 2 speakers are joining us from the East Coast of United States. And I guess it's like probably 5:30 a.m. or 6 a.m. there in the morning. So good morning to them. And thanks once again for joining so early in the morning. About the first speaker of the session, Polani B. Seshagiri is a professor and chair of MRDG department at IISc. MRDG stands for Molecular Reproduction, Development and Genetics. He's an alumnus of our department. And after doing PhD in our department, he did his postdoctoral studies at University of Wisconsin-Madison. He then briefly got associated with Primate Research Center at the same university, and subsequently joined as an assistant professor at the MRDG department. His group is interested in studying blastocyst development, and they specifically focus on differentiation of pluripotent stem cells into trilineage cell types with an aim to manage human infertility and health. With that brief introduction, I extend a warm welcome to Professor Seshagiri to share his exciting work with us. Professor
Thank you. Thank you, Professor Purushottam Rajaguru. At the outset, let me thank the BC Centenary Year Celebration Committee for providing this opportunity to speak in this forum, and importantly, to include a very dedicated session on reproductive human health. Particularly, it's my pleasure to thank my long-term associates, Professor B.N. Rao and Professor Rangarajan, for the companionship in many, many ways, as well as Professor Deepankar Nandi for steering these activities, which is really a monumental task given the time zones and many, many things. Let me just share my screen, and then I hope I don't goof up here. Just hold on.
Professor Seshagiri, we cannot see your screen yet.
Yeah, that's why I'm trying to.
Now go to the desktop and open your PPT. Yeah.
Yes, we can see it now.
Can you see?
Yes, yes, it's visible.
All right, thank you very much. I'm sorry for this little hiccup because I'm not one of those computer savvy guy to do this. Let me just get started with my talk outline. So what I'm going to be doing today, given this 25 plus minutes, is to give the need and the genesis of reproduction research, particularly how it evolved in Indian social science and what we have been doing in the last 2 or 3 decades. And importantly, I'll focus more on the biology of blastocysts and its hatching phenomenon from the particular point of view of regulation of blastocyst hatching, both from the perspective of molecular aspects as well as cellular aspects. And from this basic research that we have been doing, how important it is clinically from the point of view of managing infertility. I need to correct Professor Purushottam Rajaguru that even though we have been working for over a decade or more on the pluripotent stem cells, but not in the context of infertility management. Sorry, Purushottam, for pointing that out. Let me just get on with this. Why is there a need to look at reproduction research? And I'm giving an overview and an overarching issues here with respect to reproductive health, not necessarily confining to the female. It is both male as well as the female. The major issues on the infertility. And given the topic of today's discussion and given the background of both the other 2 speakers, I'm just trying to say something about the female reproductive health. And I mentioned a number of these, premature ovarian failure, polycystic ovarian syndrome. You could read through reproductive tract infection, recurrent pregnancy loss, preeclampsia. One of our internationally renowned subject experts is going to be speaking on that, and a number of these issues, which are extremely important in the context of managing women's reproductive health. And therefore, it becomes absolutely necessary for getting a lot of basic research done with huge translational implications. And therefore, it is necessary that we get a perspective of what's going on. And thanks to the organizing committee, and it's very fitting that these key reproductive issues are specifically covered here. And I'm going to be speaking on the topic that was mentioned here and a couple of others. A couple of statements that I want to mention here, like our chairperson mentioned, Professor Selva Nataraj is an alumni of this department. He had his PhD from Professor N.R. Mudgal, and Professor Surendra Sharma is, though not an alumni, he has been a very long-term associate of Professor M.R.S. Rao, and I given to understand that both of them had great friendship and companionship during the Baylor College of Medicine days when they were over there for a few years. Now, coming to the reason why this research is so important, particularly from the point of view of Department of Biochemistry and how this took off since the early, I would say late 70s and the early 1980s, is simply because there was a very vibrant reproduction research program initially driven by Professor Mudgal and followed by Professor Adiga. And I happen to be the student of Professor Adiga, and his interest has been a very classic biochemistry, particularly from nutritional biochemistry point of view, and how important is the vitamin carrier proteins during transplacental vitamin transport and its role in early embryonic development. And that time we had 2 of my senior colleagues who had very stellar publications with respect to the functional significance of vitamin carrier protein, particularly riboflavin carrier protein, and how immunological interference, both either by active immunization or passive immunization, resulted in termination of pregnancy. And that was a time which were very exciting times in looking at immunocontraceptive methods in our country, and our national funding agencies were pumping in a lot of money. So there was a very great national interest in this area. But my own interest when we got into this area of research is basically out of my interest was trying to figure out why is there a loss of embryos post immunoneutralization of vitamin carrier protein. And we dwelled on this in a number of ways, but it was rather difficult to figure out where was this embryonic loss occurring. And that time we didn't have the great facility to look at the embryos. And you will see in my talk how important it is to look at this microscopic minuscule entities. And so we had no idea and no clue what has been the reason for loss of these embryos during peri-implantation period. And at this time, I'm talking in 80s, Anne McLaren, which I consider as a mother of mouse embryology, and Professor Barry Bavister had visited our department, and then the biochemistry department, and had a fantastic lectures, and that really has impacted my passion to do research on blastocyst development, and then therefore went on to University of Wisconsin, worked a few years in Barry Bavister's lab, and then followed by now working as a scientist in Primate Research Center. So that's the background, and there is a Professor Adiga's legacy. A number of us have come back to the institute in our faculty capacity and carried on with our careers. Now, this is more a tutorial slide just to set the stage, more importantly for students to tell what really is early embryo development or early mammalian development. Post ovulation, the oocyte would need to be fertilized by a single sperm to form a zygote. And that zygote goes through a mitotic cleavage division, a series of them, and forms a kind of a compacted cellular entity called the morilla, because it looks like a mulberry, and it starts to cavitate and form a blastocyst. And until this pre-implantation development, because it has not implanted into the uterine endometrium, these stages of embryos, which happens in about 3 to 5 days, regardless of the mammalian species, is encased in a protective acellular elastic glycoprotein coat called the zona pellucida, and you'll see that here in the white format. And once the blastocyst is formed, this blastocyst has to rupture the zona pellucida, come out of it by a process known as hatching, and only the hatched blastocyst will oppose, adhere, and invade the uterine endometrium by a process known as implantation. You will appreciate at the end of this talk, this blastocyst hatching and implantation are extremely crucial for the establishment of early pregnancy. If there are any aberrations, either due to cellular or molecular causes, there is not going to be development of blastocysts, there is not going to be hatching of blastocysts, and consequently the implantation of blastocysts. So it becomes very, very critical. And therefore, we have been working for a number of years on these aspects. I'm just brushing here quickly that we had been working in the last more than 3 decades on sperms fertilization competent, particularly from the point of view of flagellar tyrosine protein phosphorylation, ovarian follicular development, multiple ovulation to produce multiple embryos, blastocyst development and differentiation, and blastocyst-derived pluripotent stem cell for lineage-specific differentiation. We've used both rodents and the primate model, depending on what we are actually intended to do. So this is basically what we would have been focusing in the last 2 to 3 decades, particularly on the blastocyst biology. And what I would like you to remember that these earliest stages of development occur in the female reproductive tract. These are microscopic, inaccessible for experimental manipulation in vivo, and therefore you need to either have an in vivo developed or in vitro produced embryos to study all our biology. So this is the development of blastocysts. This is the morilla picture, which I've taken some years ago. It cavitates and forms what is known as an early blastocyst, a mid or late blastocyst, and expanded blastocyst, and fully expanded blastocyst. It takes several days depending on the species, not more than 6 days. And I'm going to share you a video. I hope you'll be able to see this. Just tell you what really is this phenomenon of blastocyst hatching. And it just expands and it just ruptures the zona pellucida. And what is the mechanism of the rupture of the zona pellucida, which is intrinsically embryo driven, or is it intrinsically in an in vivo female reproductive tract mediated, or both is something which had been enigmatic for a number of years. And that has been the kind of crux of this entire biology of blastocyst hatching that we have been looked at it. So this blastocyst expands and expands and ruptures the zona pellucida, and eventually it comes out of the zona pellucida. Now, this is driven by the outermost single layer of epithelial-like cells. We call that as trophectodermal. I'll show you some pictures where you clearly see them. And this trophectodermal epithelial-like cells is the foot soldier kind of cell types to bring in this effect of hatching, A, B, in terms of invasion into the uterine endometrium for implantation. I would consider, similar to cancer biology, these cells become extremely invasive, something similar to cancerous metastatic cells. I'm not trying to tell that these are metastatic cells. These are normal developing cell types, which are critical for bringing about this hatching. The rest of the cells which are there in the blastocysts are largely completely undifferentiated cells. We call these as inner cell mass or inner stem cells. Therefore, you basically have two different cell types here. One is fully differentiated trophectodermal cells, the yellow ones, and then you have the undifferentiated inner stem cells or inner cell mass. So if there is a successful hatching, it will go through implantation and then everything is fine and there's a successful pregnancy. But for some reason, if hatching doesn't take place, it will not go through implantation. There is failed hatching and therefore failed implantation. So I consider that hatching and implantation are extremely rate limiting steps to initiate pregnancy. And in fact, this is extremely important because there's a huge amount of embryonic loss that occurs during the pre-implantation period and during the peri-hatching, peri-implantation period. And that has a huge bearing with respect to those infertile couples, particularly infertile women, where they cannot carry the pregnancy. And there is a huge pregnancy loss, and the birth rate by the assisted reproductive technology is still happening to be only about 14 or 20%. So this is a problem. So what are the reasons for this hatching implantation failures? I'm just listing here a number of things, and you can just read through. But of interest today is that we are going to be looking at the expression and function of some of these key regulatory molecules that are influencing the phenomenon of hatching. How do we know these molecules and where are they produced? Many reproductive biologists working at early development have invariably and largely worked on embryo implantation phenomenon, and very few used to be working on hatching phenomenon. And the huge literature that is available, several hundreds or maybe even thousands of papers on the implantation associated regulatory molecules, which are critical for implantation. If you take a couple of steps backward and look at it, whether or not these molecules would also be involved in the hatching process, I've listed some of them here. Could be hormone, growth factors, proteases, cytokines, addition molecules, transcription factor, etc. So whatever I have highlighted here in this kind of a light blue are the molecules, some of the molecules that we have been actively involved in the last 2 years, and I will get on with that. If one needs to work on this, you need to source embryos. Where do you get these embryos? It could be rodent model, it can be large mammalian species like domestic species, it could be non-human primates, it could even be humans. But the problems with the large mammals is it pose a huge number of technical, practical, and ethical reasons, and therefore it's not always possible to work with large mammals. And more importantly, they are invariably monotrochous or a ditochous animal, which means they'll produce only one oocyte, one embryo, or one pregnancy outcome in terms of live birth. And you need multiple embryos if you're doing it in vitro. And that's the reason why you have the rodent models, which become extremely important. Now, several years ago, we have been working on the primate. We no longer work on this for multiple reasons. The idea had been to produce multiple ovulations and multiple embryos. Therefore, you have primate embryos to deal with. And we had developed a non-surgical method to recover these nutrient stage pre-implantation embryos to study their development in vitro. But I will not go into those details except to tell that we have been intensely working on the two rodent models that I'm showing here, the hamster and the mouse. And of late, we had also dwelled into human embryos, and I'll get to that shortly. Now, how does mouse blastocyst hatch? They hatch after expansion, like I mentioned here. The zona pellucida would need to be ruptured, and then the ruptured blastocyst now can attach and invade the uterine endometrium. And please remember, that the zona pellucida remains largely intact, number one. And number two, the hatching occurs when the blastocyst is completely inflated. In contrast, and very excitingly and interestingly, when you look at the hamster model, these are our live pictures of hamster embryo development, right from the non-compact cleavage stage to compaction, morula, early blastocyst, late blastocyst, expanded blastocyst. And two major things happen during hamster blastocyst hatching. One is there is a deflation. You see the perivitral space here, enormous. And secondly, there is a global dissolution of the zona pellucida, and therefore you have an azonal blastocyst, which is now completely hatched. And importantly, this particular animal species and a couple of others too produces what are known as trophectodermal cytoplasmic projections. And I'll show you some pictures where you will see them very, very clearly. These are very exciting and interesting observations. So in summary, hamster blastocysts expand, they deflate, and there is a appearance of trophectodermal cytoplasmic projections. We call these as trophectodermal projections of TEPs, and the zona pellucida completely dissolves, and then you have these hatched blastocysts attaching. And so these are very landmark features in terms of the behavior of hamster blastocyst hatching, and there are a couple of other species which have these projections, but I will not go into the details. But just to bring in, say, I try not to put more of the data-heavy slides, just to conceptualize what we have been doing, A, and B, to make sure that we develop a story, a long story that we have been involved in the last couple of decades. Now, this is in a nutshell. So we have an experimental system where we can get these embryos to go through hatching. Over decades, we developed a culture medium, which is completely indigenously prepared, protein-free culture medium, except that we need to add BSA for hatching. And there's absolutely no serum, except that you can use a protein surface or a serum-coated surface for embryo to attach and differentiate the trophectoderm into trophoblast, simulating implantation process. So these have been very well worked out and extremely fastidious culture system. And you need to have a very high quality animal research and a good hands to do these kind of experiments. So I'll quickly go through I'm going to just go through what really is the mechanism of blastocyst hatching and its regulatory phenomenon. So these are some of the topics that I'll just quickly cover. The first thing, almost about 20 years ago, I think, one of my earliest students, Archana Mishra, did this very classic experiment which appeared in Molecular Human Reproduction. It's a big deal for reproductive biologists anyway. So what she did is when she cultured these blastocysts, they hatched out of the zona pellucida. Now, these are blastocysts which secrete out the proteases. That had been the hypothesis. So she did a very indigenous experiment to co-culture pre-blastocysts not ready to hatch embryos along with hatching blastocysts. And she observed that the zona pellucida of pre-blastocysts not ready to hatch embryos as well go through hatching. This indicates that there is enormous secretory proteases which not only digest the zona pellucida of the hatching blastocysts, but also anything co-cultured. And this invariably is of the same species. That is the hamster, whether we put 1 or 2 or 3 or 4, 8 or more, etc. Excitingly, if you put it in those conditions, either the rat or the mouse embryo, it does not hatch. That includes even the sheep or the human outside, it doesn't hatch. So what this illustrated is that the embryonically secreted proteases are competent enough to hatch the zonas of even pre-blastocyst stage embryos, and it is species specific. And that was the thing. So the idea was to look at what kind of proteases biochemically. You have a whole range of serine, aspartate, cysteine proteases, so on and so forth, metalloproteases. She and others have done in our lab a whole range of inhibitors and then see which one is affected. And it turned out that the cysteine proteases are extremely critical for this hatching. And one of my students exogenously incubated the embryos with some of these cysteine, purified cysteine enzymes, proteases. And these show up very clearly this is exogenously added enzyme bringing about zona lysis. And this is a control where you do not have exogenous. And at the same condition, when you put co-culture, both the hamster and the mouse doesn't show, but the hamster shows. So a series of experiments have been done, in terms of mRNA expression, in terms of protein localization in these embryos of these peri-hatching blastocysts expressing cathepsins. So in a nutshell, what is the working hypothesis? That these enzymes are produced perhaps by the trophectodermal cells, and may probably by forming in these trophectodermal projections. The embryonic expression of these proteases are regulated by an upstream regulatory molecules. Perhaps the initiator is sex steroid hormone, let's say estrogen, which induces growth factors, which is very well known. Also the cytokines, which is very well known in the endometrium. So we hypothesize these perhaps are involved in the embryonic expression of proteases, particularly cathepsin, and a whole range of these molecules are involved in. And this is the experimental system we had been using it for over. I just give a couple of quick examples of how this happened. This one example, this appeared a few years ago, and again in molecular human reproduction. The COX-2 expression has been shown, and if you inhibit using a potent inhibitors of COX-2, there is a dose-dependent inhibition. And this inhibition can be reversed by exogenous supplementation of prostaglandins, particularly E2 and I2, and E2 showing a much better influence than the I2. I'm not showing the data here. So likewise, there are few molecules, and I'm showing the IL-1 involvement. And this is the experiments which were done by Madhulika, where she looked at cytokines and the role of IL-1 and the antagonist IL-1, IL-1RA, and then looked at the embryonic expression of catyls, hatching phenomenon, and the number of downstream. She compared both, not only in the mouse, but also in the hamster, and see what is the relative roles of this. This is in a nutshell that IL-1 beta has a very profound role in the embryonic expression. First of all, in development and hatching rate being high, and embryonic expression of these enzymes involved in hatching, but not the serine proteases. And that's the nutshell. So if I capture all these points, this has been the kind of a working model for over years. There are hormones which regulate some of these regulatory molecules, ultimately leading to embryonic expression in the hamster cathepsins, and perhaps in the mouse, the interstitial implantation serine proteases, ISPs, and these are involved in the hatching phenomenon. And I'm going to shift quickly with a couple of slides, some of the behavior of the blastocyst hatching in the hamster that I just mentioned earlier. When we localize many of these happening molecules, and I'm giving some examples here, cathepsin or COX-2 or IL-1, which I'm not showing here, they all get predominantly, obviously localized in trophectoderm, and more importantly, and these trophectodermal projections, and you will see that in here. These are very dynamic cellular events. And what is the significance of this? A, in the context of hatching, B, perhaps in the context of these embryos crawling towards the implantation sites, and whether or not they facilitate invasion implantation is something for us to figure this out. We have been always comparing the kind of observations parallelly in the rodent model, in other rodent model, the mouse. And like I said, they don't deflate, and they, in an inflated state, hatch. Now the question arises, is there any kind of an embryonic polarity when they hatch out of the zona pellucida, say in the hamster or in the mouse? These are something which are very compelling questions as a biologist to look at it. And here is an example of the mouse. Looks like it is at the embryonic pole where the ICM is, but not necessarily. We have looked at a number of immunostaining, immunostaining, the phelidin staining, and we saw that it is not actually specifically embryonic pole or abembryonic. It could be both, but we do not know how this. But it's very difficult to start in the hamster because of this deflation, and therefore there can be a rotation of the deflated blastocyst within the zona that is still remaining. The point I'm trying to tell is this exciting observation that many of the regulatory molecules, at least half a dozen of them that we looked at, it is trophectodermal localized and enriched in the trophectodermal projection. This is the important thing. And what is the significance? Hatching regulated molecules are localized in TAPs. TAP is more abundant, more in the TE in a polar fashion. So that's the key thing. And I'm going to skip this one. These are very important stuff with respect to the mechanism of ZP digestion by the proteases. And if someone has a question, I can tell you. Very recently, we did some bioinformatics to show the specificity of proteases on one end and the ZPs on the other hand, why hamsters have a global dissolution, whereas the mouse doesn't. And I will not go into detail. These are very exciting take-home points so far. And I'm just going to quickly go through a couple of slides, which I feel is extremely important before we close this. Sorry if I overshoot by a minute or two, but I feel it's extremely important. In this, the picture that you see is Professor Bob Edwards from Cambridge. And this is, I think, 2001 or 2002 or 2001, I think, in Professor Anand Kumar's house. We had a dinner, and he and during few visits of Anne McLaren, were insisting that our department should be working on human embryos, even though it's extremely challenging, technically difficult, and ethically kind of restrictive. But I just want to give an overview of human infertility and the assisted reproductive technology current scenario. Even though it has been over 4 decades, 1 to 6 out of 6 couples are infertile, and I'm giving the statistics here, so on and so forth. The number of reasons why there is no improvement greatly with respect to take-home babies. So the question arises, where are the difficulties? And it can be multifactorial. I will not go into the detail. One important crux is, how does an embryologist make a choice on the good quality embryos versus not so good quality embryos while doing an embryo transfer? Because that is going to be the determining factor with respect to the pregnancy outcome in terms of childbirth. And that take-home baby rate is what is extremely critical. And therefore, there is always an urge to figure out, can there be a very objective embryo biomarker, which as an adjunct with the embryo scores that the embryologists give would be a way. And that gave us an opportunity. Sorry?
You have about a minute left, Professor.
Yeah, I'll finish this. So it was a multi-institutional program that we just embarked a few years ago, thanks to Imprint, MHRD, and ICMR, and roping in assisted reproductive technologies, gynecologists, andrologists, embryologists coming together. The question we asked is whether the biomarker would be having a predictive value to choose the embryo, whether it will produce a live birth or no birth. I'm giving an example of 2 molecules here, HLA-G and ubiquitin, and it is a developmental correlation that we're showing here from the cleavage stage of blastocyst, and importantly, but between the live birth and no birth. This is completely a retrospective analysis that we have carried out. It's a huge task that one needs to do. And very recently, I've been looking at the omics profiling of these in terms of live birth, and they're a very interesting story with respect to metabolome as well as the proteome. I'm just giving a data here, which I will not go into detail. So the futuristic important thing would be to propose a prospective correlative study to look at the embryo viability biomarker and embryo score criteria driven choice of embryos for embryo transfer to ensure live birth. Thank you very much. I'm going to skip all this and then just thank some of my wonderful students who were involved in embryo research and a whole bunch of others. And I need to thank the whole range of funding agencies, particularly DBT, DST, and the Imprint program to support our research. Thank you all for listening.
All right. Thank you, Professor Seshgiri, for a very insightful talk. And apologies for getting that fact wrong in the beginning. We tried to put together things from the website, but I think we goofed up somewhere. We can take some questions now. People can either raise their hand or write the question in chat box. Yeah, Dr. Selvaraj.
Yes, Sheshagiri, thank you for giving a wonderful overview. I think it makes my life easier now. The next talk, as you'll see, I have a comment and a question. The comment is, about what you made about comparing the embryo development to metastasis. And in fact, David Gardner, I listened to many talks in several conferences. He clearly makes this analogy of how the embryo development is similar to what happens in the metastasis. And in fact, one of our own alumni, Mani, who is in Baylor's College, in MD Anderson, and who is GP's student.
Oh, yeah,
Mani. Exactly. He has published a very nice paper comparing the gene expression in metastatic and to the early embryo development. So there is a very good connection between those two. And the phenomenon, if you look at, it's very similar in terms of invasion and implantation. So that's my comment. And my question is actually about this trophectodome projections that you see in hamster. And as you pointed out, it is specifically only in the hamster. So, in terms of other species embryos, how are the proteosomes or the other caspases that is required to break down this zona and also the endometrium?
Coming to your question on the trophectodermal projections that we observed primarily in the hamster, we investigated in several other species as well and also dug the literature. In fact, the other species embryos also, blastocysts also have this kind of projections. And it's very hard to figure this out because when they hatch, they are in an inflated state. So you have to come up with a mechanism of taking a kind of a series of pictures to figure. And we do observe this in the mouse. And there are reports in the human as well, and a couple of other species. So that was one thing. And I presume that these projections perhaps is the business hub of happening in terms of hatching and implantation. But people have to investigate in a bit more detail on that.
So Professor Sasagri, can you stop sharing the screen so we can see you?
Yes, thank you.
All right. So in interest of time, if there are no more questions, then I think we'll move on. Thanks again, Professor Seshgiri, for agreeing to be part of this celebration.
Thank you.
The next speaker is Dr. Selvaraj Nataraja. He is also an alumnus of Department of Biochemistry. After doing his PhD from the department, he did his postdoctoral stint at University of Illinois, Chicago. Since then, he has been associated with the pharma industry. He was the founder and head of startup Tocoferix, and he's currently the senior director at MitoBridge. His research interest includes drug discovery and therapeutics development in reproductive research. With this short introduction, I would like to invite Dr. Selvaraj to share his science with us. Dr. Selvaraj?
Thank you. Can you see my slides?
Yes, we can.
Okay, perfect. So let me jump in. Well, let me take a pointer so that it'll be easier. All right. Okay. Thank you, Chairman, for the kind introduction. First, I would like to thank, Professors Dr. D.N. Rangarajan as well as Dipankar Nandi for organizing this excellent conference and inviting me to present, as well as to Professor Seshgiri, who has been a key organizer for this session and for giving this wonderful key start of the session. And it's my great pleasure to share my journey, with the young trainees and faculty of my alma mater, where I took my baby step in research. So currently, as you said, I'm working at MitoBridge or Astralas, but I will focus on my earlier work done at Tocoferix as well as at Serono. Thanks to my mentor, Professor Maudgal, he gave me the excellent opportunity to become the confident researcher that I am today. And I just want to say that biochemistry is a great place to learn, and the training provided is second to none in the world. And in addition to the academic challenges, and learning, there are a lot of extracurricular activities that you can be involved in, and I had a ton of fun, during our days. So for all the young trainees here, make great use of this opportunity available for you to learn, and also don't forget to have fun during your stay here. And I will also like to share one old picture, taken during my days when we were the graduate student, and during one of our outing, and you can see a couple of the faculty, the current faculties from biochemistry. You can see here my good old friend Utpal Tatu, and also Rangarajan here, in this picture. You can see, they look all very young, and so we are all very young at those days. And so now let me get to the the business part. So this would be the agenda for today. first I'll give you a brief introduction for the drug discovery, followed by highlighting the unmet medical needs in women's health. And Sheshagiri provided a wonderful overview, and he identified multiple areas in the reproductive research, which working at Serono, we addressed several of them, and in each one of them we can give an hour's talk. But today I will focus on this infertility treatment, on the control ovarian stimulation and IVF, which again, Sheshagiri gave a wonderful talk. And primarily, I will focus on the novel therapies that we had embarked on the long-acting protein therapy, as well as the oral small molecule. So just to give you a brief idea about this drug development, it's a long process. It requires a whole village to accomplish the task. And it takes about 10 to 15 years, to develop a product, with a significant cost to develop a product, anywhere from 2.5 to 3.2 billion dollars to get one product to the market. I mean, this was a number which was provided by Joe DiMasi from the Tufts Center of Drug Development. And the main driver for the cost is the higher failure rate, in this endeavor. So for every one drug that gets into the clinic, for every 10 drugs that get into the clinic, only one gets approved. So overall, it's only a 10% success rate. So that's really, big, high risk, for the drug developers. However, if you get one drug that becomes a blockbuster, that can take care of the entire, the chain. And so drug development is a very high risk. However, it also has got high return undertaking. So, so the involvement of the people in this industry, is significantly risky. And so there are not too many people, who want to be involved, especially in the reproductive research. So I just want to give just an overview of the drug development process. I mean, it might be very introductory, but I just want to bring out everybody to this to the page wherein you will understand what I'll be talking a little later. So R&D is formed of the research, which is the early part, and development is where you do in the human, the clinical trials. So in the early part, in the research is to identify the disease indication is identified, and the target as well as the new molecule is developed during this early phase of drug discovery. Once you have a molecule that you want to move forward to the clinic, then you go into the preclinical stage, or what you call as the IND enabling stage, which stands for investigative new drug stage. This is primarily you're looking at the GLP and the GL tox study, and identifying the safety toxicology profile of the compound, as well as the GMP manufacturing, to make sure that you have a control over the process of manufacturing the drug. And then you apply for an IND, which is the stepping stone to get into the next phase of this drug development, that's getting into the drug development. In fact, just 10 days back, we submitted an IND for one of our compound in the current company, and it's a big milestone for researchers. This is where now it's possible to get into the development and to the human. So it's a big champion moment. And once you get into the clinical trial, it's a whole different ball game, and you need a different skill sets for this. So I just briefly mentioned, I highlighted about the different stages of drug development. The phase 1 is usually in the healthy volunteer study, and where the goal is to look at the safety profile of the compound and the pharmacokinetics. And in some indications like oncology, the phase 1 could also be patient population. But more generally, it's the healthy volunteers to get an idea about the drug, because this is the first time it's dosed in human. And the phase 2, this is where the tires meets the road. That means the molecules is tested in the disease models. And usually you will have about 100 to 500 patients in the phase 2 study. And this is also called the valley of death. The reason is I told you about the 10% that succeeds here. Most of the failure occurs here in the phase 2, because this is where where the actual human disease is tested with the molecule. And the translatable of the animal models that you have in this preclinical study is very poor. So there's a lot of drugs, fail in, this phase 2. So that's why they call it as a valley of death. And once if you are successful in phase 2, then you go for phase 3, where you have a much larger patient trial, and in the phase 3, and if you succeed, go for the NDA or new drug application, and then it's a bigger champion moment, for the company. And so this whole process, as you can see, is very costly. And if you fail in the later stage, it becomes much more costlier for the drug development. So one of the mantra, in drug development, if you are fail early. And so even if you are if you can identify the causes for failure early on, it's better for the organization. All right, now let me go back and talk about the reproductive research. And Sheshagiri again made my life easy. So as he pointed out, according to WHO, there are about 72 million infertile couples in the world, and it's about 12% of the child. Out of this, only about 50% seek treatment with the physician. Of this, only about 20% take any kind of treatment. As you can see, there's a significant drop in that. And the first line of treatment is ovulation induction by the clomiphene citrate. This is a estrogen receptor antagonist, and it is supposed to block the negative effect on FSH, so that you get an increased follicle stimulating hormone, which can stimulate the follicles. Unfortunately, this the success rate is very poor in this. And in fact, recently they've done a trial comparing a placebo, and the clomiphene citrate did not do any better than placebo. However, this is a very cheap drug, and it can be convenient because it's oral drug. So this is still preferred for an early treatment process. And people who fail here then go to the more aggressive treatment. That's the controlled ovarian stimulation followed by IVF. Again, Shreyas gave a wonderful overview on that. And as you can see, there's only about 3% of the patients who from here they go into this IVF for this most aggressive treatment. The reason for that is the cost. The cost is pretty enormous, for going through this process. And the second is also the convenience and the compliance. The patients have to take a lot of injection, so it's significantly stressful for the patient. And so that's why only 3% of the patient persists through this controlled ovarian stimulation process. So if you can have a better treatment process, then you can certainly increase the percentage of the patient taking this the treatment. So the next slide, I just gave a brief overview on what Shreyas pointed out. The controlled ovarian stimulation protocol involves multiple injection, about 10 to 14 injection. Also, these GnRH antagonists to block the premature luteinization. And the whole idea is to induce multiple follicle, as Shreyas showed in his slide. And after that, you give an injection of HCG, or human chorionic gonadotropin, to induce the ovulation, isolate the egg, do the in vitro fertilization, and transfer the egg either day 3 or day 5. And so this is the whole process of COS and IVF. So again, as I pointed out, currently there are 10 to 24 injection of FSH, which is very stressful for the patient. And these injections are self-administered, and you can imagine the anxiety the patients go through. So especially when you talk to the physician and the patient, the significant they want improvement on the convenience of this treatment process, and also the compliance of the the treatment, because sometimes when they're self-injecting, they don't give the exact drug that's required. So how can we improve the convenience? Either you can reduce the number of injections here, or you can develop an oral therapy. So this is a couple of things that I will show examples of in both this model of therapeutic development. Okay, so just taking a step back and then trying to understand what is the structure of the gonadotropins. And Rajan Dige, has done a fantastic job in understanding the structural function of this FSH hormone as well as other gonadotropin. So this belongs to the FSH, the follicle stimulating hormone, as well as other gonadotropin. They are dimers with a common alpha subunit and there's a hormone-specific beta subunit, and so the FSH, and in each of these subunits, they also have the glycosylation sites, and this glycosylation is critical for maintaining the activity as well to inducing the receptor, as well as for the half-life of the protein. So the LH, again, has the common alpha and the beta subunit, and HCG is very similar to LH. Which it binds to the same receptor as the LH, but it has an extended C-terminal peptide, what you can see here, with the 4 additional O-linked glycosylation. And this molecule is more potent than LH, as well as increased half-life, in this circulation. So clearly, I think the glycosylations provide a significant effect on this half-life of the compound, this molecule. So Irving Boimi did an excellent work where he tried to stick this C-terminal tail into the FSH subunit and the beta subunit of FSH, and so increased the glycosylation. And with this construct, he showed that the activity of the FSH increased, as well as the half-life of the FSH. So I will just briefly talk about this a little later, about this molecule as well. So this is the molecule which Irving Boimi had developed, and Organon, which is a competitor for the company that I was working for, in-licensed that technology and developed this long-acting FSH, what they call as Organon 36826, where they introduced the CTP in the beta subunit. In parallel, at our company, so what we did is, we tried to introduce additional glycosylation in the backbone of the FSH molecule. So in both in alpha and in beta subunit, we did some site-directed mutagenesis to include an N-linked carbohydrate protein. So we had either in a beta subunit or in both alpha and beta subunit, we introduce an extra glycosylation, and so to understand whether this can provide a long half-life. So before I go into the data, I just want to briefly mention about the model that we use to test these molecules. I mean, this is the immature rat modified version of Steelman Foley assay. This was developed sometime, I think, in 30s or 40s. This is a very old model, but this was adapted and for testing it. And one other thing I need to point out is this is a model which in reproductive biology people use, and I have used extensively during my days in Professor Mothgall's lab. And so it was very easy for me to adapt this model. And as you can see, in this animal model, for 2 days, you give 4 injections of FSH twice a day, and following that, you give an HCG, and you can see the eggs. And this model is very similar to what you see in the women going through the IVF, where you have multiple FSH and then HCG, and you can get the egg harvested. And in fact, this model is used to evaluate the activity of the FSH that's used in the clinic. So what I'm trying to say is this is a highly translatable model. And if you remember, I talked about the valley of death in the phase 2. So in this indication, we don't have the valley of death. If you have an activity in this model, you are sure that in the clinic you will be successful. So that's the thing. Having said that, so let me get into the data. So with the modified FSH, I'm just going to show you dose dependently here. The animals were given just 1 injection of FSH, of this long-acting FSH, and you can see that dose dependently, it can induce the number of eggs that's produced. Whereas the control in the green, when we used a single injection of FSH, it was ineffective, in inducing that. And on the other hand, if you do 4 doses of FSH, like FSH, either FSH or a compound, shows the same process. So clearly show that we have a good long-acting molecule, FSH molecule, and this could be used in the clinic. And of course, we took this molecule into phase 1 and phase 2, and Unfortunately, while we were working on this, our competitor Organon with the FSH CTP, they were already well advanced in the advanced phase 3 stage. So internally, strategically, the company decided to not pursue this product, despite that we had a good activity in the human in the phase 2 trial. And at that time, everybody was upset working in this project. In the hindsight, it was a good decision, because I'll show you why. And so just to continue this story, I'll give you an update on this FSH CTP. So how the development happened with this molecule. So they did a phase 1 study with about 24 women. And in this, they're just given a single injection of FSH. And then after 7 days or 8 days, they did an ultrasound. And so, as I pointed out, we have a good animal model. And in this, even in the phase 1 study, they could see an increase in the biomarkers, the estradiol as well as the inhibin, which is a good biomarkers for FSH. And by looking at the ultrasound, they could show that this induced, in fact, with one dose, they could induce the multiple follicles. So already, we have a proof of concept even in the phase 1 study. And then again, of course, they went to the phase 2 where they used about 100 patients. And in this study, they did the multiple doses of FSH. And again, this is the disease model. So where they did a controlled ovarian stimulation and before the IVF. And in this case, so they showed that if you give one injection of FSH, and then after 7 days, you can follow up with, one injection of the FSH CTP, and then follow up with the FSH, and they can see the success here. And again, in the phase 3, also, they went and did about 1,400 patients, and they were able to show that it was comparable to the FSH daily injection of FSH. So based on that, they applied for NDA. And by this time, Organon was taken over by Merck and Company. And so Merck applied for NDA to the EMA, the European agencies, and they approved this as a treatment as shown here with one injection of the FSH CTP. And after 7 days, you started up with the recombinant FSH. So there was, 6 injections reduced in this. It was better. But one of the issue with this FSH is that the side effect is the ovarian hyperstimulation syndrome. So some of the patients it can be fatal. So once it happens, then they have to withdraw the treatment. That was the process in the IVF cycle. Unfortunately, with this FSH CTP, once you inject, then you lose control. So the clinicians were not very comfortable with that, and so were the patient. And because of this, the uptake in the market was not pretty good with this long-acting FSH. And unfortunately, because of the ovarian hyperstimulation syndrome, it was not approved by FDA. So it's right now it is sold only in European Union and other countries, but not in US. But this also gives an opportunity to develop a small molecule oral therapy. So I think I'll part of the talk. I want to give a brief introduction about the structure of FSH receptor and how we went ahead and do that, because we had done several years back the FSH small molecule direct activator. It is it's impossible to replace a molecule like FSH, which is 33,000 molecular weight, with a small molecule of 500 or 600. So we were looking for a direct activator. And in the meantime, in our group, Julian Zhang, he published a nice paper on the crystallization of the FSH receptor, and he identified some mechanism how we can use this small molecule. And so based on that, we decided we will go and develop a positive allosteric modulator. So the terminology, the PAMs are the molecules which bind to the allosteric site. Here, extracellular region is the orthosteric site, and the small molecules bind the transmembrane domain. So by themselves, they're not active. But when you have a little bit of FSH, they can potentiate this signaling. So these kind of molecules, it would be very useful because they preserve the endogenous natural signaling, as well as they can be selective in the action. So again, just to go back and define this signaling pathway, the FSHR is a GPCR, connected with the GS, and like multiple other growth factors, it stimulates multiple signaling pathway. And so we had used some small molecule and showed that the FSH as well as the small molecule can induce cyclic AMP, the major pathway, as well as AKT, ERK, beta-arrestin, and calcium. And in the end, it induces the aromatase, which converts testosterone to estradiol. So we used the cyclic AMP as well as estradiol as the readout for our screening protocol. And so that helped us to understand the structure of the molecule. So what did we do? So we went through an high throughput screen where we screened about 1.2 million compounds. And so in this high throughput screen, we used CHO cells in expressing the human FSH receptor and stimulated with a low dose of FSH, the EC20, to look for the PAMs. And we screened this many compounds at 10 micromolar in duplicate and look for compounds that produce cyclic AMP at about 40%. And that was the cutoff. So one of the things is, the screening is a number game. So from 1.2 million, from this primary screen, we identified about 2,000 compounds. And these 2,000 compounds, then we did a dose response in a human FSH receptor CHO cells, as well as CHO cells expressing human LH receptor or a TSH receptor and the parental cell line to weed out the compounds that are non-specific and to get the selectivity. So from this, we got about 900 compounds which were confirmed hits. And from the hits, we did some orthosteric assay, looking at the different pathways like beta-arrestin and bioimpedance. And from this secondary assay, we whittled down the compound to about 23. And these 23 compounds then finally went into this functional assay, the primary granulosa cell, and looking at again with the presence of EC20 and looking at estradiol. And so from this series, you can see that from 1.2 million, we come down to 3 distinct chemical series. So this is all the whole this high throughput screening processes to come down to a manageable level that we can do. So the chemist worked on this 3 chemical series and identified. I'll just show you some of the data, early data here. So from this series, you can see that this is in the CHO cells. The compounds, clearly show a very nice dose-dependent increase in the cyclic AMP, and almost as similar to the FSH in the black. And also in the rat granulosa cells, clearly you can see that they behave as PAM, because in the blue line, that is the compound alone, had no effect on estradiol. Whereas, with in presence of EC20 FSH, you get a nice dose-dependent increase in all 3 chemical series. And so this molecules then again was worked with the chemist to develop a more potent compound. And I'm repeating this slide because I just want to show that it's an important slide, because in this drug discovery, having in vivo activity is the final nail for us to show that it can go into this. So because we are using a small molecule, we have to adapt this model system. So we have to change the model system to give FSH a low dose and then treat the animals with a small molecule orally. And here I need to take a deviation and say that this is a model system which was established by one of our alumni from biochemistry, Sriram. He joined our group. He was a graduate student from Professor A.J. Rao's lab. Following that, he did a postdoc with Professor Joint Richards at Baylor's College. Ton of reproductive biology experiment. He did a fantastic job in not only validating this model as well as running this program. Both the granulosa cell and this model system. So as you can see, there was several biochemistry persons who are the critical contributors for this project, and all the reproductive biology training experience really paid for us. And so after all long struggles, we just found one molecule where as you got a good PK, and then we also see that here this is the in vivo study, the high dose FSH and with the low dose FSH, and in presence of the low dose FSH, you can see the nice dose dependent increase with the compound given orally. So clearly, this was a fantastic moment for our group, because it's a long struggle. It was almost like 3 years by the time we got to this stage. So clearly, we could show that in a small molecule given orally, we can induce the follicular development. But there were some issues with this. You need to have very high dose of 100 mg per kg. And so, chemically also, we need to have some, there was some liability.
2 minutes, please.
All right. Okay. So this is the time when Sharono decided to close down the reproductive research, and then we started the Tocopherex. All right. So this is a virtual company. So we work with the external collaborators, and so we developed about 700 new molecules and showed that this is the lead molecule that we had. We had improved the potency in the CHO cells as well as in the granulosa cells. And in granulosa cells, we can see that, both in presence and absence of FSH, our compound stimulated estradiol, both in the rat granulosa cell as well as the human granulosa cell. And again, went to the well-written animal model. And here again, we can show that, the compound induced the follicular development. And you can see that by 10 mg per kg, you can get a good response. And, so then next we ask the question, do we really need this low dose FSH? And clearly, based on this data, we can see that even in absence of FSH, you get a good stimulation of the eggs. And so we have used the top one as our compound, and you can also see the estradiol production. So now we know that orally we can give this molecule, it can produce the eggs. But as Sheshagiri elegantly pointed out, how good are the eggs from this molecule, from this treatment? So we did this. Sheshagiri provided a great review, and so we looked at the expanded blastocyst, as he was talking about the acid blastocyst. And so you can see that these are the compounds given orally, and then looking at the fertilization rate as well as the blastocyst, because it is very similar to the FSH or also the PMSG that were used here. So clearly, we can have a small molecule taken orally, give the good eggs that's been produced by these molecules. So the next part is for the drug discovery aspect, look at the safety tox profile. So we did a toxicology study in rat at multiple dose from 30 mg to 1000 mg, got a nice TK profile. And basically here it shows that we didn't really have any toxic effect in this up to 1000 mg per kg. The only effect we had was the pharmacology, which is the increased cystic follicles, mammary gland hyperplasia, which happens even with the FSH. So we have a good molecule, with good properties, drug-like properties. And this is again looking at all the selectivity and the safety profile. Clearly, everything was good in terms of. So the next step was to get into the IND enabling studies and then get into the phase 1 study. Unfortunately, by this time, we had completed the seed funding that was given by Serono, and we were not able to raise the Series A, so the project was terminated. So to summarize, so I showed you, gave an example of a long-acting FSH that was completed development, and how, however, it was not successful in the market. So these are some of the things that happens in this drug discovery pathway. And also we identified some oral molecules that has got a clean, good bioavailability as well as good pharmacology and clean safety and tox profile. And as I pointed out, due to the lack of Series A investment, we could not continue the development further. So with that, I will thank everyone for the time, and I am open for the questions.
Thank you, Dr. Selvaraj, for such a lucid presentation. We are open for questions, and I think there has been a very general question in the chat box since a long time, so we'll take that first. Sure. It says, sir, I want to ask you that during clinical trials, how do you deal with the side effects of the drug during human trials?
That's a good question. I think, so anytime when you go to the human trials, the first and foremost is the safety of the patient. So before we get into the trial, we have to develop a clinical protocol. And in this, like I said, it's a whole different ball game. So we have all the medicals and the pharmacovigilance and all those people who will develop the protocol, saying that because based on the tox studies, we can identify the target organ toxicity. So based on that, then you say that, okay, we expect toxicity in this organ, this organ, this organ, and if this happens, this is what we are going to do. So that is all part of the clinical protocol. And so that protocol has to be approved by FDA before we can get into the chemo. So that's how, they make sure that the phase 1 study is always done in phase 1 clinic, and where there is a doctor who will monitor the normal volunteers. And in the disease model, again, because it is the clinician treating, the first and foremost is the safety. So if they see any small issues, then they will stop the study.
All right. No, that's himself. So in absence of any other questions, Dr. Selvaraj, once again, thank you for joining us so early in the morning. And I hope you will enjoy the rest of the conference.
Absolutely. It was a pleasure. Thank you for giving the opportunity.
Moving on to the third talk of the day, it will be given by Dr. Surendra Sharma. He is a professor of pediatrics and director of Center of Biomedical Research Excellence at Women and Infants Hospital, Brown University. He is a medical doctor who did his PhD from IIT Delhi, followed by postdoctoral research at Baylor College of Medicine, Houston. He has expertise in reproductive biology and reproductive immunology, with long-term research interests in placental biology, reproductive immunology, and pathogenesis of adverse pregnancy outcomes. His lab works on human and preclinical models and therapeutic options for pregnancy complications. With that brief introduction, I warmly welcome Professor Sharma.
Can you see my slides, please?
We can see you, but not your slides yet.
Oh, slides.
Sure. Perhaps you can unshare and share again.
Okay.
Professor Selva, your hand is still raised.
Yeah, I just saw that, and that was for my previous question to Sheshagiri.
Thank you. Thank you.
Dr. Sharma, we can still not see your screen.
Yeah, I'm getting there.
All right.
Can you see now?
Yes, we can see your slides now.
You need to make it full screen.
How about now?
Yes, yes, very good. Thank you. Please get going.
Okay, so let me first thank the organizers and particularly Professor Sasagiri for inviting me to participate in this session. Both these speakers did a wonderful job, and I hope I can match their intensity. So my topic is our arduous research journey from preeclampsia to Alzheimer's disease. Now, the problem with this title is that I'm mixing young age syndrome with the old age disease. And what we found, what I would like to convince you today is that both these young age disease and the old age disease share the same etiology. And we have gone through a lot of heartaches because people haven't believed into our concept. So I hope I'm able to convince you that yes, what we have been talking about for last 12 or 13 years happens to be true. I'm getting a little, some message in the middle of the screen. What is that happening?
Professor Sharma, I think it's from your side. Just try to click it or just move it away.
I can't move it. Anyway, I don't want to waste my time. So the previous slide, I have to say I have nothing to disclose. So the story starts with this severe pregnancy complication called preeclampsia. It affects 5 to 8% of all pregnant women. In some countries, the incidence is much higher, like in Peru, almost 17 to 18% all pregnant women experience this syndrome, preeclampsia. In India, also incidence is very high, although we don't have national registry there, so we don't really know exactly how many women experience preeclampsia. So let me just, the people who do not think about preeclampsia on daily basis, so let me give you some introductory comments about preeclampsia. It's diagnosed at or after 20 weeks of gestation. There are two forms of it, mid to moderate and severe preeclampsia. And basically these are defined based on the blood pressure readings and proteinuria. So in moderate preeclampsia, we have blood pressure is 140 to 150 over 90. And these are systolic and diastolic values. And these women invariably have proteinuria. They excrete huge amount of proteins in their urine. And fetal growth restriction may also associate these features. Severe preeclampsia, on the other hand, has much higher blood pressure readings, and then also associated with proteinuria. And invariably, this condition is associated with fetal growth restriction. There's another variant of preeclampsia which is called HELP syndrome. Hemolysis, elevated liver enzymes, and low platelets. That's also variant of preeclampsia. Now, the new classification by ACOG, which is the American College of Obstetricians and Gynecologists, has come out with new classification. They define preeclampsia as early onset or late onset. Early onset is a delivery that takes place before 34 weeks, and late onset delivery that takes place after 34 weeks. Now, the issue here is that we do not really have any effective therapy for prevention or treatment of preeclampsia. Delivery is the only way effectively that we can stop preeclampsia, because it's a placenta-associated syndrome. So once placenta is delivered, women get back to normal conditions. But some women still suffer postpartum effects of preeclampsia. If we go into biology of preeclampsia, it's a multifactorial and multi-organ pregnancy syndrome. And in severe condition, it affects number of these organs. And I will go through some of them later on. And particularly something goes wrong at the placenta level. And I have listed a bunch of events that may take place at the maternal fetal interface. We are working on almost all of them, and particularly the pro-inflammatory state, production of microparticles, placental microparticles, ER stress, oxidative stress, and this is the new phenomenon that I will describe in a second. Importantly, what is really a serious discussion about preeclampsia, that there's a condition beyond preeclampsia. Preeclampsia is a pregnancy-specific phenomenon, whereas beyond, these women are much more prone to developing cardiac diseases, diabetes, and now we have published a number of articles suggesting that these women are also prone to developing dementia at much higher rate. So since I'm in a clinical department, we are supposed to be doing translational research, particularly focusing on clinically relevant questions. So we set out almost 15 years ago to start studying preeclampsia, and we had goals, basically take the information from the clinic, take it back to laboratory, and go back to the clinic. So we wanted to really, as I said, preeclampsia is diagnosed at week 20 or after that. So we wanted to have access to early prediction markers, biomarkers, so we could really say that this particular woman will develop preeclampsia. If we could identify these biomarkers at week 11 or 12 or even earlier. There are no mouse models of preeclampsia, so we wanted to develop animal models for preeclampsia because we don't really understand what causes it, and we don't know what pathways we can target for therapeutic intervention. So that's what exactly we set out to do a number of years ago. So we have accomplished a number of these questions that I posed in the last slide. We have established a pregnancy-specific mouse model, which is highly pregnancy-specific, and it recapitulates all the features associated with human condition called preeclampsia. we had to use this strain of mice, IL-10 knockout mouse strain, to accomplish, although we can see features in wild type mice also, but since IL-10 is a pregnancy compatible cytokine, this strain gave much severe features of preeclampsia. And this is a humanized mouse model where we injected serum samples from preeclampsia patients in pregnant mice on day 10. And I'll show you a figure later on, the experimental design that underlies these experiments. So this is a humanized mouse model. When we found out that preeclampsia serum could induce preeclampsia-like features in pregnant mice, we then did proteomic analysis of normal pregnancy serum and preeclampsia serum. And this is what really led to the work that I'm going to describe today. And what we found out that there was an evidence of proteinopathy, which is a protein aggregation in preeclampsia. A number of proteins were dysregulated. And one of them was transthyretin, right here in this paper. And we have identified a number of other proteins that I'll show you data for today. So this was the concept that we uncovered from these studies. So what is the protein misfolding and aggregation? This is exactly what is happening. Hello? So the protein are folded, properly folded in the endoplasmic reticulum. And many times due to, in response to various triggers, these proteins may misfold and go on to form these huge fibril-like structures of aggregated proteins. But in endoplasmic reticulum, there are chaperones that can get hold of these misfolded proteins and refold them properly. So, under overwhelming conditions, this process fails, and then these proteins go on to form these huge aggregates, and these are the ones that cause problems. Now, I'm sure you all know a condition, Alzheimer's disease and many of the neurodegenerative diseases, and this is where this process has been highlighted for a number of years. I'm sure you have heard about amyloid beta plaques in the brain tissue of Alzheimer's patients. At the same time, tau proteins also form tangles. So these are intracellular tangles and these are extracellular plaques. And this is what causes the neuronal cell death. So this has been talked about in the field of neurodegenerative diseases for a number of years, but now we are finding the same etiology, same pathology in preeclampsia patients. So I'll take you through some of the evidence for these studies. So we proposed this thing almost 10 years ago, and this was the paper, one of the articles that we published. However, this concept was shot down by my peers in the field. For a long time, they did not agree with our concept that a young age syndrome can have the same etiology as the old age Alzheimer's disease. But we persisted with that, and this work has gotten some traction. This work was written about in a wonderful article in Washington Post. So now I think it has been accepted, and there have been a number of other articles that have suggested the same thing. And we recently published that we can identify these protein aggregates in nanoparticles, nanovesicles from preeclampsia placenta. So the exosome-like particles have these aggregated proteins as part of their cargo. And so we can identify these aggregated proteins in exosome-like particles. So the current focus is on the prediction. I told you that preeclampsia is diagnosed only at week 20 or after that. So we would like to have a predictive assay that we can use at week 11 or 12. And then also we would like to use some of the pathways that we have uncovered for therapeutic interventions. So let me just take you through some of the recent data that some published and unpublished data that we have come up with. So why are we interested in predictive assay? There are no well-defined rapid blood tests available because once diagnosis takes place, the game is over. We can't really do much except clinically manage these patients, give them aspirin and a few other things, magnesium sulfate to control hypertension. And so this is one reason we would like to have a predictive assay. We would also like to understand cellular mechanisms that underlie the onset of this pregnancy complications, so we can target them for therapeutic interventions. And so I'll take you through some of the strategies that we have used to understand these issues. So at this point, 3 individuals, Dr. Sevin Chang, Sayani Banerjee, and Nakashima joined the lab, and they pursued a concept that we have published a lot in the lab. I'm just listing here 3 articles. And this is the concept of autophagy. This is a cellular mechanism required for cellular homeostasis and for clearing the damaged organelles and aggregated proteins from the cells. And I don't have time to take you through. It's all published information. You can check in this major article on autophagy that preeclampsia is associated with impaired autophagy. So this concept turned out to be very useful for us. And this is what we have followed or pursued to develop a predictive assay. And so this is the first prediction. Thematic strategy includes this concept that autophagy deficiency in the placenta allows accumulation of protein aggregates. So Sebin, Sayani, and Akitoshi have done this work where they showed that the preeclampsia placenta do not express some of these lysosomal biogenesis machinery proteins. This is LAMP1, lysosome associated membrane protein 1, LAMP2, cathepsin D, that's the protease that is present in the lysosome and that degrades all these proteins. And there's another protein called P62 that should be degraded if autophagy is activated. So what you see here, LAMP1, LAMP2, cathepsin D are poorly expressed in preeclampsia placenta. These are placental tissues. And whereas P62 is accumulated in preeclampsia placenta, suggesting that autophagy is impaired. And this is a quantification in number of placental tissues that we have done. So that's what the concept people in the lab used. And we just published a very popular article that just came out in Nature Scientific Reports, a novel blood test for early biomarkers of preeclampsia and Alzheimer's disease using the autophagy as the conceptual basis. So let me just take you through the data from this article. So this is the strategy that we used where we established a trophoblast cell line, which is autophagy deficient, by stably transfecting trophoblast cells with this mutant autophagy gene. And what it does, it basically inhibits the formation of autophagosomes or phagosomes early on because it inhibits the conversion from LC31 to LC32. And then we expose these cells to serum from patients with the hope that these protein aggregates present in the serum would not be cleared by the cells because they are autophagy deficient. Then we can come back with a fluorescence dye, which is proteostat dye. And this dye has a unique property to bind to aggregated proteins. So we can then detect the signal. And this is the validation of the process where we have taken native transthyretin, which is not aggregated. And then we have aggregated this protein in vitro. This protein has a high beta seed content and has propensity to undergo aggregation. So you can detect easily the aggregated protein when we use it in these cells. But this is a pure protein. It does not guarantee that we can detect these proteins in serum when we take serum samples from patients. So this next slide shows the serum samples from early onset. Remember, I told you that preeclampsia are now classified into 2, early and late onset preeclampsia. So you can see we have taken a huge number of samples, as you can see on right-hand side. And we have done kinetic exposure of these autophagy deficient cells with serum samples. And you can see from early onset, which is the severe form of preeclampsia, we start detecting the proteostat signal by 3 hours of incubation. And then this signal becomes robust after 6 and 24 hours, whereas in the late onset, which is the mild form, and you can see that we can still see signal, very robust signal by 24 hours. And these are the ROC curves, and the association is very strong for the assay. So now question is, what proteins are present in these complexes? So we have already identified transthyretin. This is a transthyretin protein. And this is amyloid beta, which is, I told you the amyloid beta is specific to Alzheimer's disease. And you can see in serum samples from these preeclampsia patients, we can detect TTR, transthyretin, and amyloid beta. And what is so important about that, the signal co-localized with proteostat signal, suggesting that these proteins are present in the form of protein aggregates. So that's what is really important, and we can detect A beta, just like Alzheimer's disease. This protein is present in preeclampsia serum samples in the form of protein aggregates. I'm not showing you the data on the placenta, but we can detect the same proteins in the aggregated form in the preeclampsia placenta. And these are quantification and ROC curves here. And this just simply shows that when we did non-denaturing gel, we can detect these high molecular weight aggregates only in the preeclampsia serum here and here. This is for transthyretin and A beta, and actin is just the protein load. But that simply suggests that a huge amount of protein aggregates are present in serum samples from preeclampsia patients. So question is the predictive value of this assay. So we are collaborating with a group in Sweden, from Linköping, Sweden, and our own hospital, and we collect serum samples from all pregnant women at week 11 to 13. And our colleagues in Sweden have followed these patients, these pregnant women, for a long time, and have now data on number of women who develop preeclampsia and who go on to have normal pregnancy. So we have shared these serum samples with them, and you can see, and we have almost 70 serum samples from preeclampsia group and normal pregnancy group, and these were given to us in a blinded form, so we really did not know what is what in the beginning. Now they have decoded the samples for us because we have analyzed all the samples. And at this stage, we analyzed 21 samples from preeclampsia group and 19 samples from normal pregnancy group. And 11 to 13 weeks itself, we can identify proteostat specific signals suggesting that there are aggregated proteins present in serum samples, even at 11 to 13 weeks. So these protein aggregates are present at much earlier stage than the diagnostic time for preeclampsia. And whereas the normal pregnancy serum samples do not show us any proteostat signal. And by the way, I don't have time, but these protein aggregates already contain transthyretin and tau proteins that we can detect by immunostaining with the specific antibodies for those proteins. So the question is, since I've been talking about the Alzheimer's disease and preeclampsia share the same etiology, can we use the same assay to predict Alzheimer's disease? There's a condition called mild cognitive impairment, which is pre-AD stage. And so we have collaborated with our colleagues at a neighboring hospital, Rhode Island Hospital. And Dr. Art is the director of the Alzheimer's Disease and Memory Clinic. And they have given us huge number of serum samples and a CSF sample from these patients. And so we use this assay to see whether we could use this assay to detect these protein aggregates, because right now there's no blood test available for Alzheimer's disease. Only thing that we do in the clinic is the PET scan, brain PET scan, or CSF biochemistry to detect tau proteins or amyloid, but those are very expensive procedures and time-consuming procedures. So not everybody can afford to have those assays done in the clinic. So this assay would turn out to be so useful if it is approved, by clinicians. And that's what we are trying to do. We are trying to collect a huge number of samples, because in the clinic, you can't convince clinicians just by having 10 or 20 samples. You have to go through hundreds of samples and validate your assay. So this is what we have done. This is the beginning of the project. So here you can see these are samples from, yes, these are samples from Alzheimer's patients, and these are from mild cognitive impairment, which is the pre-AD stage. And you can see amyloid beta, transthyretin, alpha-synuclein, which is mainly associated with Parkinson's, but we can detect in Alzheimer's patients. You can see we can detect all these proteins in serum samples using our assay. And the signal co-localized with proteostats, suggesting that these proteins are present in the form of protein aggregates. So this is really soothing to us. My colleagues in the Alzheimer's clinic are very excited, and they have already started using. And these are the quantification of the data and ROC curves, which suggest that there's a very strong association with the positive information from this assay. So again, I talked to you about tau proteins. I have only showed you the evidence for amyloid beta, transthyretin, and alpha-synuclein. Is tau protein also present in preeclampsia serum? And this is when Dr. Sukanta just joined the lab, and he wanted to study this issue, this question, whether preeclampsia is also associated with tau protein, just like the Alzheimer's disease. And we are collaborating with our colleagues from Harvard Medical School, Dr. Liu and Dr. Zhao. They have worked, published a huge number of articles on this protein called this phosphorylated tau protein, which is associated with AD patients, Alzheimer's patients, and the patients with traumatic brain injury. And one of the articles that they published in Nature Communication, they have published huge number of articles in Nature and Cell, but one of the articles that they can use this protein, antibody against this protein, identify tau phosphorylation in football players, boxers, and accident trauma patients, so that they can use the specific antibodies against this cis-p-tau. So cis-p-tau, and normally we all have trans-p-tau, which is a physiological tau protein, and it's involved in microtubule assembly. So you can just read it. I'm not going to read the whole thing here, just simply to tell you that trans-p-tau is good protein. It's involved in healthy neurons, promotes microtubule assembly, is susceptible to phosphatase, so it can be dephosphorylated, and it can be degraded, and it has no propensity to aggregate, whereas the cis form of this protein cannot promote mitochondrial assembly, and again, it is prone to aggregation. It causes tauopathy and neurodegeneration in AD. So my colleagues have published a number of articles on this protein. So when Sukanta looked in preeclampsia placenta, this is a control normal pregnancy placenta. This is a preeclampsia placenta, you can see, and this is the trophoblast layer of the placenta. You can see huge staining for cis-p-tau in the preeclampsia placenta. We don't see much in the control pregnancy placenta. And what is important, that there's a huge proteostat signal in the placenta, and the both signals co-localize, and this is the amplified picture of this inset here. You can see a very nice co-localization of cis-p-tau and proteostat, whereas we do not see anything for trans-p-tau. Trans-p-tau does not localize with proteostat signal. There's in preeclampsia placenta, there's no trans-p-tau, whereas trans-p-tau is expressed in normal pregnancy placenta. And we
Professor Sharma, you have about a couple of minutes.
Okay, I'll just try to finish it. So we can even induce it with hypoxia in primary human trophoblast. So this is the humanized mouse model. The question is, cis-p-tau is really important for preeclampsia pathology. Sukanta did this depleted cis-p-tau from serum and injected. An important thing is that preeclampsia serum induces elevated the proteinuria, elevated blood pressure, and there's a fetal growth restriction. whereas depletion of cis-PTR can normalize all these features and bring it back to normal pregnancy. And this is evidence for glomerular endotheliosis. In response to preeclampsia serum, you have a complete disruption of glomeruli here, whereas cis-PTR depletion can again restore the structure of glomerular units in the kidney. So kidney pathology is a major feature of preeclampsia, and that's why we see huge proteinuria in preeclampsia patients. So therapeutic interventions, we screened a huge number of our reagents to see, but we wanted to target autophagy and protein aggregation. And this is the molecule that we came up with, trehalose. And I call it sweet relief for preeclampsia and Alzheimer's disease. This is a disaccharide, non-mammalian disaccharide. And what in our results, what we found that trehalose can restore autophagy and inhibit protein aggregation in human trophoblasts. So we used it, and this is a mouse experiment again. And you can see here, no matter how we gave trehalose, preeclampsia serum elevates blood pressure, proteinuria, fetal growth restriction, whereas trehalose, given in any scenario, can normalize these readings and can also repair glomerular endotheliosis, whereas maltose, that's another sucrose, does not do that. So it's a very specific that it can inhibit preeclampsia and inhibit all the features associated with preeclampsia. So this is the conclusion side. Basically, I have talked about predictive assay. I have talked about therapeutic interventions, and this is important. Selva was talking about how you go through the clinical trial. So we have applied for IND. I have joined with a biopharma company in Illinois. They have the exclusive rights for trehalose. So they are helping us with initiating these clinical trials. And let me just go through that. So this is a very important slide. All the people who did the work here are listed, and our collaborators, national and international collaborators. And of course, the work won't be possible without the support from funding agencies. So thank you very much.
Thank you so much, Professor Sharma, for this perceptive talk. We have a question in the chat box. It says, sir, I want to ask you, is there any benefit of detecting Alzheimer in pre-Alzheimer stage? Will this reduce the disease pathology?
So, well, detecting protein aggregates would not reduce the pathology. What will reduce the pathology if we can, like I'm talking about trehalose. So the initial evidence in serum samples and in this neuronal cells that we have taken, trehalose works like a charm, but working in a test tube is not the same thing working in humans. So as I said, we are initiating these clinical trials. So trehalose might help because autophagy deficiency and protein aggregations are two major pathologies associated with Alzheimer's disease or any neurodegenerative disease. So we are hoping for the best.
May I ask a question?
Yeah, go ahead, Dr. Silvaraj.
This is Subhash from NCL Pune. I have two quick questions to Professor Sharma. The first one would be, how would be the status of post-pregnancy? Have you tested amyloid beta or alpha-synuclein?
Can you just repeat it again and go slow, please?
All right. Yeah. Can you able to hear me out?
Yes, I can.
I can hear you. Go ahead.
All right. Yeah. The first question: have you also tested post-pregnancy during this, amyloid beta or alpha-synuclein?
So you mean first pregnancy means very first pregnancy when they develop preeclampsia? Or what,
Yes.
what do you mean?
Yes.
Okay. Okay. So this is a very interesting question. Yes. So the 11 to 13 week samples that I showed you, they are from first pregnancy pregnant women. And what happens with preeclampsia and second or third pregnancy, preeclampsia incidence is not that prevalent because now we know if if a pregnancy takes place with the same partner and the duration between two pregnancies is not very long, then preeclampsia does not come back because there's a paternal education of endometrium. So the second pregnancy, third pregnancies are not as severe with respect to preeclampsia as the first pregnancy. So all these samples that I'm showing you today are from first pregnancy.
The second question, may I? Or do you have maybe half a minute?
Sure.
Yeah, I think this will be the last one because we are running
All right.
out of time.
Yeah.
So.
Okay. So what is the specific idea to look at PIN1 as far as the tau is concerned? Why can't, or are you also interested to look at GSK3 beta or other, key kinases which is responsible for tau phosphorylation?
Yes, I think my colleagues, my collaborators from Harvard have already looked at them. And PIN1 is very specific for tau isomerization. So the PIN1, if PIN1 is activated, it's induced, then it will keep tau protein in trans isoform, whereas if PIN is inactivated by oxidation or phosphorylation or by point mutations, then the tau will be transformed into a cis-p tau. So the PIN1 is the only one that does that thing. Other kinases, I haven't done it, but my colleagues tell me that other kinases don't do that. And so PIN1 is very specific for this purpose.
All right, thank you.
All right, I can see a question in the chat box and Dr. Selvaraj's hand, but I'm afraid we'll have to stop here because we are running late. So thank you once again, Dr. Sharma, for joining so early in the morning. And I thank all the three speakers for giving wonderful talks. I pass it on back to Big's team.
Thank you. Silva, you can send me your question by email.
Yes, I will do that.
My personal thanks to
questions.
Selva and Sharma.
So thanks to all the speakers for their wonderful talks and Dr. Rajaguru for chairing this session. Now I request Satyaranjan to take over from here and proceed for student talks.
Thank you, Sweta. In the second part of this session 3, we will have 6 upcoming brief talks by alumni and students of this department. This part will be chaired by Dr. Mahipal Ganji. Dr. Mahipal Ganji is an assistant professor in this department who joined in 2020. His multidisciplinary research group is interested in understanding the interactions that help in 3D genome organization. Now I would like to request Dr. Mahipal Ganji to start the session by introducing the speakers.
Thanks, Satya. So our next speaker is Professor Swati Saha from Delhi University. She was a PhD student in Professor D.N. Rao's lab. She graduated in 1997 from the department with Giri Memorial Award, actually. So she is now Dean of the Department Interdisciplinary and Applied Sciences in University of Delhi. She's going to deliver a talk on investigating epigenetic regulatory mechanisms in Leishmania de novo.
Are my slides visible? And am I audible?
Yes, yeah. You have to make it full screen, Swati.
Okay. All right. Okay, so good evening, everybody. It gives me great pleasure to congratulate my alma mater on completing 100 remarkable years of pioneering research, and of course, wishing the department at least another century of the very same. So my lab is at the University of Delhi South Campus at the microbiology department, and we basically look at the DNA biology of Leishmania parasites. So I'd like to thank the organizers for giving me the opportunity to tell you all a bit about the epigenetic regulatory mechanisms in Leishmania donovani. So Leishmaniasis are a group of diseases manifested in 3 forms, and in India, the prevalent form is visceral Leishmaniasis, popularly called Kala Azar, and being caused by Leishmania donovani. The disease is fatal if not treated in timely manner, There are drugs to treat the disease, but due to emerging drug resistance as well as the toxic side effects of existing regimes, the search for new drug targets continues. And thus, there are several groups around the world who are actively involved in studying the basic biology of these parasites. So our lab has been interested in looking at two aspects, DNA replication as well as chromatin modifications. And today I'll be telling you about some of our findings from the latter. So histone post-translational modifications, as you all know, occurring both on the tails as well as the globular domain, impact cellular processes either by altering chromatin structure or by modulating the binding of effector proteins. So these PTMs, as well as the functional roles of specific PTMs, are largely conserved in eukaryotes, going from yeast to mammalian cells. But because Leishmania are highly divergent eukaryotes, the histone sequences they carry are quite different. And to date, the landscape of histone post-translational modifications in Leishmania parasites remains unknown. So we have primarily so far been looking at histone acetylation events in these parasites. And early studies from our lab found that of these 4 mist family hats which were identified by genome annotation, 3 were nuclear while 1 was cytosolic. 3 of these targeted the same histone, albeit at different residues. And we were able to find that 2 of these actually targeted specific residues in vivo as well. So taking the studies forward to address the functional relevance of these specific modifications, we adopted the route of creating genomic knockouts since siRNA the RNAi machinery is absent in these parasites. So looking at the role of the HAT3 protein, we found that this was not essential to the cells because we could create nulls, played no role in transcriptional regulation based on our microarray analysis, no impactful role in DNA replication either, based on our cell cycle and EDU labeling studies, but did modulate the cell's response to UV-induced DNA damage, where we found that at doses where wild-type cells could recover, the mutant cells could not. So we then started digging further into the why of this, and primarily addressing the H4K4 acetylation event. But no matter which way we looked at it, nothing we found could account for this very dramatic effect which we were seeing. So we turned our attention now to non-histone angles as well. Now, as you all know, UV will cause the formation of these pyrimidine adducts in the DNA, which causes the stalling of replication forks. And if the cell does not rectify the problem, then these forks could collapse and the cell would die. So one of the ways the cell gets around this is by switching the replicative DNA polymerase with the translesion DNA polymerase, which has a wider pocket and can accommodate this pyrimidine dimer, thus allowing the cell, polymerase to synthesize DNA past the lesion, albeit this is an error-prone process. So we looked at these angles through various experiments, and the bottom line is that we found that while in wild-type cells, in response to UV, PCNA, so what I forgot to mention is that early studies in yeast actually found that the monoubiquitination of PCNA is essential for stabilizing the TLS polymerase on the DNA template. So what we found was that while in the wild-type cells, in response to UV, PCNA was getting monoubiquitinated, this was not happening in mutant cells. And we showed this by 2 different sets of experiments. So PCNA monoubiquitination in response to UV is abrogated upon HAT3 depletion. But then the obvious question is how and why? Now, around the same time as we were doing these studies, an Italian group showed that actually the human P300 can acetylate PCNA in vivo in response to UV. So we latched on to that and started a series of experiments to examine those angles. And the short of it is that we found that in response to UV, HAT3 mediates PCNA acetylation. So you see that in the nulls, that is not detected. So we concluded from all of these sets of experiments that HAT3 mediated acetylation of PCNA is a prerequisite for the monoubiquitination to occur, which in turn is probably playing the role in the TLS-based DNA synthesis. And that was why the cells could not recover in the case of HAT3 depleted cells after UV treatment. Now, when we looked at the role of HAT2, the story was quite different because this turned out to be an essential protein. And so all our characterizations were done with the heterozygous knockout cells, where we found that the heterozygous knockout cells showed very compromised growth and cell cycle patterns. And again, our experiments led us to find out that Psych4 and Psych9 downregulation in these HAT2 depleted cells was probably the reason why we were seeing these aberrant patterns. Now, if you look at the genome organization of Leishmania, it's very unique in that not just Leishmania, all trypanosomatids, in that functionally non-related genes are arranged in these long unidirectional arrays, which are coordinately transcribed from these divergent strand switch or HT regions that lie between the gene clusters. So these polycistronic transcripts are then co-transcriptionally processed by transplicing events and then translated. No conserved sequence motifs have been identifiable at these DSSR and HT sites, and transcription is constitutive and, as I said already, polycistronic. And at the time when we did the study, no histone modification marks had been identified yet at these sites. So based on the fact that Psych4 and Psych9 were getting downregulated in HAT3 depleted cells, we conjectured that probably H4K10 acetylation was regulating transcriptional events in the cell. And so the first step was to examine these DSSR and HT sites for that modification mark, which we did using chip assays with our H4 acetylcatein specific antibody, and found in fact that yes, these sites were enriched in H4K10 acetylation. And these the extents of these acetylation events were downregulated upon HAT2 depletion. So if reduced H4K10 acetylation at the DSSRs was responsible for downregulating cyclin expression, then it was logical to conclude that the expression of the entire gene cluster must be coordinately downregulated, since transcription is occurring coordinately. But when we carried out microarray analysis, we found in fact this was not so. And while the vast majority of genes genome-wide were not getting affected by lowered levels of H4K10 acetylation, there were about 200 odd genes whose expression levels were getting downregulated upon HAT2 depletion. So this led us to thinking about there being internal promoters lying within these long clusters. And to test this, we used two kinds of experiments. First, we linked these upstream regions to a reporter gene to check if they could drive transcription, and found in fact that the Psych4 and Psych9 upstream regions could drive transcription as compared to the upstream regions of these other genes which could not. And second of all, we carried out nuclear RNA transcription assays to look at ongoing transcription as microarrays look at processed transcripts. And so from these two sets of experiments, I'm not going to the details of them, but we could conclude that yes, there are internal promoters lying within these long clusters. So how widespread is this phenomenon?
Swati, 1 more minute.
Oh, okay. So we have now taken this study chromosome-wide with 2 chromosomes. And again, without going into the details, we find using nuclear run-on assays across these 2 chromosomes that most of the genes are being polycistronically transcribed, with small subsets being transcribed only at specific cell cycle stages. And so when we looked at their upstream regions, okay, I'll skip all that. So basically what we've concluded is that there are 2 stages of transcriptional activation in these parasites. The primary tier of transcription is from the DSSRs, which is constitutive and which is not affected by slightly lowered levels of H4K10 acetylation. And the second tier of transcriptional activation is occurring from these promoters lying internally within these clusters. And this second tier of transcription is being activated in a cell cycle stage-specific manner and also is more sensitive to H4K10 acetylation events. And these are some of the other projects in the lab. I should end with thanking my team of students who carried out all this work. So they won and initiated all the studies. and also took forward the HAT3 study, Udita the HAT2 study, Aarti the HAT4 study. Arushi is taking forward our study with the ELP3A protein, which we find mediates the cell's response to chronic stress. Jyoti and Varshini are working with the set domain proteins, which we find modulate the cell's response to oxidative stress. Then Ashish and Vishal are working on the transcription project. And finally, so it all started 30 years ago in 1991 when I joined GN's lab for my PhD. And as I'm fond of telling anybody who's willing to listen, I knew Zilch when I got to his lab, but I had very patient seniors who taught me basics and a very patient boss who gave me the time and space to learn the drill. And so from there to where I am today, it's been a long journey, but I'll always be grateful for the opportunity I had to do my PhD at BC under the supervision of DN. Thank you for your time and attention, and I'll be happy to take any questions you may have.
If there are any questions from audience, we're actually running short of time. Okay, Deepayan, go ahead.
Hi, ma'am. A great talk. I just had one question. How does the levels of the acetylation of the histone 4 change across the different stages of the parasitic infection? Like when is it in the sandfly versus when it is in humans? How does it change?
Yes, and nobody has looked at it yet because so all our work has been in the sandfly stage really. Nobody has actually looked at the acetylation levels in the intrahuman stage, in the macrophageal stage. So we don't know the answer to that. Am I audible? Sorry.
Yeah, ma'am. Thank you.
Yeah, thank you, Swati, for a great talk. Okay, next we will move on to the next speaker. It's Dr. Trishna Dey from Professor Rangarajan's lab. She recently graduated from Professor Rangarajan's lab, and she's working on a very exciting study in Pichia pastoris, and she developed a method which is a methanol-free glutamate-inducible yeast expression system. Trishna, are you there?
Yes, I'm here. Can you hear me?
Yeah.
Okay.
Looking forward to your talk. Thanks.
Okay. Are my slides visible to everyone?
Yes, yes. You just have to make it full screen.
Yeah, I'll do that. So I hope I'm audible and the slide is visible on full screen.
Yes, go ahead.
Okay. So first of all, thank you for the introduction and the opportunity. So today I'll be talking about my work that I have been carrying out in the laboratory of Professor P.N. Rangarajan post my thesis submission, which is about development of a novel methanol-free glutamate-inducible expression system. So our lab has been working on the methylotropic yeast Pichia pastoris, which is not only an academically important model organism, but is also an industrially relevant yeast. It can utilize vast variety of carbon compounds as the sole source of carbon, which include not only glucose and glycerol, but also C2 carbon compounds like acetate and ethanol, C1 carbon compound like methanol, fatty acids like oleic acid, and most uniquely, amino acids. By and large, amino acids are utilized by yeasts like Saccharomyces cerevisiae as a nitrogen source. But Pichia pastoris not only utilizes amino acids as a nitrogen source, but unlike Saccharomyces, it can utilize amino acids as a sole source of carbon as well. Our lab focuses on understanding the transcriptional regulation of carbon metabolism. And in the process, we have identified several transcriptional regulators like MXR1, TRM1, and ROP1. While MXR1 and TRM1 are transcriptional activators, ROP1 is a transcriptional repressor. My research focus was to understand the post-transcriptional regulation of carbon metabolism. And in the process, we identified several post-transcriptional regulators of glutamate metabolism, which includes a cytosolic protein called RTG1 and metabolic intermediates of the glutamate utilization pathway. So we have shown that in Saccharomyces cerevisiae, RTG1P functions as a transcription factor along with another protein called RTG3P. In Pichia, we have shown that RTG3P is absent. Hence, in the absence of RTG3P, RTG1P functions as a cytosolic regulator of carbon metabolism. It functions as a post-transcriptional regulator. So we have shown that it regulates the translation of two key enzymes of glutamate utilization pathway, which are glutamate dehydrogenase 2 and phosphoenol pyruvate carboxylase. We have shown that the RTG1 mediated regulation happens via the 5' UTR. That is, the cis regulatory element is present within the 5' UTR of these mRNAs. We have also shown a unique crosstalk between GDH2 and PepsiK protein during utilization of glutamate as the sole source of carbon. We have shown that translation of GDH2 and PepsiK during glutamate utilization is also facilitated by the respective substrates, which is glutamate and oxaloacetate respectively. Glutamate and oxaloacetate not only facilitates the translation of GDH2 and PepsiK from the respective mRNA when they are added exogenously, we have shown that they also, the intracellularly generated intermediates, that is glutamate and oxaloacetate, also facilitate this entire process. And we have shown this to be, and this usually happens during extreme conditions of nutrient deprivation like carbon starvation. And we have shown it to be an autophagy or autophagy related process. So all this work led us to hypothesize whether the 5' UTRs of GDH2 and PepsiK can function as riboregulators. While my colleague Neetu is focusing on understanding this aspect of the project, I went ahead to develop a novel methanol-free glutamate inducible expression system. As I already mentioned that Pichia pastoris is an industrially relevant yeast. So why is Pichia a popular choice? So Pichia pastoris offers simple, fast, and very cost-effective platform for recombinant protein production. It being a respiratory yeast, it can grow to very high cell densities, and which ultimately yield in very high protein levels per volume of the culture. It has several advantages of eukaryotic expression systems such as protein processing, protein folding, and post-translational modification. It is very easy to genetically manipulate, and above everything, it also has the added advantage of secreting the highly expressed protein into the medium, which makes the downstream processing very easy. Now, what is this platform that I'm talking about for that, which is already well established in Pichia pastoris? So it is the methanol inducible expression system, which utilizes the strength of the promoter of the gene encoding alcohol oxidase 1 protein. So this system has a lot of advantages, like it is a strictly regulated inducible system. It is very cost effective, and it has very high expression levels, which ultimately result in high yield of protein. Despite these advantages, this system also comes with some disadvantages. Here the inducer is methanol, which is toxic, flammable, and it requires special handling. The metabolism of methanol also results in the generation of toxic byproducts like peroxidases and peroxide and formaldehyde, which often result in the generation of biologically inactive protein. So there is a lot of research going on for the generation of methanol-free Pichia pastoris expression system. So during the course of my PhD, we stumbled upon one interesting finding where when we were trying to understand the expression profile of GDH2 and PepsiK across different carbon sources, we found that GDH2 is specifically induced when it is cultured in glutamate-containing medium, while PepsiK is not only induced when it is cultured in glutamate-containing medium, but it is also induced in ethanol and acetate-containing medium, but the expression was manifold higher in glutamate condition. Interestingly, when we went ahead to analyze the RNA profile For GDH2, it was very surprising. Although the protein was found only when it is cultured in glutamate-containing medium, RNA was found to be present in all the carbon sources, even though the protein was not seen. For PepsiK, as expected, it was found to be under catabolite repression in dextrose-containing medium, but in other non-fermentable carbon sources, RNA was present, but at different bedding levels. To further confirm this result, we resorted to reporter assay, and we can see clearly that when GFP is expressed from GDH2 promoter and PepsiK promoter, its expression profile is just like the endogenous GDH2 and PepsiK protein. And there was a huge correlation with respect to the GFP mRNA profile as well. So this gave us the idea that GDH2 and PepsiK proteins are synthesized in a carbon source specific manner. And based on this premise, we went ahead to develop glutamate inducible yeast expression system, wherein we would like to express recombinant proteins under the under GDH2 and PepsiK promoter. So we wanted to shift to a inducer which is readily available and is cost effective. So we resorted to glutamate, monosodium glutamate, which is a sodium salt of glutamate and is known as MSG. It is also very famous under the trade name of Ajinomoto, which is used as a flavoring agent in many Asian cuisines. This compound is US FDA approved. It's a well-known food additive. It is readily soluble in water, easily available, inexpensive, and toxic. So our next goal was to see whether it can induce the expression of GFP. Yeah? Okay. Our next goal was to see whether this compound can induce the expression of GFP from GDH2 and PepsiK promoters as good as glutamate or not. And it is very evident that it is an efficient inducer of GDH2 and PepsiK promoter. And to further get a clear picture with respect to the amount or the quantity with which the induction has happened compared to the already established AOX system, we resorted to Western blotting. And here we can see that expression of GFP from GDH2 as compared to the well-known methanol-based AOX assay system was approximately 60%, and that of PepsiK GFP was 85%. To further get a clear idea about
Krishna, you have a minute to go.
Okay, so to further get clear idea about the yields of the protein, we resorted to nanobody mediated GFP pull down, where we cultured cells in YPD medium overnight so that we can generate biomass, transferred them to the YNBM and YNB MSG containing medium and induced for 24 hours, followed by pull down. So we can here appreciate that when we harvested 20 OD cells, we were able to obtain 14.4 microgram of GFP from AOX promoter, 18.3 from GDH2 promoter, and 12.6 from PepsiK promoter. And this correlated well with our Western blot finding. Now we can clearly appreciate that the strength of PepsiK promoter is way more than that of GDH2. So in order to increase the efficiency of our system, we resorted to a strategy where we expressed GFP from 2 PepsiK promoters cloned in 2 different vectors and then co-transformed in the same strain. And here we can see that upon co-transformation, we were able to increase the yield of GFP compared to AOX1 promoter by 1.24-fold. We do understand the limitation with working a GFP protein per se, because it is inherently very stable, and the protein expression varies from protein to protein. The level of expression would vary from protein to protein per se. So we resorted to use an extracellular protein. In this case, it was SARS-CoV-2 receptor binding domain, and an intracellular example, which is the human actin. And we can see that from a 20 ml culture, we were able to induce the expression of SARS-CoV-RBD at a level of approximately 75% with that of AOX promoter. And similarly, from 50 OD cells for actin, we were able to induce the expression of actin for PepsiK promoter at a level of approximately 58%. So just to recapitulate, single transformations resulted in the yield of protein percentage-wise from 58 to 87% with respect to AOX, and co-transformation, we were able to increase the yield and efficiency of the system. As of
Krishna,
now,
can you please summarize fast because you're already over the time.
okay. So we are working on with other examples as well. And here we can see that when we co-transform SARS-CoV-RBD as 2 copies, we were able to induce the expression. And here it is almost, we can see that it is almost 1.75 times more. Right now, we are also in the process of making versatile glutamate inducible vectors, which with various combinations of recombinant combinations of markers and either GDH2 or PepsiK promoters, of which 4 will be the for intracellular expression and 4 are going to be for extracellular expression. So with this preliminary and encouraging data, we have filed patent, and a preprint is also available for further reference and details. With this, I would like to end my talk. Thank you.
Thanks, Trishna, for the great talk. So are there any questions? Okay, since we are running out of time, so thanks, Trishna, for the wonderful talk.
Thank you.
Yeah, so Deb.
Yeah. Thanks, Krishna. So now this time to speak for Dr. Amrita Bhosle. She is graduated from Professor Nagasuma Chandra Lab, and she is now working as a postdoctoral fellow at Broad Institute of MIT and Harvard. So look forward to your talk, Amrita.
Yep. Thank you so much for the kind introduction. And I would like to start off by saying that it is my privilege to come back and speak to this audience, and I'm very grateful for the opportunity. So I'll dive right into my material for today. So the interactions between human health, microbes, and human activities such as drug use or diet can have several outcomes. And these outcomes can be good or bad, like what I have basically said in my title, depending on the kind of microbes that we harbor, whether they are beneficial or disease-causing, or the outcome can be downright ugly, such as antibiotic resistance, which is now a global health concern. And in conversations about public health and the role of microbes, we often ask questions such as which microbes are good for us, which metabolites that they produce are good or bad for us, and how to tackle problems such as antibiotic resistance. However, answering these questions is not easy. Because these involve the study of complex phenotypes, complex communities such as the gut microbiome, and complex interactions such as those between microbes, the immune system, diet components, and so on. However, what can really help us study this complexity is omics data and smart computational methods. So what I'm going to talk about today is one of my PhD projects where I've applied transcriptomics to study resistance mechanisms in E. coli and identify a strategic target. This was published in iScience. And the next thing I'm going to talk about is just give a brief overview of what I'm involved in my current lab. So to start off with the first story, which is studying the resistance mechanisms in E. coli. So what we did was first generate trimethoprim resistant E. coli from trimethoprim sensitive wild type MG1655. What is trimethoprim? It is a competitive inhibitor of dihydrofolate reductase, and it basically acts by inhibiting the production of DNA. So the way we did this was expose the sensitive E. coli to concentrations below MIC and then go on increasing the dose of trimethoprim 2 times in every step until we had a culture that was resistant to 32 times the original MIC of E. coli. And we called this the 32XR E. coli. And what we did next was compare the whole genome sequences and the transcriptomes of the sensitive E. coli and the resistant E. coli that we had developed. The very first thing to check was whether the resistant strains harbored any drug-resistant mutations. And we identified 2 mutations that basically result in the upregulation or the overproduction of a resistant DHFR. And I say resistant because the mutation that we identified was the L28R mutation in the binding site, which reduces the affinity of DHFR for trimethoprim. And the second one was a promoter of mutation, which actually overproduces this DHFR. When we compared the transcriptomes of these 2 strains, we identified several differentially expressed genes. There were 190 genes that were upregulated and 155 that were downregulated. And we first carried out an enrichment analysis to just see what pathways are perturbed in the resistant strains as compared to the sensitive E. coli. And we found that there were multiple biological processes that were actually perturbed. And among the upregulated ones, we found SOS response and biofilm formation and response to pH. SOS response is something that is expected when cells are exposed to DNA damaging agents. And in the downregulated ones, we found chemotaxis and flagellar proteins and so on. So this was a lot of information to intake, and what we wanted was a systematic way to analyze this. So what we do in our lab usually is integrate transcriptome changes into genome-wide knowledge-based interaction networks. So what we did was constructed an E. coli genome scale interaction network. This is knowledge-based, which means it is just going to put together interactions that are previously studied, such as those between a transcription factor and gene, direct protein binding interactions, co-expression profiles, gene neighborhood, and so on. And then what we do is use this network in which we have integrated the fold change data and look at paths that are associated with significant perturbation. So what we were actually hoping to find was like different clusters of biological processes that are perturbed. But what we instead found was this whole connected graph where we basically saw that the perturbed processes were involved in a crosstalk with each other and were not acting in isolation. So among the resistant mechanisms, we found things such as the SOS response, acid stress response, biofilm formation, and folate pathway that was significantly perturbed, and that there was a crosstalk between all these response mechanisms. Now, to find a strategic target in trimethoprim resistant E. coli, we looked at this graph in great detail and found that SHMT or GLI-A, which lies downstream of DHFR, which is FOL-A, was upregulated in the trimethoprim resistant E. coli. And we saw that it also interacted with transcription factors involved in biofilm formation. So that was really interesting. So then we went and looked at the perturbation profiles of enzymes in the folate pathway. And to give everyone a brief recap of what the folate pathway has, so FOL-A, that is DHFR, converts dihydrofolate to tetrahydrofolate, and then tetrahydrofolate is converted by 2 different enzymes into 5,10-methylene tetrahydrofolate, which is the cofactor of DNA synthesis. And the 2 enzymes are GCV-TPH, which is the glycine cleavage complex, and GLI-A, which is SHMT. Now, in the resistant E. coli, we found that DHFR was upregulated, which I spoke about earlier. And when you look at this particular arm of the pathway, we saw that GLI-A was upregulated and then glycine cleavage was actually downregulated, which means that the responsibility or the burden of forming this very important metabolite, that is 5,10-methylene tetrahydrofolate, was mostly on GLI-A. So then we wondered whether the dependence on SHMT or GLI-A was an emergent vulnerability in trimethoprim resistant E. coli. So then what we did was carried out a deletion of GLI-A in the resistant strains. And then we had duplicates of resistant strains, that was 32XR1 and 32XR2. Compared to the MIC of wild type E. coli, which is 0.5 micrograms per ml, these strains had a very large MIC. But when we deleted GlyA, we found a 100-fold reduction in MIC in R1 and like a 32-fold reduction in MIC in R2, which was very, very encouraging. So what we also carried out was a comparative evolution of wild type and the delta GlyA E. coli, just to see whether they gain resistance at equivalent rates. So I had 6 replicates of a wild type, which are shown in the shades of red here, and 6 replicates of delta GlyA. And over a course of 10 days, I went on subculturing them in higher concentrations of trimethoprim once I observed visible growth. And what I saw was that the MIC on each day, the comparable MIC between GlyA and wild type MG1655, the GlyA strains always had a slower rate of gain of resistance as compared to the wild type MG1655. So this is what the whole story is about, that by analyzing the transcriptome of wild type E. coli and resistant E. coli, we could identify GlyA as a strategic target in trimethoprim resistant E. coli. So I'll shift gears a bit and then speak about the other part of my talk, which is my postdoc work, in which I am focusing on prioritizing bioactives produced by the gut microbiome. So this is basically the good and the bad microbes or metabolites that they produce that are very health relevant. So most of us know that microbiome derived bioactives, which can be the microbes itself, their gene products or small molecules, influence host health and immunity in several different ways. And although a lot of these are known, for example, we know that lactobacillus is really good for us. We know that metabolites such as butyrate is really good for us, and TMAO is bad for us. Actually, very few is known, and there can be thousands and thousands of health relevant gene products and metabolites that are currently unknown to us. So what we do in our lab is actually build computational tools to interrogate metagenomes and metabolomes and
Also, you have less than a
prioritize.
minute. Sorry.
Yep, thanks for the reminder. So and prioritize bioactives of interest. So the tool that I'm developing is called Macaron, which prioritizes metabolites of interest, and my colleague has developed another tool called MetaVibler that prioritizes gene products of interest. And to just give a brief overview of how these tools work, so the input is case control, metagenomes, and metabolomic data. So metagenomes have like millions of genes, metabolomes have tens of thousands of metabolites. So these are features, genes or metabolites, and then we rank each feature based on some phenotypic or ecological indicators of bioactives, such as how prevalent it is, how abundant it is, and what is the effect size in a phenotype of interest. And then we combine ranks from all these different indicators of bioactivity and then prioritize them to identify the most interesting microbial bioactives. So what we are doing, in other words, is looking at millions and thousands of genes or metabolites respectively, and then just prioritizing a few hundred ones that we can experimentally study in greater detail. We are currently applying this pipeline to the HMP2 cohort that has individuals with inflammatory bowel diseases and healthy controls to identify microbes and metabolites that are of great importance in IBD. And for those of you who are interested, my lab actually develops a lot of computational tools for microbiome analysis. It is called the BioBakery suite of tools, and it is freely available to everyone for use. And with that, I will thank the Department of Biochemistry again for the kind invitation, and my PhD boss, Professor Nagasuma Chandra, all the collaborators who have really helped me through my PhD, as well as my current boss and mentors. Thank you, and I can be reached at this particular email ID as well as on Twitter. Thanks again.
Thanks, Amrisha, for the nice talk. Now I'd like to ask if there are any questions from students or from any audience. Okay, so if there, yeah. Okay, so if there are no questions, so then I'd like to thank again Amrisa, and yeah. So now Savanun, continue.
Thank you so much.
Okay, our next speaker is Deepash Kotiwal. Deepash graduated from Professor Shikala Loria's lab and is currently works as a postdoctoral researcher in Harvard Medical School. He's going to talk about understanding the multi-level regulation of eukaryotic transcription, insights from cohesin and RNA pol 2. Deepash.
Hello, am I audible?
Yes, yes, you are.
Oh,
It's
okay.
also full screen.
Okay, thank you. So I would like to, first of all, I would like to thank the organizers for giving me this opportunity to discuss my work on this platform. Today, I'm going to talk about understanding the multi-level regulation of eukaryotic transcription and role of cohesins and RNA pol 2 in this process. So, sorry. Sorry, just give me a minute. I'm not being able to move my slide.
We can see your slides, Deepash. Go ahead.
Oh,
now you unshared. Okay, can you share again?
Yeah. Okay, yeah. So this diagram here shows a kind of a textbook view of gene expression and the multiple steps that it can be regulated at. My interest lies in the first part of this process, that is transcription and its regulation. Transcription can be regulated in multiple steps, including chromatin organization level. So, chromatin organization level, chromatin can be organized as a closed chromatin or an open chromatin. And of course, we know that closed chromatin are not accessible to transcription machinery and hence not conducive for transcription. For example, facultative and constitutive heterochromatin. In contrast, permissive heterochromatin allows binding of sequence-specific transcription factors, which remodel nearby nucleosome, thereby creating an open chromatin structure, which allows binding of a full set of transcription machinery and initiation of transcription. So for the major part of my talk, I'm going to talk about the role of cohesin in the formation of this closed chromatin structure and its impact on transcription. And later on, I'll talk a little bit about my current work on RNA pol 2. So in budding yeast, cohesin complex consists of 4 proteins, SMC1 and 3, along with 2 non-SMC protein, MCD1 and SCC3. Cohesin binds to centromere, pericentric region, and at the specific sites along the chromosome arm, and as well as subtelomeric region. It is required for sister chromatid cohesion, double-stranded break repair via sister chromatid recombination, chromosome condensation, and regulation of gene expression. So when I started my PhD work, we knew about cohesin's role at centromere, pericentric region, at the specific sites on the chromosome arms, but we did not know what the function of cohesin is at the subtelomeric region. But what we did know at that time is that cohesin is required for regulation of gene expression. And interestingly, one of the conserved feature of telomeres is that they exert position effect. That is, that genes that are present near telomeres are expressed at a lower level compared to rest of the genome. So this schematic here shows the establishment and spreading of telomere silencing in budding yeast. From the published studies, we know that genes that are present within 20 kb from the telomere are expressed at a lower level. interestingly, only sub-repressed region lies only within 6 to 8 kb from the telomere end, and the proteins required for repression of this extensive silent domain that lie outside the subbinding region remain poorly understood. So we asked the question, could cohesin be required for repression of telomere proximal gene? To address this, we carried out RNA sequencing analysis using cohesin mutant MCD1-1. And interestingly, we found that a mutation in cohesin leads to upregulation of a large section of telomere proximal gene compared to only a small fraction of other genes are upregulated. When we take a look at this data in terms of numbers, we see that telomere proximal 42% of the telomere proximal genes were upregulated in cohesin mutant compared to only 10% for the rest of the genome, suggesting that cohesin is required for repression of telomere proximal genes. We went on to analyze repression of these derepression of this telomere proximal gene with respect to distance from the telomere, and we observed that though cohesin repressed region lies within 30 kb from the chromosome end, the maximum derepression was observed within 4 to 24 kb. This was an exciting and interesting finding to us since telomere proximal genes are repressed at least until the 20 kb from the end, and subprotein binds only in the first 6 to 8 kb. So this shows that cohesin is one of the first factor identified that can repress the extended telomere silencing domain, which is not overlapped with subbinding proteins. So having established cohesin's role in telomere silencing, we ask the next obvious question: how does cohesin perform this function? So one of the conserved features of telomeres are that they are tethered to the nuclear envelope, and this helps in telomere silencing. So we ask, could cohesin be required for this function? To test this possibility, we use a strain where LACO repeats are integrated near telomere 5R and LACA is tagged with GFP. This strain also has NUP49 tagged with GFP to illuminate a nuclear envelope. These images here shows a tethered versus untethered configuration of telomere. And when we analyze this data, we observe that mutation in cohesin leads to a moderate but statistically significant increase in untethering, suggesting that cohesin is required for telomere tethering to the nuclear envelope. Another conserved feature of telomeres is that that they are organized in a heterochromatin-like structure, and it has been shown that this structure is important for telomere silencing. So to assess the telomere heterochromatin structure in cohesin mutant, we carried out dimethyl assay. Briefly, if the telomere heterochromatin is compact, it is refracted to dimethylase, and DPN1 digestion will be inefficient. And if telomere heterochromatin is more open, or telomere chromatin is more open, there will be efficient digestion by DPN1. And we observed that after performing this assay, we observed that cohesin mutant cells are highly sensitive to DPN1, suggesting that telomere heterochromatin structure is lost in the cohesin mutant. We went on to delete SER2 in the MCD1-1 background, and we found that in double mutant cell, the telomere heterochromatin structure is even more open compared to any of the single mutant, suggesting that both cohesin and SER protein makes independent contribution toward formation of this structure. We use South3A1 as a control. So based on these data, we present a model wherein wild type cells, telomere are tethered to the nuclear envelope. They are organized in a heterochromatin-like structure and present in this peripheral silent region. And altogether, this leads to telomere silencing. In mutant cells, there could be two possibilities. One, that telomeres are still tethered to the nuclear envelope, but there is loss of heterochromatin-like structure near telomere, which will pull these telomere proximal genes out of the nucleus and lead to out of the peripheral silence region and lead to their derepression, whereas a telomere proximal gene remains silent. Another scenario could be where there is a loss of telomere tethering along with loss of heterochromatin structure. And in this case, all the telomere proximal gene will be de-repressed. Now moving to the second part of my talk, after analyzing transcription regulation at chromatin organization level, I joined Professor Steve Boratowski's lab to understand transcription regulation, this time at the very heart of transcription, that is RNA Pol2. Due to the lack of time, I may not be able to, I won't be able to show all the data and will only be able to briefly introduce my project here. So we all know that RNA Pol2 is a conserved enzyme that transcribe DNA into mRNA and many non-coding RNAs.
Dipash, one more minute.
Yeah. So RNA Pol2 carries this unique C-terminal domain on its largest subunit RPB1. The CTD is composed of 26 heptad repeats, and interestingly, each heptad has 5 amino acids that can be differentially phosphorylated during a transcription cycle. Another interesting, so surprisingly, most of the data on CTD phosphorylation comes from serine 2 and serine 5 phosphorylation, whereas tyrosine, threonine, and serine 7 phosphorylation remains understudied. In fact, they are so understudied that sometimes they are together called as orphan residues. So I'm studying, it is important to study these residues, especially since tyrosine 1 along with serine 2 and serine 5 are essential for viability. Moreover, very little is known about the kinases and phosphatases that modify these residues. So that's another angle that I'm working on. Second part of my project involves spatial and time resolved analysis of CTD phosphorylation. So here we are combining in vitro transcription on immobilized template with mass spectrometry. So with this, I would like to conclude my talk and thank my mentors, Professor Shikala Loria and Professor Steve Boratowski and all the lab members. I also thank Professor Jared Marto and his group for their help with mass spectrometric experiments. Thank you for listening, and I'll be happy to take any questions.
Nice talk, Dipash. Are there any questions from audience?
Dipash, I have a question. I don't work in this field, but this is Surendra Sharma from Brown University, your neighbor from Boston. So I have a question. I have always thought about aging cells have shortening telomere, and cancer cells maintain their length and for their active growth. So the transcriptional machinery that you described, how does that change? How does that interplay between cancer cells and aging cells?
So I do not know the exact answer to that question, but that could be one of the interesting area that we can look at, since I would like to say, like, the telomere proximal genes are under wild type condition, they are already repressed. But in aging cells, since there will be a telomere shortening and probably loss of telomere silencing, sorry, I do not know the exact answer. I think that transcription.
That's fine. I think maybe
probably he can move to our lab and discuss. Just next to you, right? All right. So we'll move to next speaker in the interest of time. Saran will take over.
Yeah, thanks. Thanks, Dipash. So our next speaker for this session is Raju Rai. He's a PhD student from Professor Rajguru's lab at biochemistry, and he's working on RGG motif exclusively on SPB1 is required for P-body disassembly. Raju, are you there?
Yes, I am here.
Okay. Looking forward to your talk.
So first of all, I want to thank the organizer of this meeting to provide me a wonderful opportunity in this platform to present my work to all of you. My work is about an RGG motif containing protein SBP1, which is required for post-processing body, P-body disassembly. So to start with, what are RNA granules? So RNA granules are microscopically visible RNA and RNA binding protein complexes, which are majorly found in cytoplasm. These are non-membranous and they are very rigid and insoluble in lysine. RNA granules are also have been implicated in translation repression, mRNA decay, and also can coexist for changing components within themselves. RNA granules are also found to be associated with neurodegenerative diseases, where fat proteins are implicated. Fat are a class of proteins which includes fusion sarcoma, Ewing sarcoma, RNA binding protein 1, TAR DNA binding protein 43. Mutation in any of these protein causes mislocalization of the protein from nucleus to cytoplasm, resulting in pathological inclusions, which are implicated in neurodegeneration. So, assembly of RNA granule is mostly well studied, and here I want to bring your focus into the intrinsically disordered region containing protein, where here is an example of EDC3 YGEF domain, which is intrinsically disordered, and here in PGL proteins we have RGG box, which is a disordered. These protein bind self, they can interact with self and can also bind to RNA and other proteins, RNA binding proteins, to form P bodies and also P granules.
Raj, you're there? Raju.
I can't hear you. Raju, you're muted. Also, it stopped in between a little bit.
Okay, sure. I will. So we hypothesize that as the role of low complexity sequence in assembly of RNA granule is well characterized, it is possible that low complexity sequence could contribute to disassembly of RNA granule, which is not known. To test the hypothesis, we took two proteins, low complexity sequence containing protein, SCD6 and SBP1, which contain the RGG motif, which is an intrinsically disordered sequence. Both these proteins are granulocytin protein and also translation repressor. These proteins are known to repress translation by binding to EF4G. We use the following experimental method where we ectopically expressed PAB1, which is a stress granule marker, and EDC3, which is a P-body marker in the cell indicated. Cells were grown till 0.4 OD. And then we stress the cells with sodium azide, which is an oxidative stress for 30 minutes, followed by recovery for 1 hour. All these steps were documented using live cell microscopy. In unstressed condition, we don't see any stress granule and very few P-body forming, which is well known. Upon stress, both stress granules and P-bodies are found, which is well studied. But interestingly, when during recovery, we found there is a defect in EDC3 RNA granule disassembly in del SBP1 as compared to wild, and also the its RGG counterpart SCD6. So this says that SBP1 might play an important role in RNA granule P-body disassembly. EDC3 is not the only protein that is rising to P-bodies. Therefore, to check that it is not specific to EDC3, we try to test other two P-body protein, which is DHH1 and SCD6, and we found that in del SBP1, both DHH1 and SCD6 are defective upon recovery. So this is that SBP1 plays an important role in P-body disassembly. Next, we try to test which domain of SBP1 is required for P-body disassembly. As an interest of time, we are not showing all the results, but I'm going to implicate that when you use the RRM1 mutant and RRM2 mutant, we found there is no disassembly defect of EDC3 RNA granule during recovery. But interestingly, when you took the del RGG mutant of SB1, we found there is a defect in EDC3 RNA granule disassembly upon recovery, which is also shown in the graph. In addition to that, we also tested what happens if we add the only RGG mutant of SBP1 in the cells, and we found that the only RGG mutant is sufficient to drive the RNA granule disassembly as compared to its DGG part. So this suggests that the RGG is very important for disassembly of EDC3 RNA granule or P-bodies. So till now, what we got to know is SBP1 is required for P-body disassembly during recovery from stress, and RGG motif is important P-body disassembly. Next, we try to test is that test the role of SBP1 in P-body disassembly mediated by EDC3 SBP1 interaction. To test that, we took purified wild type SB1 and fragments of EDC3, which is LSM, FDF, and YGF, and did a glutathione cephalus pull down, where we found that SBP1 can bind both LSM and the YGF motif of EDC3, as also shown in the graph. YGF motif is interesting because YGF motif of EDC3 is required for self-interaction of EDC3 and to form a mature P-body in the association of mRNA and NADH. So next we tested that does EDC3 form granule in vitro. For that, we purified EDC3 with mCherry and added in a phase separation buffer. We found that EDC3 form tiny structures when grows in both size and intensity when RNA and NH is added, also shown in the quant. So as the assay system is working, we next asked that what happens if we add wild type SB1 in this reaction system. So added wild type SBP1, we found that the P from granules are decreased both in size and intensity as compared to buffer system without SBP1. Interestingly, the low complexity region deletion of SB1 does not have any effect in the droplet size or density. So it shows that RGG motif of SBP1 is required for P-body assembly. But what or how? So as it already known that EDC3 can self-associate with using the IGF motif. And also from our the previous result, we got to know that SBP1 can bind to EDC3, bind to YGF and LSM domain of EDC3. So we next ask the question, is the disassembly is because of a competition between EDC3 and SBP1 to bind to the YGF motif. To test that, we purified full-length EDC3, wild-type SBP and the YGF motif of EDC3 and did a glutathione pull down, where we found that, as reported earlier, EDC3 can bind YGF, and also as I have shown earlier, SBP1 can bind YGF. But when you added both the
Raju, you have 1 minute. You're already
Okay.
past. Yeah, finish it off.
Yeah. So when added both the protein, we found that there is a decrease in EDC3 interaction with YGF, which shows that EDC3 SBP1 interaction is important for granule disassembly. So as mentioned earlier, fat proteins has been implicated in various neurodegenerative diseases, and recent publications suggest that yeast is a well-established model system to understand fat proteins. Therefore, we overexpressed EWSR1 in wild type and delta SBP1, and found that EWSR1 granule dissociation is defective compared to the wild type. Consistent with the microscopy result, also found a growth defect in delta SBP1 compared to wild type as depicted in the growth, suggesting that the granule disassembly is important, as persistent granule could impart toxicity to the cells, which is well established. So taking all the result into consideration, we propose the following model where EDC3, which can self-associate and form P-bodies in association with mRNA, and NADH in presence of SBP1, it comes with a binding to YGF and leads to P-body disassembly. So this is my lab. I thank Raj for the driving factor and being awesome, and thank Aishwarya and Geetarth for helping me with this project. Thank you.
Thanks, Raju, for a great talk. Are there any questions from the audience? Okay, if not, I have one short question, Raju, that
Yes.
this in vitro assembly, right? The phase separated, the purified M-cherry thing. Did you measure the
Yes.
size of those granules? How similar it is with the in vivo? So if you measure the size of the granules.
So as compared to it's an in vitro system. So it is reported that the granules that is formed in vitro are compared to size, it will be more greater than the in vivo granule we see.
Okay, so it is not like in between somewhere between the in vivo and in vitro. They're not matching. So they are totally different. That's what you're saying.
Yeah, it is actually reported. Like, so we have found it and also known.
Okay, sure. Thanks. Thanks for the time. So next, Deb.
Thanks, Raju, for the nice talk. So now it's time for Abhinava. Abhinava Banerjee is a PhD student at Dr. Mahipal's lab, and he joined 2020. Yes. And he's one of the PMRF fellow in our institute, and we look forward to your talk.
Yeah, am I audible? Hello?
Yes, go ahead, Abhinav.
Okay, yeah, thanks a lot, Devaprato, for the introduction. I would like to start off by thanking the department. It's been a great pleasure to be here. It's just been about a year that I'm here, and in this 100-year-old department, we are the youngest lab. We started off just last September or October, and I was the first member along with Mahipal to start off. So without wasting much of the time, I would like to go through what are the things that our lab actually works on and what are the various projects that we are planning to go ahead with. So first and foremost, our basic interest is to actually understand the DNA, the biophysical aspects of DNA, and the various dynamics the DNA molecule actually goes through. So as we all know that DNA actually carries the genetic information to the next generation, and it also dictates various cellular functions under various different conditions that a cell is under. So as this explains that DNA is a very crucial molecule and it needs to be protected very carefully, but at the same time needs to be accessible and also needs to be compacted and organized and stored because the information is large and the space inside the cell is really, really less. So if we go through the eukaryotic way of compacting genome or compacting the entire DNA, it's first wrapped around nucleosomes, and these actually form various topologically associated domains, also known as TADs, and they are actually segregated as domain A and domain B based on their active marks and based on whether they are more open or compacted. And finally, the entire chromosomes, big chromosomes, actually take up something called chromosomal territories within the nucleus. So if you remove the histones, which was done way back in 1977, when you treat it with high salt concentration, you see that there are these huge loop kind of structures which are actually attached to the chromosomal scaffold. So this was done on metaphase chromosomes. If we try to see in what's the state in bacteria, because they also have a very complex genome and they also need to compact it, and they are much, much smaller than eukaryotic cells. So we can see that even the bacterial genome is very, very similar, as they have, even though it's circular, but they actually have a central anchor, and where there are these various different loops with different supercoiled structures, which is actually considered to be an added level of gene regulation. But for all these various compaction, accessibility, and various different DNA-oriented processes, a key player is the proteins that the DNA actually codes for. So how do we see the DNA and the protein interacting? So till now, there have been various different techniques, which are classical biochemical assays that have been used widely. And they are like DNase footprinting assays, electrophoretic mobility shift assays, various different southwestern blotting, east-1 hybrids, and with more recent technologies, which are antibody-based, we can do chromatin immunoprecipitation, and with further development in the sequencing throughput of various technologies, they went to DAM-ID and various chromosome conformation capture techniques, which allows us to actually see various different interactions between various genomic loci. But the problem is that these don't actually give us, these actually only give us static pictures and static interaction information, and it does not give us a dynamic information about how this DNA protein interaction is actually occurring and its dynamics. So we actually use various techniques, and I would be talking briefly about two techniques and how we utilize this. So firstly, we use single molecule imaging, and it opens up a whole new world to actually visualize single molecules at their details and to see every single molecule individually and how they interact. So here on the right, we can see that this is basically a lambda DNA, which is actually 48 kilobases long, around 48.5 kilobases long. And we see that it's actually what we do is basically we have a very, we process a surface, mostly glass, and we can actually tether flow stretch the lambda DNA on the surface, which are actually biotinylated on each ends. And we can see the entire molecule under a fluorescent microscope. So this was the assay that had a breakthrough in 2018, where actually what we are seeing here is actually condensin molecule, which is actually performing loop extrusion, and the condensin molecule, which you can see here in red, is actually tagged with ATO647N, and it's performing a loop extrusion event. And this was one of the first visualization of how the condensin molecule actually performs loop extrusion. Further, we also use atomic force microscopy to actually see individual molecules in great details and how various DNA protein interactions can occur. So let me first come to the first important part, which is protection and compaction. And for this, we actually use we target some mycobacterium proteins, which is, as we know, mycobacterium is a deadly disease causing tuberculosis, and they actually have to survive in very harsh conditions in our body until they get an opportunity to actually unleash their weapons. So to actually protect the nucleoid of the bacteria, they use two different nucleoid associated proteins, which is HU and LSR2. They actually are known to have DNA binding capabilities and have great level of DNA compaction, as I will show you. So these are bare double-stranded DNAs under atomic force microscopes. And once we add the nucleotide associated proteins. In this case, the HA protein, which is also known as a histone-like protein. We can see this complex meshworks that they actually form, and these are actually single strands, but here you can see that they actually bridge various large molecules and they form this complex meshwork. And the dynamics of this is still yet to be understood. It's reported that these actually form these kind of structures, but we are really interested.
Muted and your presentation is gone. Can you unmute and share again? Yeah.
Tell him. Sorry about it. I don't know what happened.
That's fine. Go ahead.
Okay, fine. So further, I would like to talk about accessibility and the proteins that are involved in accessing the genome. So we are interested in seeing the RNA polymerase and how it actually moves along the DNA. And this has actually been visualized many a times, but actually what we are more interested is in the way how supercoiling actually affects the movement of RNA polymerase. This is really, really crucial in case of bacteria, because bacteria have circular genome and they are more prone to getting supercoiled as in contrast to eukaryotic genome, which are usually linear. And there are far more proteins like topoisomerases that are involved in removing these supercoils. So back in 1989, Liu et al. Actually proposed something called a twin domain supercoiling model, where polymerizing RNA polymerase actually forms positive supercoils downstream of its region. And actually this actually also generates negative supercoils upstream of where it has actually passed through. What we want to see is actually how these supercoils are involved in regulating and whether the underlying sequence present in the genome actually allows to pin certain supercoil structures to certain regions in the genome to actually guide where RNA polymerase can actually come sit and start accessing the genome and transcribing the target gene. Finally, we are also interested in the global organization of the genome and how the entire vast information is actually organized into the genome. So for this, people have used various different techniques like chromatin conformation capture techniques, which speaks a lot about the spatial point-to-point interaction, but it actually does not talk about the spatial positioning of different loci inside the entire nucleus. Though there are various different imaging techniques that have been used till now with confocal microscopy that has allowed to visualize this, but we want to visualize the entire organization of the genome in much greater detail. So for this, we use a technique called DNA paint, where there is various different targets are actually tagged by a certain docking strand. Here we can see the docking strand. And there are certain imager strands which carry a fluorophore. These imager strands are actually complementary to the docking strand, and these imagers go and transiently bind and interact with the docking strand for short durations of time, around less than a second. So this allows us to get blinking events throughout the entire cell, and this allows us to visualize it at much greater resolution than what conventional diffraction limited imaging can actually allow us to do. As we can see on the right, this is a diffraction limited image of telomeres. And if we zoom into this red box here, this is what we see in the diffraction limited image. This is the best that we can go to. But if we use DNA paint, the super resolution technique that we plan to employ in understanding the chromatin architecture, this is how we see the entire structure of the telomere regions. So we further want to use this technique to actually be able to understand the underlying components of underlying gene and its underlying histone modification mark, because this is a very crucial aspect. And what chromatin immunoprecipitation or chip data actually gives is the information that is actually ensembled average from a mass of cell. Single cell chip data is definitely present, but it's really, really difficult to perform single cell chromatin immunoprecipitation. And we want to actually be able to develop a tool that can visualize the gene and the underlying histone marks. So with the interest of time, I would like to wrap up quickly, and I would like to introduce my team This is my entire team, and with special thanks to Professor Muniappa, Nagaraj, and Professor Deepak Nair for allowing us to use instruments and to be able to do our stuff. Thank you.
Thank you, Abhinav, for the excellent talk. Are there any questions? Okay, if there is no question, I have very general question that I'd like to ask you.
Yeah.
So for instance, when you said that you want to do the genome organizing this chromatin study, so which organism you would prefer?
So at first we are trying to, sorry, continue. I'm sorry,
Yes,
I cut
yes,
you.
Go ahead.
Yeah. At first we want to basically, so DNA paint has never been done to actually study the chromatin conformation, chromatin architecture, except for visualizing very specific targets in the nucleus. There have been only, I think, a handful of papers that only see a single protein, but they don't see actually an end symbol of proteins. They don't see many different proteins. So at first, we would like to use normal HeLa cells or MCF7 cells that is easy to culture and easy to grow, easy to standardize. But we further want to use this technique in primary cells so that we can actually see how the entire chromatin is organized in primary cells. And that's, I think, has a great amount of information to be explored there.
Great. Thanks a lot.
Can I add a comment? Sorry.
Sorry, but I can't see who's speaking.
It's Harinder Singh.
Oh, yeah.
And I wanted to first of all compliment you on a wonderful talk. And
thank you so
much.
And
in terms of applications, I'm actually extremely excited that Mahipal's lab is developing the last approach, because I think many of us, including my own lab, are interested in the architecture of transcriptional hubs. So these are interacting promoters and enhancers, and that are part of the higher order structures within the nucleus. And having approaches like the one you are describing will be extremely valuable.
Thank you. Actually, we are also really interested to see, because the field of super enhancers are really new, and there is a lot to explore in that sense as well. We actually want to see interactions, long-range interactions. What we actually want to see is to be able to visualize these long-range interactions. Chromatin conformation techniques definitely give us data that there is long-range interactions, but what's more interesting to see is how every single cell actually has, what's the cell-to-cell heterogeneity. I think it would be really interesting to see in that direction.
Okay. If there is no question, then thanks again, Abhinav. So now
Thank you.
Thanks to all the speakers and the chairs of the session. Now I would like to request Sweta again to take over from here and initiate the session 4.
Thank you, Satyaranjan. Welcome everyone to the 4th session of the day. This session is based upon genome biology, and it consists of talks by 2 professors, Professor Harinder Singh and Professor Rachel Green. The session will be chaired by Professor Shikha Laloria. She joined ISC in the year 2000. She is now a professor in the department. Her area of research majorly includes SMC complexes and post-transcriptional protein modification. Now I welcome Professor Shikha Laloria to kindly introduce the speakers of the talks.
Thanks. Am I audible to everyone?
Yes, you're audible.
Yes, Shikha. Yes.
Yeah, okay, great. So it's my pleasure to chair this session, and I welcome all of you to the genome biology session. We are running a bit late, I think about 12 minutes or so. So let's see how we can catch up, or maybe we need a little more time. So this session has got 2 speakers, Professor Harinder Singh and Professor Rachel Green. Let me introduce the first speaker, Professor Harinder Singh. He's from the University of Pittsburgh, and he's a professor of immunology, and he's also the director of the Center for Systems Immunology at the University of Pittsburgh. Harinder obtained his PhD at Northwestern University in 1984 under the guidance of Dr. Lawrence Dumas. And he was later on a Jane Coffin Child's postdoctoral fellow at MIT with Dr. Phil Sharp and David Baltimore. He joined the faculty of the University of Chicago, and there he became the Louis Bloch Professor of Molecular Genetics and Cell Biology, and also an investigator of the Harvard Hughes Medical Institutes. I think he also had a stint in Genentech as a senior director of discovery immunology and a staff scientist for some time. And after that, he was on the faculty of the Cincinnati Children's Hospital Medical Center and also the director of the Division of Immunology over there. And in 2019, he has moved on to the University of Pittsburgh, where he's currently the director at the Center for Systems Immunology, and he's been there since. His lab's interest is mainly in the analysis of transcription factors and gene regulatory networks that regulate the development and functioning of innate as well as adaptive cells of the immune system. So today, Professor Harinder Singh will be talking about the nature of combinatorial interactions of transcription factors underpinning the decoding of mammalian regulatory DNA. Over to you, Harinder. Please start your presentation.
Shikha, thanks so very much. I want to thank Mahipal and of course the organizers of this very special conference to celebrate the 100 years of the Department of Biochemistry, which is a storied department. So I am honored to be to play a little role in the celebration. Let me just make sure, is my slide fully visible?
Yes.
Yes,
Okay,
it
great.
is.
All right, thank
Slide,
you.
please.
Okay, so as Shikha said, currently, I'm at the University of Pittsburgh, where we've launched the Center for Systems Immunology. And I just want to say one thing, since a lot of next generation biologists are in Bangalore. And I think at this point, modern biology is going to be greatly impacted, not only by experimentalists, but by computational biologists and also biologically inspired engineers. So I think that's sort of the triad that is going to fuel discovery in 21st century biology. And we're trying to assemble the talent to be able to play this game. Shikha pointed out that, I trained as a PhD student with Larry Duma at Northwestern, and then as a postdoc with Phil Sharp and David Baltimore. And since this is a day of reflection, I hope you'll indulge me in reflecting a little bit in terms of my own career. It really has been riding the roller coaster of DNA. And I thank both Professor Duma and Sharp and David Baltimore for inspiring a lot of the work that then my own lab has pursued over multiple decades. And at the end of the day, as a molecular biologist at heart, I'm actually what I would call an accidental immunologist. I have no formal training in immunology, but my training is entirely in molecular biology and genetics and genomics. I became interested in discovering transcription factors and the nature of the regulatory codes that are operative in the mammalian immune system. And so over the years, we've explored, discovered and explored the nature of these regulatory molecules, the circuits within which they're embedded, both in the context of development of different cells of the immune system, as well as their functioning in response to antigen encounter. But the interests are very fundamental in nature. So the immune system for us is the model system, but we think we can, learn rules that apply across the board to many, many different cell types. Now, what I would like to turn to now is that transcription factors, as you know, are really the key components embedded within cells that are decoding regulatory information. And they're controlling the flow of information within cells as they transact different processes. And This particular schematic just shows you several transcription factors that we either discovered or began to work on over a period of time. Just the structures of the DNA binding domains, recognizing particular DNA sequences are shown in the schematic, and I'll have more to say about them in a bit. Now, I think to the previous talk, actually, Abhinav's talk introduced you to some fundamental problems when it comes to the control of transcription in metazoans, higher organisms, multicellular organisms. And that is that the regulatory information, the bits of DNA that control the spatial pattern of gene activity or the temporal pattern of gene activity turns out to be fairly complex. And we'll come back to the nature of the complexity of that code, but essentially, there are numerous binding sites for transcription factors that comprise what is denoted here as an enhancer. The other, and we're still trying to understand the nature of this combinatorial complexity, and that's the problem that I'm going to briefly talk about in the rest of the talk. But the other fundamental problem is that these regulatory sequences, complex in nature, can act at very, very large distances in cis, and in some cases even in trans, in terms of communicating their action to the promoters of genes whose activity they're controlling. And these distances in DNA length can be in megabases. So there is the problem of how do you transact molecular interactions at a distance with the fidelity that's needed to spatially and temporally and very precisely controlled gene activity. And of course, Mahipal's lab, as you know, is taking on this problem in terms of getting at the underlying molecular mechanisms. But I'm going to focus on the nature of the combinatorial code for the rest of the talk. So at this point in time, we know what the nature of the transcriptional regulatory code is in terms of its fundamental units of organization. And those fundamental units are short but redundant DNA motifs, typically 6 to 10 base pairs in size, that represent the binding sites for transcription factors. And the transcription factors, for those of you who like to think about the genetic code rather than the regulatory code, the analogy would be they're analogous to amino acyl tRNAs that are interacting with codons in the mRNA. But the analogy falls apart beyond that. Now, so it turns out, though, that these, as I mentioned, that this code is actually quite complex. The bits are these 6 to 10 base pair sequences, and they have redundancy within them. But then they are organized in these rather diverse combinations to generate higher order structures. And it is that higher order organization that actually is critical in determining, when a gene is on, where it's on, and which signals it responds to, and so on and so forth. And so this problem has remained as an interesting fundamental problem for those of us who are interested in eukaryotic gene regulation to try to solve. Now, the approaches at this point in time, and I say this point in time because when I started as a postdoc, as a molecular biologist, we were identifying actually these bits as well as the transcription factors that recognize the bits. In fact, my work, along with others, was amongst the first to identify new bits and new transcription factors. But at this point in time, we know all the bits, essentially, all of the binding sites, and we know all of the transcription factors that are encoded within the mouse or the human genome. And so that part is clear. What isn't clear, of course, is the higher order structure. And now there are many methods, and actually they were covered partially in the last talk, that are being used to define the nature of the regulatory sequences. And literally, the catalog of regulatory sequences at this moment in time is building daily. We have tens, if not hundreds of thousands of regulatory sequences to mine. The problem, though, is in deciphering the patterns. Now, one of the patterns that's been noted are composite DNA regulatory elements, or I will abbreviate them as CEs. Now, this pattern involves stereospecific juxtapositioning or two or more binding sites of distinct transcription factors. And the stereospecific juxtapositioning involves strict spacing between the two sites, and actually the orientation of the sites matters as well. So these combinatorial elements actually have been, to date, 20 to 30 have been rigorously biochemically and molecularly analyzed. They're very attractive means of organizing regulatory information because, as you can imagine, they enable combinatorial control of gene activity. And they're very nice signal integrators because if you have one transcription factor induced by signal A, another by signal B, the DNA would do the integration in terms of enabling their cooperative action. Now, of the well-described composite elements, we've contributed to the analysis of two of these. And you can see in the right here with my cursor, this is a composite element that's comprised of binding sites for the ETS family transcription factor PU1 and the IRF family transcription factor IRF4. They cooperatively assemble on this DNA sequence. And interestingly, these members of the IRF family can recognize a distinct set of composite elements, in this case, partnering with members of the AP1 family. And what was very surprising to us when we discovered this particular composite element was that it came in two flavors. You can see one of them, the two sites are juxtaposed. In the other case, there's a 4 base pair spacing. Those base pairs do not matter in terms of the sequence. And one of the motifs is actually flipped in its orientation. And I won't go over all of the experimental work that was done over the years to demonstrate rigorously biochemically, as well as molecular biologically, that the spacing and the orientations of these sites mattered in terms of enabling cooperative assembly and action of these transcription factors. So now the problem that we've been confronted with is the following. We know as I said, 20 to 30 of these composite elements that have been described in the previous several decades. And the issue is, do these composite elements actually represent a second order feature of the genomic regulatory code? If that's the case, then they should be prevalent in regulatory sequences and not outliers, that is, albeit interesting outliers. So we needed a way to systematically analyze for the occurrence of composite elements in a large set of regulatory sequences, which I mentioned to you are available now for analysis. So a postdoctoral fellow, Viren Chaudhary in my lab, who was interested in this problem, developed an R-based computational pipeline called CE-seq, which will systematically search for composite elements in large sets of regulatory sequences. And just to schematize the problem, if this is the binding site for a transcription factor A, binding site or motif for transcription factor B, and a priori, you don't know the number of base pairs that could be used to create a composite element, nor the relative orientation of these individual motifs. For each spacing that you allow for, there are 4 configurations. So what CE-seq does is you provided a set of test sequences, a set of control or background sequences, as well as the motifs for transcription factors that you want to search in terms of the occurrence of potential composite elements. And it goes through and rigorously scans all of the sequences and bins them in their stereospecific configuration, and then tests for statistical enrichment. And the outputs that you get are position frequency matrices of likely composite elements. Now, to validate this whole computational approach, we took a set of 10,000 enhancer sequences that we had previously described that are active in B cells of the immune system, and we searched them for composite elements that would be comprised by the binding sites for these transcription factors. And the CE-seq or BitSeq actually accurately predicted the occurrence of these AP1 IRF composite elements, as well as the ETS-IRF composite element that previously we'd described. and also the NFAT AP1 composite element that Anjana Rao's lab, who I know has connections with the department and the Indian Institute at the Indian Institute of Science, we were able to recover that composite element as well. So given that this approach works, we decided now to systematically apply it to an extremely large sequence space. And in 2019, a landmark resource paper was published in Cell by the ImGen consortium, where they determined open chromatin regions in essentially 90 different immune cell types and states. And these regions, each 180 base pairs in size, likely represent combinatorial binding of different transcription factors that are controlling the activity of genes in these cells. And in terms of the complexity of the sequence space, it is 500,000 accessible chromatin regions delineated by ATAC-seq. And so we use CE-seq to systematically search for composite elements in this space using all transcription factor binding motifs known to date. And so this is an all-by-all TF scan of these 500,000 sequences. It took several months actually on the PIT computing cluster to complete the task. So the question was, are CEs prevalent? And what the analysis said was a resounding yes, because we generated 22,000 composite elements that were predicted to be operative in the regulatory space of the immune system. So mind you, we go from 30 or so to 20,000. Now the task at hand though is these are predictions, and the issue is how can we validate such a large set of predictions? I want to say that we are in the process of organizing ourselves to do this biochemically and molecularly. And in the questions, during the question session, I can certainly elaborate on that. But I'll very quickly try to convince you through the different kinds of validation that we've done that we think most of these predictions are in fact correct. Now, I'll just show you an example of the kinds of predictions that we generate. So here is the motif for the RUNX1 family of transcription factors, RUNX family of transcription factors. This is PRDM14. CE-seq recovers this composite element, the two motifs separated by 8 base pairs, and then the orientations are denoted here. Here's another example, STAT5B with RBPJ, which is the DNA binding component of the Notch signaling pathway, and this is the particular composite element. Now, each of these position frequency matrices are generated by aligning sequences within these open chromatin regions, and each of them are hits that are found in tens, if not hundreds of regulatory sequences. So, and we know the precise coordinates of these. Not a surprise, simple motifs, as I said, tend to be small in length, whereas the CEs are longer in length, and you can compute that they have, of course, greater information content than the simple motifs from which they derive. Now, one big surprise for us was that half of these composite elements that are predicted are precisely the kinds of stereospecific juxtapositions that we had been working on before, we and others. And that's shown on the left. But the other half are an odd set of configurations where apparently the distance separating the two motifs does not matter. They show far greater flexibility, and I show examples of that on the right. And so we're calling the motifs on the left, the CEs, digital composite elements, and the motifs on the right, fuzzy composite elements, because they seem to be using a fuzzy regulatory logic in terms of their actions. Now, I'll end by just providing you 3 bits of validation. There was a very nice study about 5 years ago published in Nature by UC Tapali, where he did basically a biochemical experiment motivated, or a biochemical screen motivated by the same considerations as us. But what he decided to do was to express as many transcription factors, mammalian transcription factors, as he could in bacteria, purify each of these proteins, and do DNA binding assays with pairs of these, and use an affinity selection protocol with oligonucleotides that carry the motifs for those two TFs at variable spacings to see if they could cooperatively bind and what would he select for in terms of DNA sequence. And he identified about 300 new composite elements based on this biochemical screen. And what we can do is ask, are those biochemically delineated composite elements recovered in our much larger set of composites? And the answer is yes. And on the right, I just show you alignment of the motifs that were discovered by Topali using his biochemical affinity selection and the motifs that we recover using this systematic scan. Another thing we can do is, at this point in time, ChIP-seq assays have been done, which define the DNA binding regions of particular transcription factors in the genome. And we can ask, are our composite elements statistically enriched in the DNA binding regions of the transcription factors that have been profiled in this way? And this just shows you some examples where we see very strong statistical enrichment of composite elements in the binding regions of the transcription factors that are indicated, both in a murine B cell line and the human B cell line. And these are the lines that the ENCODE project has been using over the years. Perhaps the most interesting thing is the following, that if we take the binding regions of TFA and the binding regions of TFB, experimentally delineated, and then we ask, we're predicting a composite element for TFA and B, well, then that would argue that that particular composite element should be highly enriched in the overlapping binding regions of TFA and B. And that is indeed the case. So that argues, so the biochemical evidence is cooperative DNA binding in vitro, and the biological evidence is co-binding to a region that contains the composite element in vivo. We have 20,000 elements. And so what we are embarking now on are high throughput biochemical and functional approaches to test these. But you can, of course, do it the old fashioned way too. Here's an example of a new composite that comes out of the screen, and it juxtaposes NFAT and IRF binding sites. So this is, I think, one that Anjana would be amused by, because she had described an NFAT AP1 composite element with Steve Harrison many, many years ago. So here's the ETS-IRF composite element. Here's the new one, which we call NICE for NFAT IRF composite element. And of course, you can see here's a critical base pair that is altered to enable now cooperative assembly by a distinct pair. And again, using DNA binding assays, you can readily show that in this case, recombinant NFAT-C2 and IRF8 cooperatively recognize this nice motif. And one can show from a functional standpoint in straightforward reporter assays that the nice motif activates transcription. That's this experiment. And if you mutate the binding site either for NFAT or IRF, you impair activity, and this is the double mutant. So I think we now are embarking, and by we, I mean the larger genomics molecular biology community, on this major goal. I know that Sarah Teichmann is participating in this symposium, and Sarah, along with Aviv Regev, have led the development of the human cell atlas. And so each cell can be described quite nicely in terms of its transcriptome. But of course, that molecular description, as rich as it is, doesn't tell us how that transcriptome is regulated, how each of these distinct states is regulated. And for that, we need to provide, we need to know the codes, the regulatory codes that these cells are using to control the genes, and also the network of transcription factors that are operating on those codes. So I think that is the grand goal that we now are focused on as a community, and hopefully in the next decade, we will have achieved this. So I'll end by just thanking many, many people over the years. I've just singled out some people in this. Abraham Brass was an MD PhD student with me at the University of Chicago. He began the work on the Etzaref composite element. And I'll just fast forward and say Varen is currently a postdoc with me who has developed this computational pipeline. And there are several computational biologists that I want to thank, Matt Weirach, Tongli Zhang, Nathan Salamonis, and there are others as well that I've really enjoyed working with over the years. So I'll stop there and be happy to take questions.
Thanks, Dr. Singh. So this talk is now open for discussion, and I hope if any of you have questions, you would raise your hand. I see several questions, so I'll go to Suma first. Suma,
yeah,
you may
thank
ask
you.
a question.
Yeah, thank you, Shikha, and a fantastic talk, Dr. Harinder. My question is, are we in a better position to explain selectivity using a combination of transcription factors than with individual ones, given that the recognition regions have only a few bases?
Yes, in fact, that's been, thank you for asking that. That really has been the fundamental motivation. So I think I'm not going to argue that we can account for selectivity with these binary, these composite elements, but I think they address that problem of selectivity quite clearly in giving us, more complex elements whose activity is dependent on at least two inputs. I do think there's yet a higher level of organization of the code, a tertiary, perhaps a quaternary level. But I think what I guess I'm going to argue is that we have missed these binary elements all these years in the sense that, I mean, we've certainly analyzed some of them, but our analysis now suggests they are quite prevalent and they involve really a diverse set of transcription factors, which we are inferring from the motifs that are turning up in these combinations.
Thank you.
So there is a question by Rishi.
Hi, thank you for a very clear talk. I have a tangential question, sorry for that. So in cancer cells, where you might have like a gain or loss of binding sites or diversification of binding site, how would that be represented in your in the motif description?
Right. No, it's not a tangential question. It's actually very relevant at the moment. So our search space has been normal cells that have been profiled by the ImGen consortium. Now we can perform the same search. This tool is generally useful for any genome, and we are interested in, because as you point out, cancer genomes are highly mutable, highly evolvable, and it would be interesting to perform this kind of analysis with the accessible regions within particular cancer cell contexts and see how they are evolving. So I think it's a really interesting question. We just haven't addressed it in this case. I would add one other thing, actually, and that's those fuzzy motifs. My suspicion is that in the context of cancer cells, actually those fuzzy motifs may turn up. A lot of the variation may be in those fuzzy motifs. There's another feature that we've found about those, and they tend to accumulate P300, this critical co-activator in the genome. And so I think that's something we'd like to follow up on.
Thank you so much.
Dipankar, could you?
Yeah, wonderful talk processing. I was just wondering, how do you address the role of these interactions in different, which depends on the differentiation status of different immune cells and upon different conditions. So how do you address these functional aspects?
Right. So, so what we are doing now, it's an absolutely important question. One of the things we're doing is we are constructing a library of elements where we can assay the activity of each of these elements in isolation. Now, the library will carry these elements, and it's configured in what is called a massively parallel reporter assay format. So we can introduce all of the elements into cells of interest, these different immune cells, and assay their activity in all of these different cell contexts. That will tell us whether they have cell type specificity of the kind that you're asking us to test for. And we also will determine whether, because we have mutations of these sequences, whether they're functioning in a given cell type or a given cell state is dependent on the binding of A and B and so on and so forth. So we think we have a systematic way of exploring that functional, their functional properties.
Thank you very much.
So there'll be the last question by Abhinav. Are you unmuted?
Yeah, hi, Arinder. Great talk.
Yeah, hi.
And it's a really novel to see how multiple transcription factors actually play a role. Did you have a look into how, are these kind of composite structures actually more important in seeding super enhancers rather than just a single lineage determining factor? Because it's well known that there's only a very specific set of lineage determining factors that actually seed super enhancers. But are these composite elements more crucial in seeding these kind of long-range lineage determining regions of enhancers?
Right. So I'll say two things about this. I left out, in the interest of time, a figure that I think is quite revealing in terms of the information these composite elements carry. But the gist of it is, if we take composite elements that are prevalent in cell type specific accessible regions, and that's the only bit of information that we use, and we ask, for example, we ask for relatedness of cell types on the basis of the CE code, we can actually nicely describe the immune system without any prior knowledge of the developmental relationships between B and T cells and other cells in the system. In other words, those composite elements do carry, we think, cell type specific information. The question you ask is, are there particular composite elements that are found, in super enhancers and distinguish those enhancers from other types? Actually, we need to do that. We can readily do it. And I'm actually rather surprised that we haven't, because we think about super enhancers quite a bit. So I can get back to you within a week or so and answer that question.
Thanks a lot, Harinder. Yeah, that would be great, actually.
Okay, thanks. Let's thank Professor Harinder Singh for his very informative and engaging presentation today. And thanks for taking the trouble to join in the morning and helping us celebrate this event, very important event for our department.
Thank you. It's been an honor, and I just wish I could be with you in Bangalore, but that'll have to be another day.
We hope to welcome you here sometime in the future when we are beyond the pandemic.
Thank you.
Thanks. So moving on to the next talk of this session, it's my pleasure to welcome Professor Rachel Green from the Johns Hopkins School of Medicine. A brief introduction about the speaker. Professor Rachel Green is the Bloomberg Distinguished Professor in the Department of Molecular Biology and Genetics at the Johns Hopkins University School of Medicine in USA. She received her BS in chemistry at the University of Michigan, and she performed her thesis work in biochemistry in the lab of Jack Szostak at Harvard University, where she was studying the structure and function of catalytic RNAs and the implication of RNA molecules for the earliest evolution of life. Very interesting topic. Subsequently, she was a postdoc fellow in UC Santa Cruz, where she worked on the ribosome with Harry Noller. And there she was exploring the diverse functions of rRNA and tRNA in translation. And she joined as assistant professor in 1998 in the Department of Molecular Biology and Genetics at the Johns Hopkins University School of Medicine, where she's now a full professor. She has won many awards and fellowships, such as the Burroughs Wellcome Career Award, the RPI RNA Award for Young Scientists, the Howard Hughes Medical Institute Assistant Investigator Award, the Johns Hopkins University School of Medicine Teacher of the Year Award, to name just a few of them. Her lab focuses on diverse aspects of translation and its regulation in bacteria, yeast, and higher eukaryotic systems as well. And this includes defining also the molecular mechanisms that specify the fidelity of protein synthesis during translation. So the title of her talk is Colliding Ribosomes Function as Key Triggers for Cellular Stress Response Pathways. I invite Rachel to start her presentation and enlighten us on this topic.
How does that look?
Yeah, we can see the slide.
And how about the pointer?
Yeah, we can see the cursor as your pointer.
Perfect, thank you. All right, Shika, thank you so much for the invitation and for the introduction. I'm really happy to be talking to you this morning, and like Dr. Singh, I wish I could be there. I've never been to India and hope someday to go, but this clearly isn't the year. I also was going to add that, you know, as his life was a roller coaster on a DNA ride, mine really has been focused on RNA from the beginning of my career. And while it's been a terrible year and a half, I think it's been a pretty good year for RNA. And so I'm happy at least to have contributed in some way or to have had some role in our understanding of RNA in a meaningful way. So what I thought I'd do today is tell you one main story about how we've come across the idea that ribosome collisions are central signaling hubs in the cell to turn on stress response pathways. And to get that story started, I like to show this slide, which is a cryo-EM tomography image from Liam Holt's lab at NYU. And it's a slice of a yeast cell. And you can see in gray the organelles in the cell. In yellow, you can see some nanoparticles that his team put into the cell actually to look at the viscosity of the cell. And then false colored here in aqua are the ribosomes. And in this study, they were able to actually quantify ribosomes at 23 micromolar concentration, or 20% of the volume of the cell. And in fact, there's so many ribosomes that they directly impact the viscosity of the cell. So I think this sort of orients you to thinking about the idea that ribosomes could be central signaling molecules shouldn't be too surprising or too outlandish. So with that in mind, our lab for a number of years has been interested in protein synthesis and the mechanics and what happens when it goes correctly and what happens when it goes wrong. So under normal circumstances, the way we think about translation is that you load at the, sorry, I'm setting a timer. Oh my gosh, forget it. You'll tell me when I'm running out of time. Messenger RNAs get translated, ribosomes get loaded at the 5' end and translate through to the stop codon and get released. And we think that under normal conditions, the flow of ribosomes on a messenger RNA is dictated primarily by elements in the 5' UTR, and that flow is dictated such that there are rarely collisions. And we know, just like with traffic in the morning, that the fewer collisions or slowdowns there are, the more efficiently you get to your office. For a number of years, we've been focused on a number of different pathways that ask the question, what happens when ribosomes encounter a problematic sequence in the messenger RNA? And indeed, such a problematic sequence that the ribosomes get stuck. And what we know is that if ribosomes don't make it to the termination codon, they're going to make an incomplete peptide product, and we think that's likely to be toxic. And cells have evolved mechanisms to deal with that, to target the nascent peptide for degradation. We know that the cells also couple mRNA degradation typically to this nascent peptide degradation, so you don't keep making the same protein that's incomplete. And we got interested in this area with an interest in how ribosomes get rescued when they don't naturally reach their stop codon. And for a number of years, so, the analogy is you pull your car off to the side of the road when there's a small problem. If you have just an mRNA with a problem, you fix the problem. So we, for a number of years, wondered how it was that the cell would recognize that this ribosome was problematic and slow and stuck, And some really key experiments from Hanny Zayer's lab at Washington University showed a number of years ago that in fact, it's not that a ribosome is slow per se, it's that a ribosome is so slow that the lagging ribosomes collide, and then that collision presents a new interface to recruit an E3 ligase that ubiquitinates small subunit proteins, and that process then leads to the recruitment of all of these factors. And we and others have studied, we've focused on the ribosome collision and its role in promoting this whole set of quality control events. However, in recent years, what we've stumbled on is the idea that you can imagine that under certain circumstances where there are too many mRNAs that have damage, that the solution to the problem isn't to pull the car off to the side of the road, but instead to activate general stress response pathways to more broadly deal with a big problem. And what we found is that the colliding ribosome is a signal also to activate these responses when the machinery for mRNA-specific quality control becomes limiting. So just to orient you to the colliding ribosome, the Hanny Zayer's work showed biochemically that collisions were important, and structural biologists have given us a framework for thinking about that. What's shown here on the right is the lead ribosome that's run into the problem, where its A site of the ribosome is empty. The lagging ribosome collides, and if you look carefully at the structure, you'll see that the two small subunits come together to form this beautiful interface that we believe recruits the E3 ligase. Ribosomal proteins that are ubiquitinated are found right in this patch here, so we think this is where the E3 binds, though no one's captured it yet. And importantly, you can see here the messenger RNA extending from the lead to the lagging ribosome, and if this were space filling in a way that you could get a sense of it, you would appreciate that RNases are unlikely to be able to target this mRNA here in between two ribosomes, and that ends up being a key biochemical tool for us. But suffice it to say that these structures are real, and they've guided a lot of our thinking about how these processes take place. So usually I tell two stories, and what I'm going to do today is tell you one story. We have two main stories where we've identified factors that specifically recognize colliding ribosomes and are critical to dictating altered cell fate. And today I'm going to focus on a MAP3 kinase called ZAC that we've identified as critical for recognizing collisions and impacting cell fate. And this is entirely the work of a former postdoc in the lab, Colin Wu, who's now got his own lab at the NCI. So Colin had been studying in yeast oxidative stress, and he had identified, we had been studying the activity of ribosomes during oxidative stress, and he stumbled again on what was known, which is that oxidative stress in yeast leads to the activation of MAP kinases. However, this ends up being true in mammalian cells as well, is that oxidative stress activates the MAP kinase signaling pathways. And in yeast, he had shown that there were ribosomes that were stuck on proline codons, and we wondered if ribosome activity might be related to these events. But as we were thinking about these ideas, we called in our colleague downstairs who studies these MAP kinase signaling pathways in mammalian cells. And he says, when I think about, MAP kinase signaling pathways, the most effective mechanism that I use to activate these pathways is actually to use a collection of drugs like anisomycin, cyclohexamide, and ricin. And we appreciated when he said that these are simply drugs that target elongating ribosomes. So if you look at the typical elongation cycle of translation, a ribosome takes in an amino acyl tRNA, makes a peptide bond, and moves the mRNA tRNA complex to go around again. And in fact, these three drugs that activate the MAP kinase signaling pathways in mammalian cells all target independent steps in the elongation cycle. And so this was long ago appreciated to suggest that the ribosomes, instead of some more complex event like oxidative stress, which targets lipids and proteins and RNA and DNA, but that some simple stress that targets ribosomes, in fact, activates these pathways. And Bruce Magan's lab referred to this as the ribotoxic stress response. And so we reasoned that we as ribosome people could focus on this pathway a little bit more carefully and ask how exactly it is that ribosome targeting drugs leads to the activation of these pathways. And to foreshadow, I'll just mention that Bruce Magan's lab also identified UV irradiation as a stress that very potently activated the MAP kinase signaling pathways in a way that he argued was effectively the same as the ribotoxic stress response. And I'll get to that at the end. So with that an idea in hand, we began to wonder the same questions we had wondered for quality control pathways, which is what kinds of ribosomes activate these pathways. So to begin exploring this question, we did a very simple experiment, which is we simply treated drugs with or without anisomycin, and we asked whether the MAP kinase signaling pathways were activated, and indeed they are. So this is the positive control. We then wanted to ask the question, okay, fine, is it just ribosomes targeted by the drug that are activating these pathways, or do the ribosomes need to be moving? And we asked that question by putting in a drug called harringtonin, which binds to the initiating ribosome, but allows all the other ribosomes to run off the end of the messenger RNA. And we could then ask the question whether elongating ribosomes are critical for activating this response by simply pre-treating with the drug harringtonin and then exposing them to anisomycin. And you can see that you abrogate the response, suggesting that ribosomes had to be moving to activate the response. So with that experiment behind us, we were then ready to ask a critical question, because we, for a number of reasons, began to wonder if, like for the mRNA quality control pathways, whether stuck or instead colliding ribosomes were the key trigger for activating these cellular stress response pathways. To do this, we did a simple experiment that took a page from Hanny's book, which is the following. You can imagine that in untreated cells, ribosomes are typically not colliding. However, if you treat cells with a high dose of a drug, an elongation inhibitor, like anisomycin, what we imagined would happen would be that all the ribosomes in the cell would bind the drug and would be sharply arrested, and there would be very few collisions. However, if you treat drugs with an intermediate dose of the drug anisomycin, we anticipated that the occasional ribosome would bind the drug and the lagging ribosomes would collide, and these samples would be enriched in colliding ribosomes. We can experimentally confirm that by doing a simple experiment where you digest free messenger RNA with RNase. And in these two different samples, you would anticipate you'll have primarily monosomes, single ribosomes on a messenger RNA. As shown here, this is resolved on a sucrose gradient. And with no drug and high drug, you have predominantly an ADS population. By contrast, when you treat this intermediate sample, treated with intermediate doses of drug with RNase, what you see is an enrichment in disomes, consistent with the idea that this treatment enriches for a colliding ribosome. And with that tool in hand, we are able to ask the principal question we were interested in asking, which is it single stuck ribosomes or colliding ribosomes that activate these kinase signaling pathways? And the data are shown here, where we simply titrated anisomycin across a broad concentration range and looked at the activation of downstream MAP kinases P38 and JNK. And what you can see is that at intermediate but not high doses of drug, we maximally activate these pathways. Along the way, we began thinking about other stress response pathways that might be critical for having cells to respond to stress. And we knew that a very important one was the activation of EIF2 alpha through phosphorylation, which results in the downregulation cell-wide of translation and is a very important stress pathway. And so we asked the same question for the activation of EIF2 alpha. What you can see is, again, at intermediate but not high doses of drug, EIF2 alpha phosphorylation happens and P38 activation happens, consistent with the idea that colliding ribosomes rather than single ribosomes are key signatures that tell this, they alert the cell to a problem. So with that tool in hand, we wanted to begin to think a little bit more mechanistically. How is it that colliding ribosomes lead to the activation of MAP kinases that we think are free in solution? We actually did a CRISPR screen to think about which MAP3Ks might be at the top of the cascade. And out of that CRISPR screen for sensitivity to anisomycin, emerged a MAP3K known as ZAC, which had previously been implicated in these pathways. We made ZAC knockout cell lines, shown here. And you can see that in the ZAC knockout cell lines, we are no longer able to activate P38 phosphorylation with the application of anisomycin. In experiments that I won't show you, so this experiment suggested that this is the MAP3K that's upstream of this whole MAP kinase signaling pathway. In experiments I won't show you, we were actually able to show that ZAC is also critically upstream of this activation of EIF2 alpha phosphorylation, and that phosphorylation is promoted by one of the GCN2 kinases, GCN2 kinase itself, which is known to be implicated in sort of interactions with the ribosome and typically in responding to amino acid stress. So this was a nice set of experiments that now allowed us to get the top of the signaling cascade ostensibly closer to the ribosome. We also knew that MAP3Ks, like their downstream substrates, are typically activated by phosphorylation. And so we did a simple experiment where we asked if ZAC itself became phosphorylated as a consequence of ribotoxic stress. And those data are shown here, again, in anisomycin titration, activation of p38 phosphorylation at intermediate doses of drug. And then using a phos-tag gel, we can clearly demonstrate that ZAC itself, its migration is dramatically slowed, indicative of phosphorylation, only at intermediate, but not at high doses of drug, allowing us to suggest that ZAC gets phosphorylated at the top of this cascade. So we were excited about that. And as ribosome people, we wanted to ask the question, well, does ZAC itself, is it really the top of the cascade? Is it really the factor that directly senses ribosomes? And to ask that question, what we did was we took cells that had been treated with drug or not, and we fractionated cytoplasmic extracts across a sucrose gradient. And those data are shown here. These are just fractionated lysase across a sucrose gradient at low, medium, and high doses of drug. And we simply then probed for ZAC across the fractions. What you can see is, in fact, this MAP3K is, in fact, under all conditions, it appears to be associated with deep moving species, and in particular with ribosomal species. Is there a problem? With ribosomal species. So this was exciting at first. I can tell you that this is is there a problem? No? Okay. That this is a signature that's unusual. Proteins that interact with ribosomes. This is a very clearly a ribosome interacting factor, but we were a little disappointed initially because ZAC was migrating under all conditions, suggesting ZAC was sort of monitoring the status of elongating ribosomes. We asked a more refined question by taking these cytoplasmic extracts and digesting them with an RNase to resolve monosomes and disomes. And those data are shown here. Again, this is the monosomal fraction, and this is the disome fraction. What you can quite clearly see is while ZAC may migrate generally with elongating ribosomes, this activation event where ZAC gets phosphorylated happens very specifically on the disome peak, suggesting again that collisions are the key activator of ZAC phosphorylation that leads to these downstream signaling events. So we did a lot more biochemistry. I won't share more of it, but I'm happy to take questions on this topic. At this point, we began sort of reflecting, anisomycin is a wonderful way to activate pathways, but it's not very physiological. And we remembered that Bruce Magan had shown that UV irradiation also, he thought, activated the ribotoxic stress response. And that sounded like a more important physiological system to study. We began to wonder how it was that UV irradiation might lead to the activation or to problematic ribosome species. The first thing we did is to validate that UV irradiation actually led to the exact same activation of ZAC phosphorylation and downstream signaling. And I'm not going to show you those data, but it looks identical to treatments with anisomycin. But that didn't tell us how UV might impact ribosomes. At that point, we began thinking about what we all think about with UV irradiation. And what we all talk about is that UV irradiation damages DNA, forms pyrimidine dimers, that ultimately leads to the recruitment of kinases in the nucleus like ATR and ATM, and ultimately downstream of them to the activation of other MAP3 kinases, and downstream of them MAP1Ks. So this is clearly a pathway that's relevant to DNA damage, DNA repair, and cellular stress. However, we knew that we were activating p38 as well using UV irradiation. And I can tell you that when you look at the studies on DNA damage, these studies are done in the time course of hours. And those data that I've shown you so far are studies done at 5 and 10 minutes. So we were looking at a very potent activation of p38 on a 5 or 10 minute time scale. And we began to wonder if the DNA damage caused by UV irradiation could be similar to RNA damage caused by UV irradiation and similar pyrimidine dimers that would lead to ribosome stalling genome-wide and the activation of these same stress response pathways. And we did a number of different experiments to address those questions. These are sort of the bread and butter of my lab, and I'll just show you 2 of them, but we did polysome profiling, which I'll show you. We did deep sequencing approaches to show where ribosomes might get stuck if there were problems, and we found that they in fact get stuck on pyrimidine dimers. And then I'll show you an mRNA transfection experiment that allowed us to eliminate the possibility that signaling was happening through the nucleus. So first, I'll just show you quickly polysome profiling, where we took cells that were either untreated or treated with UV irradiation. What you can clearly see is the appearance of this disome and trisome RNase-resistant peaks, suggesting that UV irradiation does indeed cause widespread mRNA damage and the accumulation of colliding ribosomes. genome-wide. Next experiment I'll show you is the mRNA transfection experiment, which allowed us to ask the question, what we wanted to make sure that we knew was that UV irradiation, while it does cause DNA damage, isn't primarily responsible for this ribotoxic stress response that we're observing. And so to do this experiment, we simply took GFP mRNA that we either exposed or didn't expose to UV irradiation and transfected into cells and looked to see whether we activated those responses. And you quite clearly see here that in the presence, when we pre-treated the messenger RNA with UV light, ZAC is activated, as is P38, junk, EIF2 alpha phosphorylation, and GFP is in fact no longer synthesized under these conditions because the mRNA is so damaged. So with that, we were pretty excited, and this was sort of the conclusion to our paper, where I outlined the ideas I've presented, which is that under broad cellular stress, where collisions exceed the available quality control factors, what we found is that these colliding ribosomes then activate a collection of cellular response pathways to allow the cells to respond to stress. The first thing we think likely happens is that at medium levels of collisions, EIF2 alpha phosphorylation takes place, which leads to the blocking of ongoing translation in the cell and what's known as the integrated stress response. This is a cell survival pathway that allows the cell to recover. We then reasoned that when collisions get a little bit more intense, that maybe ZAC leads to the activation of P38, which is implicated in cell cycle arrest and really somewhat of a survival response. And then under really, really intense stress responses or collision levels, that ZAC then activates junk, which is typically associated with apoptosis. Shika, can you tell me how long I've been going?
Yeah, there's 1 more minute.
One
So
more
please
minute.
try
Okay,
to
I'm going to skip many slides because I really mistimed this. So I would say that this is our working model. Our working model is to understand how these 3 different pathways are integrated and how we can learn how the cell measures collisions in order to activate the correct response. And the slides I'm going to skip through are our current efforts to address that using phosphoproteomics and sort of careful titrations of collision amounts to understand how the cell might integrate these different responses in order to have the appropriate output. And so in closing, I really misjudged the talk, I'm sorry. I'll just go to my final slide. So in addition to thinking about how the cell moderates which cell response, we're also trying to understand how the cells balance this whole set of choices between mRNA-specific quality control, cell survival, and apoptosis. And we're trying to think about how that might happen differently in different cell types as well. So you can imagine neurons might have different requirements than reticulocytes, long-lived versus short-lived cells, translationally stressed or not stressed. And those are the big things we're thinking about going forward. So in closing, I'll just thank again Colin Wu, who did all the initial work on ZAC, Naladri Sinha, current postdoc in the lab who's carrying this on and doing phosphoproteomics, collaborators along the way, and thank you for listening.
Thanks, Rachel. So because of shortage of time, I will just entertain one question. I think Harinder has a question. Please go ahead, Harinder.
Yeah, Rachel, fascinating talk. Actually, can you go to your model, because something puzzled me, but your model seems to resolve that. And it had to do with the activation of EIF2 alpha versus the MAP kinases. You know, the titration experiments that you showed with the inhibitors, it looked like EIF2 alpha activation was seen at lower concentrations. I forget the inhibitors that you were using. Versus the MAP kinases, suggesting perhaps 2 distinct mechanisms of sensing. And in this model, you do invoke the idea that EIF2 alpha is activated at lower levels of this kind of ribotoxic stress.
Yeah.
Whereas
I wish it were true. I wish it were true. You may have, here's like sort of an example of a titration of collisions. And we follow collisions through ubiquitinated ribosomes, which is right here.
Right.
And you can, I think if you look at all of these, GCN2 phosphorylation, EIF2 alpha downstream of it, ZAC, P38 and junk, they don't look terribly different to us.
I
and
see.
So we're struggling actually, and we're using phosphoproteomics because it turns out ZAC has at least 10 sites, GCN2 has 20, and we're wondering whether there's cascades.
Right.
we're wondering whether the affinity for the ribosome of these factors after they get phosphorylated and the rate at which they fall off is what differentiates. But what we had hoped, as you suggest, was that we would do this titration and
Right.
GCN2 would come up first and then ZAC
Right.
would come up next. And it's turning out to be much more complicated than we had thought.
I see. Okay.
But thank you for the question.
Mm-hmm.
Thanks, Rachel, for this fascinating talk.
Thank
And
you.
also, I'd like to thank both the speakers for taking out the time and joining us on this momentous occasion of our centenary. It was lovely to listen to you. I'm sure all the students and alumni benefited from this presentation. So now I will hand over to the BIGS students representatives. Thank you very much.
Thanks to all the speakers for their wonderful talks and Professor Laloria for chairing the session. Now I request Sukriti to take over from here and proceed for plenary talks.
Thank you, Shweta. I welcome all to the third plenary talk of this seminar. The upcoming talk will be delivered by Professor Janet Thornton and the session will be chaired by Professor Nagasuma Chandra. Professor Nagasuma joined the institute in 1998. Her laboratory works on systems modeling of complex biological processes and applying them to study human health and disease, and also comparing patterns of perturbation across diseases. I would now like to request Professor Nagasuma to kindly introduce the speaker.
Thank you, Sukriti. It's a great pleasure to chair the session and welcome Professor Dame Janet Thornton from EMBL EBI at Cambridge, UK to deliver this plenary talk. When you think of interdisciplinary work, interfacing structural biology, biological chemistry, bioinformatics, chemoinformatics, the first name that comes to your mind is Janet Thornton. It appears right at the top of the list, and she's contributed immensely to shaping the field of studying protein sequence, structure, function, evolution, and how they apply in health and disease, and applying that knowledge to facilitate healthy aging, from what I can see from her recent research work. So Janet's early education was in physics from Nottingham, followed by a master's in biophysics at King's College, which I understand drew her closer to doing a PhD in biophysics at National Institute of Medical Research, London, after which she's worked at the University of Oxford, UCL, Birkbeck College, occupying prestigious chairs, and then moving on to the European Bioinformatics Center at Cambridge as its director. This is on the Wellcome Trust Genome campus at Cambridge. Needless to say, she has won several awards and a lot of recognition, including election as a Fellow of the Royal Society UK, a Foreign Associate of the US National Academy of Sciences, and Fellow of the Academy of Medical Sciences. Most notably, she's decorated with the Commander of the Order of the British Empire, CBE, and later on, the Dame Commander of the Order of the British Empire in 2012. Okay. For me personally, it's a great privilege to welcome Janet to this session. I wish we were doing this in person. Although I have not had the opportunity to work with her directly, I think I have benefited from her work enormously. In fact, that has been inspirational for me to get into this area. So in other words, I've been indirectly guided by her by reading all her papers and following her work closely. And she continues to be at the frontier of this field even today. And today she's going to talk to us on enzyme flexibility, transformations, and mechanisms. May I now invite Professor Janet Thornton to the stage, please.
Thank you. Okay. Can you see my screen? Can you hear me?
Yes. Brilliant. Yeah, all good.
Brilliant. So first of all, I would like to thank Suma for such a kind introduction. But I'd like to congratulate all of you in the biochemistry department at Bangalore. 100 years is a long time, and my goodness me, has biochemistry changed over those 100 years? And we have lots to celebrate. You have lots to celebrate. And I just, as I'm sure other speakers have said, it would have been wonderful to have been there with you. but that's not to be today. I'm going to talk about computational enzymology, but before I do that, I'd just like to dedicate this talk to Professor Srinivasan, who, as you know, sadly passed away not very long ago. For me, he was my direct link to Bangalore, and I visited him several times there. I knew him, of course, when he was at Birkbeck College at the same time I was at Birkbeck College as a young scientist. And he really, to me, encapsulated a true scientist of the very best kind. Of course, he was superb. I'm not going to go into his scientific achievements. Suffice to say that.
Oh, Janet, sorry, I think you got muted. Sorry to interrupt. Janet, I think you're muted.
Let me unmute. I don't know how.
I think it's now on.
That's okay.
Yes.
So when did that happen? Did you, oh.
In the last slide, when you're talking about Srinivasan.
When I was talking about Srini. Okay. He was a total scientific enthusiasm and a really dedicated teacher and supervisor. I've had the fortune to work with many of his, or some of his proteges, and they always speak so warmly and kindly about Srini. He was this picture to me, which I took when I was in Bangalore last in, it's now almost, well, when I was in Bangalore in 2013. Of course, he was a dedicated family man, and our hearts go out to Minnie and Jayashri. And so to me, I really wanted to dedicate this talk to him and to remember him. So the talk is about enzymes, and I wanted to start off with a bit of a historical perspective. In fact, enzymes were used in food production 7,000 years ago, well, more than that now. It was used to make cheese and bread and alcohol, and you can see here in ancient Egypt and also in Japan making sake, and I'm sure in India it was used in some similar ways. In the 1800s, enzymes were really first discovered. The term catalysis and enzyme were coined, and cell-free fermentation was observed towards 1900. And during the 1900s, enzymes were really characterized in great detail. All the curly arrow diagrams describing mechanisms were derived. The metabolic pathways, the complicated metabolic pathways, were derived during this period. And of course, we began to get sequences and structures of proteins, the first enzyme structure being determined just before I joined the lab in Oxford of hen egg white lysozyme by Sir David Phillips. And now in the 2000s, we really have enzymes galore. We have huge amounts of information about enzymes. We know that on average, 25 to 50% of proteins coded by the gene, by an individual, by an organism's genome are enzymes. We have many, many sequences. This is now somewhat out of date. And also we know the reactions, which were the beginnings, if you like, of enzyme studies. People use quantum chemistry to analyze their mechanisms, and recently a few successful enzymes have been designed. And in looking through to give this talk, I noticed that enzymology and bioenergetics were central to the department. It said enzymology was a major thrust area in the department right from the beginning. So this feels like an appropriate subject to think about. But despite all that knowledge, there are still a lot of questions that aren't answered. How do enzymes work? They work at neutral pH often, and they work extremely well. This is the green chemistry that we need for our sustainable world. How does their function evolve over time? Can we predict transformations and mechanisms? And can we predict specificity and function from sequence and structure? So these are academic questions that I think many of us are interested in, but there are also a huge number of applications that will be made easier by understanding more about enzymes. We know that many drugs target enzymes. We want to design new enzymes for the benefit of mankind. We want to redesign pathways so that they work to produce the molecules that we are interested in. And of course, the idea of having plastic processing enzymes that can help us to remove all the plastic that's both in the oceans and in landfill sites. And so this area, I believe, remains one of great interest. And as I say, they are very relevant to medicine, in particular drug targets. About 54% of all drug targets are enzymes. Most of those are to humans. And you can see here how they're distributed amongst the different type of enzymes that we know from the Enzyme Commission. And so if we look just in 2020, in fact, of the top of the novel drug targets, I think there were about 13, 5 of those targeted enzymes. And so they remain a classic target for novel drug design. So what do we want to do in my group? We really want to capture, to analyze, to validate and ultimately predict enzyme mechanisms and to discover how enzyme function in a family changes during evolution. We know that one family of enzymes will evolve over time to form to have different members, and those members often have different functions. Quite often they have no enzyme function at all, and that is also interesting, the pseudoenzymes. So I'm going to talk a little bit about how we capture the data. I'll talk about how enzymes evolve and what we've learned about that. Then I'll go on to finding enzyme transformations and also trying to predict enzyme mechanisms using an informatics approach. And lastly, I briefly talk about looking at some of the AlphaFold models that have been generated and asking whether they can help us to predict function. So I'm sure that all of you in a biochemistry department are well familiar with the enzyme classification scheme. But for those who perhaps aren't so familiar with it, just to remind you, there are 7 different classes of enzymes denoted by the primary EC number here, the class. There are then 3 other numbers. The middle 2 define a subclass and subclass, and the last one is a serial number. This is very useful computationally, and in fact, we began our studies on enzymes now some 30 years ago because of this Enzyme Commission scheme, because it's a very good way, it has its problems, but it is in principle a very good way to capture the function of an enzyme. And it defines nothing to do with the protein, it just defines the small molecule reaction that is catalyzed by an enzyme. Of course, since the Enzyme Commission was set up, and this is all done manually by a group of enzymologists, there are also many other databases that have been critical in gathering some of the data. I've already said that the percentage of enzymes in genomes varies between about 25% up to 54%, and is usually higher in bacterial organisms. And they distribute between the different classes, as we see here. The major classes, the hydrolases, the transferases, and the oxidoreductases, really occupy more than 3/4 of all enzymes, and there are smaller numbers of the other 4 classes. So when we started this, of course, my interest for many years has been in protein structures. And we really, within the protein data bank, the PDB, there were coordinates of enzymes, there was the name of the enzyme, but there was very little else. And we used that to look through papers and identify the catalytic residues. So this became the Catalytic Site Atlas, which has since evolved into the MCSA, which is our database, which is available online. And I think it's a great teaching tool, actually, to teach students about how enzymes work. But in the MCSA, we now have information for the enzyme reaction, the set of reactants and products. We have information about the protein in its sequence and its structure and its homologous family and also the species. We have information about cofactors, often metals, but other cofactors, organic cofactors. In particular, we captured information about those residues that are directly involved in catalysis, the catalytic residues. And this database has been updated over the years by students. We have interns in summer. This year it was done entirely remotely, and each intern tries to annotate from the literature the information about the 4 areas that I've just described that are in the MCSA. So we have about 1,000 entries, and each one has a reference PDB structure. Of those, about 680 odd. Now, in fact, this has grown and is slightly larger, about 700 detailed mechanism descriptions. And although this doesn't seem so large, in fact, it covers about 80% of the EC numbers down to the 3rd level, i.e. it doesn't always cover all the substrates, but it covers the reactions that have a structure in the PDB. And we can use these reference structures to, of course, annotate millions of UniProt sequences that are close homologs and to annotate the 50,000 PDB enzyme structures. And this is all freely available. You can go and look at online at this database. And so, as well as having the information about the sequence, the active site, the structure, the reaction, we also have information about the mechanism, i.e. how this protein, this enzyme, catalyzes this reaction. And this is described in the famous curly arrow diagrams beloved of biochemists and enzymologists that basically tell you how the enzyme catalyzes this reaction to change, in this case, isocitrate to oxoglutarate. And I'll come back to this. So we have this large database of information, and we can look at it and we can ask very simple questions, such as how many of our 1,000 mechanisms involve each type of amino acid in the catalytic role through a side chain. And what you see here is the distribution of the 20 amino acids and how many in our database of about 1,000 enzymes involve histidine, aspartic glutamic acid. You can also see that the vast majority of the residues that are involved are either charged as being the most common, or also the polar residues. As you might expect, the aliphatic residues, the hydrophobic residues, are less involved. And when we normalize according to the frequency of amino acids in the active site, we find that actually histidine, asp, GLU, and cysteine, the size of the circle represents how significant it is. And we find that these residues really do dominate catalysis. Oops. We can also say, we know that sometimes it's the main chain, the backbone, carbonyl and NH group that's involved in catalysis. And when we look at the residues that use their main chains in catalysis, we now see a completely different distribution. And what you can see it here, that the dominant residue type is glycine, followed by serine, alanine, cysteine. And when you look at these, as you can see here, when you normalize the data, you can immediately think, well, actually, this depends on the size of the amino acid. If you have a small side chain, then the backbone, the NH and carbonyl, are much more accessible to be involved in catalysis. So we also looked at what sort of catalytic roles do these amino acids perform. And we separated them into 3 types. One, and probably the most important, is the reactant. That's those residues that are directly involved in the change in the substrate, that are directly involved in the chemistry, if you like, the reaction. We also have some that are interacting, and we have some that are spectators, albeit important for catalysis. And here we see in particular the electrostatic stabilization is very important. But you can see when you look at the reactant role, the dominance of proton acceptors and donors. Sometimes I think that the enzymes really just work by moving the protons around on the ligand. That is by far the dominant sort of reaction that occurs. And we can not only do that, but we can assign a given reactant, such as proton acceptor, we can find which are the most common residues of the 20 residues. So we begin to develop a sense of the way that proteins work. Proteins perform these amazing array of catalytic reactions with a relatively little catalytic tool set. They have the 20 amino acids and they have the cofactors. I'm not going to talk about cofactors today, but you can see here that if we're looking at proton acceptors, that the histidine, glutamic acid, and aspartic acid again dominate. But you can also see that cysteine can not only be a proton acceptor and donor, but it can also do a whole variety of other reactions that occur in enzymes. So cysteine is, it can do many things, more perhaps than His, GLU, and ASP can do. And we can see that if we look at electron acceptors and electron donors here, that most of these amino acids are able to be involved in that sort of a reaction. So we have the beginnings of a table that tells us what the amino acids do during catalysis. Okay, so I'm going to, if you like, diverge temporarily to talk about what we learned about evolution of new enzyme functions. And I'm not going to mention the non-catalytic, the pseudoenzymes, because there really isn't time for that. And so here, the question was, if we look about what we know about enzymes today from UniProt, basically, and also from the literature, what can we learn about the way that enzymes evolve new functions? So they change their active sites in order to do a different transformation. How do they do that? What's involved? And how many times does that happen? So we basically use public data for this. And we selected all examples where we found a change in functional, in the function of the enzyme, as measured by its EC number. And we identified those sequences associated with novel functions. We only did this for small molecule metabolism because it gets complicated for macromolecular enzymology. But we only use those functions that have been validated experimentally. And I should say at this point, we exclude the non-enzymes or the pseudo-enzymes because frankly, it was difficult to be confident in these annotations. And so this is just one example of what we did. We grouped the enzymes into families or superfamilies. And in this family, the PIPDE superfamily, this is a very large family of enzymes. It's found in almost 3,000 species. We have 26 members of this family in our genome. We have, at this point, when we did it, and we started this some time ago, there were 39 structures associated with members of this family. And those structures included 18 different enzyme functions and 212 GO terms. So their enzymology was diverse and their role in the body was even more diverse. And so in this is one example, phosphoinositol phospholipase C. And in this example, this member of the family, its enzyme number is 3.1.4.11. What this enzyme did with the help of magnesium was to cleave this molecule at the OP bond to create two products as shown here. So basically this, it cleaves as a hydrolase, this PO bond. And when we do the evolutionary tree based on sequences and picking out where we have these changes in function as defined by the EC code, we find that there are a lot of different EC numbers. Most of those are 3.1.4. That's some sort of OP, cleavage of OP using a hydrolase function. But we also see that there are some liases here, 4.6. So they're in a different class altogether. Now the three that you can see, the two pink here and the blue, we had structures of those with some ligands. You will also note that this one needed magnesium, this enzyme needs calcium, this enzyme doesn't have any cofactor at all. And so we looked at their three structures that you can see here that I've now pulled out. You can see them superposed on the left here, a classic tin barrel. And you can see also that when we look at these, we can construct this matrix of functional changes as classified according to their enzyme commission numbers. So we can see here that we have a 31446 changing into a 3144. That means just that the substrate has changed. And so we have 4 examples of this hydrolase changing into a different hydrolase, a different substrate, if we look across the whole lot. We also have an example of a liase, a 4, changing into a different liase here. So we've got a 4, EC4 going to EC4, and we have one example where we have the hydrolase changing into the liase here. And so we have one here. So we can construct this matrix of functional changes. And this is just for one of our enzyme families. We did this across 276 superfamilies, and this was very much a collaboration with Christina Rengo using the CAF database that we've worked on. She's led and I was involved originally in setting it up. And what you can see is when you combine this information creating this exchange matrix, most of the changes that we see lie along the diagonal. That is, that most changes are for a change not in the substrate, not in the class, the primary class of the enzyme, but in a more subtle aspect. And so you can also see that in the off-diagonal terms, which mean you have a change in the primary class of the enzyme, we observe changes in every box. And so we can say that during evolution, it's possible to change almost any enzyme into another enzyme class. And we can represent this. This is shown here in more detail. We can say, for example, for the isomerases, we can say that an EC5 isomerase there are examples where it has evolved to be an oxidoreductase or a transferase or a hydrolase or a lyase, and also the ligases, the EC6. And in fact, you can represent all of our enzymes by these diagrams like this. And this gives us a functional overview. It was, we put this together some years ago now, of evolution of enzyme function. And it shows, first of all, how that it's quite common, even though functions are often retained in the orthologs of genomes, but you also get these functional changes. But you also see that, in fact, most often, the number of changes in substrates is much larger percentage-wise. 89% of these changes only change the substrate, and a much smaller number change the reaction chemistry. And so it's clear that during evolution, it's much easier to tinker with the binding site, to change the substrate, but the chemistry is very often conserved, but occasionally it does change. Okay, so we were keen to look at, you'll be familiar, I hope, with the work of Chothier and Lesk, who showed that as the sequence identity between two proteins dropped, similarly, the structural identity fell off as the sequence became more distant. And we asked the question, well, suppose we look at sequence identity and functional similarity. Are we going to see the same, are we going to see the same sort of relationship here? And we thought we could measure functional similarity as substrate specificity. How similar were the substrates in these families? So we thought we could look at this, and we were hoping to see for each family some sort of a drop-off between the sequence similarity of the members and the functions that they perform. So this was our hypothesis. I should say that when we plotted these things out, this was what we got. And this was one of our not best research days, shall I say. Because you can see that although on average, there is a correlation as the sequence identity falls, the similarity of the substrate that the enzyme works on drops on average. But you can also see that there are lines all over the place, and it isn't a beautiful, clean picture as we hoped it was. And to some extent, this has driven us back to look at families perhaps in more detail and to understand more about the way that enzymes work. This really drove us back to basics, if you like. So there's no simple correlation, we felt, and we needed to look in more detail. So in going back to basics, I'll just describe three different sort of strands of work. One is looking at flexibility and how enzymes use flexibility in catalysis. So we wanted to just ask, to what extent are enzymes flexible in their enzyme active sites? And so we can use enzymes with multiple structures in the PDB to investigate their flexibility. So we used all the MCSA annotation, the PDB annotation, and also the RIA and KEGG databases and our cofactor database. And we reconstructed, if you like, in 3D, we superposed these active sites and asked whether they were similar. So we compared the sequence and structural similarity in enzymes, and we found that the whole structure and the active sites follow very clearly different trends. And I'm afraid this is drawn the other way around from what I showed you before. So if we looked at the overall similarity, so this shows one example where the sequence similarity is just 15%. And we find that as the sequence similarity between members of a family decreases, the root mean square deviation for all the alpha carbons gradually increases. And you can see this here, a clear line, fairly clear cut. But of course, when we look at the active site structure, the picture is extremely different. So now we're just looking at the catalytic residues. And what we find now is, first of all, of course, that these are immensely well conserved, that they don't, even as the sequence of the whole protein diverges to be less than 15% identity, even at very low sequence identities, the structure of this catalytic site is extremely well conserved, with the average RMSD being less than 2.5 angstroms, even down at less than 20% sequence identity. So we know that these are very, very well conserved. And we've been through analyzing about 307 different enzyme families. And we actually found what we described as 4 different paradigms of flexibility. Many of them, 42%, using the data we've got at the moment, the active sites really were completely rigid. And that means less than 0.5 of an angstrom RMSD between the catalytic residues. So we have the largest fraction, in fact, is very rigid. We also find that some are more flexible. They're not radically flexible. The RMSD is shifted, but they have more flexibility. They're not so rigid. Whoops, sorry. Others have this open and closed form, where you have 2 distinct conformations when the ligands bind. And others, again, have a lot more flexibility and are extensively variable, but that's quite a small fraction. So we're in the process of looking at this in more detail. This isn't yet published, but we hope it'll be submitted very soon. But we also, I said we wanted to go back to basics, and flexibility was one aspect. The other aspect was changing from the biology to the chemistry and using cheminformatics tools to learn how enzymes work. So can we use our knowledge of enzymes to predict what they do, their transformations of small molecules? So given an enzyme sequence and structure, can we predict its substrates and products? Now, the fact is we can't do that yet. And I think this is a very important area of research to consider. And our aim was to combine the power of cheminformatics to handle small molecule reactions, if you like, and reactions, and combine that with our knowledge about enzymes, their structures, their evolution, and how they work, looking at the roles of the different amino acids. And 2 particular things we've looked at. One is the transformations, i.e. what happens here. The other is the mechanisms that are used. So for the transformations, John Tyzak, a postdoc in the group, worked on something called Transform Miner. And the idea was, if you provide a molecule, you want to find those enzymes, transformations, that act on a given source molecule. The other question was, you want to find a path between source and target molecules using enzyme transformations. Again, this is available as a web server that you can look at. So we know that we have a lot of diverse enzyme chemistry. We know how to handle the data. We can generate these reaction files. And we can also provide atom-atom mapping from, if you like, the substrates to the products from the reactant. So we can map this oxygen corresponds to this oxygen over here. You need that atom-atom mapping. So these are molecular equations. You conserve the atoms. You don't lose atoms. You conserve them. And so that's one way to look at reactants. And so we had to, we use reaction decoder toolkit that Asad Rahman in the lab had developed now probably 15 years ago. And we use this way to look. So you can, in this case, you can submit a molecule, in this case it's propanol, and you can find what that molecule might be transformed to by which enzyme. So here, this gives the transformation. And on the right here, you see the reaction and also the EC number that transforms this molecule into this molecule here. So this gives you a way to, if you like, mine reaction space, to look at the chemistry of this reaction. And you can see, in this case, there is one enzyme here that clearly will perform this transformation, but there are also quite a lot of other enzymes that also likely would be able to transform this molecule, in this case to this molecule, or in this case to this. And you can look at those enzymes that might perform that reaction. You can also start with a source molecule, the same one again, propanol, and you can say, I want to find a way using enzymes at each step to transform this molecule into this target molecule. So you can find a pathway, and in this case, you can see there are several pathways that would allow you to form this. And it's just based on the similarity between the substrate and the substrate of a given reaction or a given enzyme that you know about. So this is one way to delve deeper into the transformation capabilities of the enzyme. And we're using this. John is working to identify opportunities for enzyme repurposing using various filters, for cleaning up wastewater, etc. The other question we wanted to look at was enzyme mechanisms. And you can ask, well, what is an enzyme mechanism? It's the movement and interactions of atoms in the active site of an enzyme that drive the transformations of the reactants into the products. So here you can see you go from step one, I think this goes step one, step two, step three, step four, and you can follow it round. So this is what you want to, given you know. So what we wanted to do was to create an algorithm that, given an active site and the reaction, will suggest possible enzyme mechanisms based on the information that we've got stored in the MCSA. So we know the active site, we know the reaction. Can you predict this curly arrow diagram, the enzyme mechanism? So Antonio Ribeiro, a postdoc, is very actively working on this. And basically the way he's done it is, first of all, to extract the rules, then to create the algorithm to apply the rules, then to view the predictions and to see if they are correct or not, and then to apply to all the MCSA entries that we've got and check if we can get the correct mechanism. So Antonio is actively working on this. I would say I'm hoping that he's 9/10 done. We keep finding more difficulties every step of the way. It's quite a long and tortuous route, but we think that this should be possible using the mechanism. Antonio is a quantum chemist, and he is always very keen to point out that all we can do is suggest possible mechanisms, and we would have to use much more sophisticated quantum mechanics tools to look at the energetics to see which is the mechanism, rather than actually define a given mechanism. So, Suma, have I really almost run out of time? I really have no idea, because I'm not sure what time I started.
No, I think we're good. We've got 15 minutes. We started a bit.
We've still got a bit of time. Okay, good. So I wanted to finish off by talking a little bit about the AlphaFold database and the impact that that is likely to have on our world of proteins. So as you're aware, DeepMind created a complex pipeline involving various types of neural networks and model building tools to predict protein structures from sequence. And this time last year, actually, it was all excitement because the announcement came from Cas14 that their pipeline had been extremely good at predicting accurate structures, and you can see some of them here. And since that time, so it's sort of 1 year on, DeepMind decided to release coordinates for their predictions, and they did this in collaboration with EBI, Embl EBI, and a database was released in July. And in fact, I think it was yesterday, this database was updated so that there are now almost 800,000 structures covering the whole of Swissprot, so that you have now double the number of structures that we had before. And remember, in the PDB, we only have structures for about 55,000 unique sequences. So this is really having a huge impact in the number of potential structures that we have. These structures are open access, they can be used by everybody, and they can be downloaded. And we have, this number is slightly out of date, but since the database was launched, we have a huge number of people who have used these structures. And also, well, this, you will be aware that they are planning to release about 130 million structures. That will probably be in the middle of next year for the whole of UniRef 90. So we will have effectively structures for most of the sequences, or at least representatives of those sequences that we know. So in terms of the AlphaFold models, if you've not looked at them, this shows one example of an AlphaFold model. The models are annotated, each residue according to the quality of the prediction. We know that some parts are very well modeled, as shown in blue. The yellow and the red parts are much less accurately predicted. And so you have to beware about those regions. When you compare that to known structures, they are often extremely accurate. They are the best homology models, if you like, that they have. The root mean square is deviation for the C alphas for many of these models to the known structures is very, very low. So for these, the average for this particular protein, the average confidence score is called a PLDDT, is about 90. And that is a really confident prediction. However, there are many proteins where we had no PDB structure, and this shows one of them. And in this case, the predictions are less than less than, I don't know whether convincing is the right word, because this protein almost certainly is a flexible protein, it's dynamic, and it interacts with other proteins before it adopts a unique structure that we're used to with globular proteins. And so in this sort of a protein, the confidence score is much lower. The PLDD score is only 66. And that means that actually most of this protein, the predictions are not. There is not sufficient information to make a good prediction. In fact, when we look at these models, what you can look at those proteins which have homologue in PDB and look at their confidence score, PLDDT, and you see that for those proteins, the majority of them have confidence scores over 60, over 80. They're very good predictions. For those proteins where there isn't a corresponding PDB, the predictions are much lower, but the reliability, the confidence in those predictions is lower. And that in part reflects the fact that some of these proteins are flexible. It reflects the fact that some of these proteins belong to very small families, and there's not enough data really to make confident predictions. But what about enzymes and the enzymes that are predicted? So if we look at human proteins, we find that about 26% of the proteins in human, in the PDB, are enzymes. So that's quite a small fraction, but that's human has less enzymes than, as I said, than the bacteria. If we look at humans with no structures in the PDB and no easy models, then we find about 5,000 proteins. And of those, some of these are actually enzymes. And so we can ask for these enzymes, can we make any predictions? And so we began to use these models to search for enzyme functions using our catalytic templates that we derived some time ago. And we derived those for, in this case, you can see the catalytic triad, but we also have them for ligand binding residues and DNA binding residues. And we can use these templates to scan each model to see if it provides a region which matches one of the templates. We can then highlight the best hits, and we can potentially make some prediction from the similarity about what these enzymes might do. And in fact, this was built with ProFunc, which is a server that we developed many years ago in the structural genomics era, when there were many structures without any functions. And this is the same sort of situation. So we have lots of enzyme templates, in fact, 3,500 of those, involving just 3 residues in this case to define what the active site looks like. And so you can, this is one example. You can see this is the AlphaFold model for the human transmembrane protease serine 2. And you can see here, they know already that it performs this 3,4, so it's a hydrolase, although they don't know what the substrate is. And when you use the catalytic triad template, you find you get an extremely good match of these residues. The RMSD of these all these atoms is only 1 angstrom. It's very small. And so in this case, this template corresponded to 1 RTF, the catalytic domain of tissue plasminogen activator. And when you looked around this catalytic template in this AlphaFold model, you found that much of the structure was the same as you saw in this PDB structure. And in fact, we had 26 identical residues within 10 angstroms of the catalytic center. So in this case, this shows an example where these templates were able to pick out what clearly is a homologue of a serine proteinase, but it's a distant homologue where only the catalytic site has been conserved in some ways. We also have ligand binding templates, in this case for a cofactor, PLP. We have different set of templates. We have DNA binding templates, lots of those. And we also, Roman also developed this reverse template approach, where you take a query structure, such as an AlphaFold model, you identify the conserved residues, and from those you derive a set of 3 residue templates, and you then scan the whole of the PDB for examples of these 3 residue templates. And so this is a set of proteins, human proteins from AlphaFold, where there was no Swiss prop model. In total, there were about 1,000 of these. And when we took our templates, what we found was that we could find certain enzyme templates hits, 13 in total. And we also found 14 probable, but not absolutely certain hits. And we could do that along here. And so I think this shows a way that the 3-dimensional predictions can be used to help to predict function. And in this case, if we look at enzymes with no templates in the database that can be identified by reverse templates, this is one example, sphingolipid delta 4 desaturase, there's one. And we look at this model, we know what it does, we have its enzyme number. If we did the sequence searches against PDB, we got nothing, no structure of an enzyme with that particular EC number. But we did get a reverse template match to this particular PDB code, which you see has the same first 3 EC numbers and a 1 there. And this template corresponds to a region where 2 zinc ions are bound. And this was present in this AlphaFold model and also in this particular enzyme structure. And so in this case, we have a region that is where zinc ions are coordinated by histidine residues. And when we look at this enzyme, what we find actually is that, well, if I go further on, the bottom line is that both proteins, this one and this one, have similar, though not identical substrates. So you have a change here in the substrates. And so this is one way that we can use these structures to say something potentially new about function. So that really concludes. I've talked a lot about a lot of different things about enzymes, but there's still a lot to do. It amazes me how detailed one needs to be. And we really need to look at different enzymes and enzyme families to understand more about whether we can make these predictions. So to conclude, I'd like to, first of all, acknowledge the people in my group, in particular, the people along the top here, Antonio, John, Yanis, Roman, and Neera. And also in the past, these 4 people who really made a huge impact. There are others as well, I should say. Perhaps these are more recent ones who've really helped in doing this analysis. And I'd also like to thank Christina Rengo, my long-term collaborator, who runs the CATH database and has done some brilliant work on using that to help to interpret function. So if I could just close by lastly remembering Srini. I wish he'd been in the audience, and I hope he would have enjoyed the talk. And now thank you to you, and I'm very happy to answer any questions you might have. Thank you.
Thank you so much, Janet, for a wonderful talk. I'm sure there are questions. Sodamini?
Hello.
Oh, hi, Minnie.
I am fine, thank you. Thank you so much for remembering Srini and dedicating this wonderful talk in his memory. I am deeply moved. And also, thanks for a brilliant talk. It was great to see the smooth flow. And I had one question, if I may. It is to do with the EC classification, because looking at, I'm sorry, I'm in the dark, but looking at the functional residues, I wondered if you had any situations where you had to differ from the ascribed EC classifications. You know, looking at a functional residue, you would think it is some other EC number rather than what was ascribed in the database.
So I don't think we not really looked at it in that way. We looked at some years ago now, we tried to look at the EC classification, first of all, to understand it, because, you know, it's very complicated. And you look at them and you think, well, I'm not sure. The other thing is that each class it's not self-consistent. So you can't use the EC classification to sort of navigate between classes. It doesn't make sense, which is very frustrating. However, when you then go and look at a given enzyme and what it's called, you realize why the Enzyme Commission have put it in that class, and you realize that there are compromises to be made. It's often difficult to know exactly where to put a given enzyme. And so we came to the conclusion, and we did actually try to write some papers about how we might change the Enzyme Commission nomenclature. But those weren't very well received, because I think everybody quite rightly felt, and I feel also, we have this wonderful classification. We can use it for what it's good for, but it's no good for navigating through enzyme space. You have to do it at the level of the reactions and look at what's going on. So that's how I mean, that's a long answer to your question, Minnie, but it is really complicated. And we did try to improve things, but we felt we couldn't really, and that it was better to leave it as it was, but develop tools to navigate through the space, which is what Transform Minor is, and some of the search things, EC Blast, that we did some time ago.
Thank you so much.
Yeah, thank you, Minnie. It's lovely to see you, and take care.
Yes, can I ask?
Nagsuma, this is Rangarajan here. Can I just intervene?
Yes, please. Yeah.
We are running behind the schedule, and the next speaker is waiting. Sorry, so we have to curtail the discussion.
Sure. Yeah, there are many questions, and we can pursue them at a later time, perhaps. Thank you so much, Janet. Really appreciate you taking time to join us in the Centenary celebrations. It was a great pleasure to listen to you. Thank you so much.
Very nice to see you. Thank you.
Thank you. Over to the BICS organizers.
Thank you, Professor Janet, for this wonderful talk, and Professor Nagsuma for chairing this session. We will now proceed to our next plenary talk by Dr. Vishwa Dikshit. This session will be chaired by Professor Sandeep Eshwarappa. Professor Sandeep joined the department in 2015. The primary interests of his lab include understanding the mechanisms and significance of translational read-through events in mammals and mechanisms of angiogenesis. Another goal of his lab is to study resistance mechanisms to extreme stress in tardigrades. I would now like to request Professor Sandeep to kindly introduce the speaker.
Thank you, Sukruti. Good evening, everyone. It's my pleasure to introduce Dr. Vishwa Dikshit. Dr. Dikshit completed his medical training, that is MD, from University of Nairobi. He is associated with Genentech since 1997, and currently he is a vice president of early discovery research at Genentech. Actually, he doesn't need any introduction, especially to those who are working in the area of inflammation, because he is known for his pioneering work in inflammasome biology. Some notable contributions are demonstration of caspase in component of FET receptor-induced apoptotic pathway, showing the importance of mid-88 as a key adapter in innate immune signaling. He is one of the highly cited scientists with more than 124,000 citations so far, and with an 159H index. He is a member of National Academy of Sciences, AAAS, Royal Society, and many other prestigious societies. Dr. Dikshit, it's a great pleasure to have you at this meeting, and I look forward to your talk. Over to you.
Great, thanks. So let's just do an audio-visual check. Everybody can see my slides and hear me okay?
Yes, we can hear you also.
Great, well, it's a tremendous honor to be giving this talk to celebrate the 100th anniversary of the biochemistry department at Indian Institute of Sciences. What I'll do today is discuss an aspect of my talk, of my work really, that has to do with an important question in cell biology, which is that how are leaderless cytokines released? So by leaderless cytokines, I mean cytokines that don't have a signal peptide, but they gain access to the external milieu. So they're not going through the Golgi apparatus. So how exactly are they getting to the exterior of the cell? This is a very important question in immunology because a number of very potent cytokines, like IL-1 beta and IL-18, cytokines being entities, proteins that are going to orchestrate the inflammatory response, they do not have a leader sequence. So that's the question. Now, in order to address that question, I must introduce you to the inflammasome. And the inflammasome is a cytosolic apparatus. It's composed of a sensor, an adapter, and an effector. It is present in all cells of the innate immune system, so macrophages, dendritic cells, and other cells as well, like endothelial cells. And it protects the sanctity of the cytosolic compartment. So if the sanctity of the cytosolic compartment is challenged by pathogens, then the inflammasome is activated. What do I exactly mean? I mean that a number of pathogen products like flagellin, nucleic acids, toxins are detected, their presence is detected by the sensor. Also, insults that mediate sterile inflammation. So this could be protein aggregates, as you see in neurodegenerative diseases, could be aggregates of chemicals like uric acid. Those too are detected by the sensor. Mediators of sterile inflammation like ATP that activates the purinergic receptor P2X7. That too is detected by the sensor. So essentially the sensor is the police person monitoring the cytosolic compartment. Now the sensors themselves are, they largely fall into a family termed the NOD family of sensors. There are about a dozen of these in the mammalian genome. There are over 200 in plants, and in plants they form the central backbone of their immune response. Generally, the sensors have a prototypical structure. They often have a protein-protein interaction motif. Here I've just showed a sensor NLRP3. It has a pyrin domain, a nucleotide binding domain, and a series of leucine-rich repeats. How exactly these sensors sense the insult? I mean, that's a story for another day and a story of much activity. But suffice it to say that once the sensors sense the insult, they form this complex between an adapter and the effector. And the effector in this case is a protease, a caspase-1. So just to remind you, caspase-1 is a member of the caspase family. In fact, it was the first caspase. These are cysteine proteases, but they have this very unusual substrate specificity in that they cleave after aspartic acid residues. And generally, you can divide caspases into apoptotic caspases that mediate apoptosis and inflammatory caspases that mediate inflammation. And so this is a protease, caspase-1, that's an inflammatory caspase. And in fact, its original name was interleukin-1 converting enzyme. Regardless, once this complex is formed, which was dubbed the inflammasome by the late Jörg Chopp, caspase-1, which is a single polypeptide zymogen, gets activated to give you the catalytically competent protein that's composed of two subunits, P20 and P10. It's actually a tetramer, dimer of dimers. But for our purposes today, let us appreciate the fact that the zymogen is getting processed in this inflammasome complex, and you're getting an active protease. Now, this protease is going to proteolytically process pro-IL-1 beta and pro-IL-18 to give you the immunologically active cytokines, IL-1 beta and IL-18. Now, here's the problem. These cytokines, even after proteolytic processing, don't have a leader sequence, don't have a signal peptide. So how do they get out of the cell? Well, it turns out that they get out of the cell because a second role for caspase-1 and for inflammatory caspases is to induce a form of necrotic death termed pyroptosis, in which the cell membrane is perforated. And this perforation of the cell membrane allows for the cytokines like IL-1 beta and IL-18 that don't have a signal peptide to exit the cell and gain access to the extracellular milieu. Not only these cytokines, but there are other mediators of inflammation like IL-1 alpha that doesn't have a signal peptide, HMGB1, histones can contribute. So, and these mediators of inflammation that are released from self are referred to as mediators of sterile inflammation or DAMPs, danger-associated molecular patterns. Now the story gets a bit more complicated. Actually, the literature is quite complex, and I'm at the risk of oversimplifying it, but for our purposes today, that's fine. Now in 2011, we discovered what a non-canonical pathway, which seems to be a dedicated pathway in which the presence of LPS, which you know is the predominant structure of gram-negative organisms, that is detected in the cytosolic compartment, and it engages caspase 11, another pro-inflammatory caspase. But importantly, caspase 11 works through caspase 1. So in the final common pathway to the proteolytic processing of IL-1 beta and IL-18, we only have to worry about caspase 1, because that is the caspase. Whether you activate the canonical pathway or the non-canonical pathway, that's the caspase that's going to cleave pro-IL-1 beta, pro-IL-18, and give you the mature cytokines. And right, remember the mature cytokines don't have a signal peptide. So how are they getting out of the cell? And that turns out because pro-inflammatory caspases, such as caspase 1, caspase 11, can induce this form of cell death, which is characterized by membrane perforation. And this allows the newly processed, the mature cytokines to exit the cell. But how exactly does membrane perforation happen? Now, in order to address that question, we did a forward genetic screen in mice. So this is an ENU screen. ENU, as you know, is a mutagen, introduces single nucleotide changes. We developed over 10,000 pedigrees. The screen is ongoing. Each pedigree has on average 30 to 40 mutations. Okay. And what we did was we asked a very simple question, which was from each pedigree, we obtained cells of the innate immune system, bone marrow derived macrophages, and we simply activated the inflammasome. In our case, we activated it with cytosolic LPS, and we measured the release of IL-1 beta. And so this would happen in wild type mice. And what we were hoping was that we would find a strain where this was broken. So we would stimulate with LPS and not see the release of IL-1 beta. And this is the genetics is a bit complicated, but for our purposes today, you can then narrow down the mutation to a single gene. And so this is an example of such experiment. We are measuring the release of IL-1 beta on the y-axis. In this case, we're stimulating with LPS to engage the inflammasome. And as you can see, the vast majority of the strains make really copious amounts of IL-1 beta. Now, the caspase-11 mutant doesn't, but that's our positive control, right? So you don't engage the inflammasome if you're in response to LPS if you're mutant in caspase-11. But look here, there's another mutation, ENUB11, that is just as compromised as the caspase-11 mutation. And what was this? This turned out to be a protein, Gesdermin D. We'd never heard of it. In fact, going to the library didn't help. Because when we went to the library, it said it's a member of the Gesdermin family, contains an ill-characterized Gesdermin domain, no obvious signal sequence or transmembrane segment, and function unknown. So that wasn't much of a help. But what we did realize is Gesdermin D is required for the exit of mature IL-1 beta to the outside. So what is the experiments we did that convinced us? So remember, I said caspase-1 will cleave pro-IL-1 beta. Pro-IL-1 beta is about 31 kD. And it's going to give you the mature biologically active form of IL-1 beta, which is about 17 kD. Okay. And the question is, how does this get out of the cell? So we're going to do immunoblot. We're going to blot for pro-IL-1 beta and processed IL-1 beta. And we are going to look at the situation in the absence of any treatment and in the presence of ATP. So now what we've done in this experiment is we've taken wild type macrophages from wild type mice, and either not treated them in the absence of any treatment or treated them with an inflammasome activator. And you will see immediately, we see the prompt presence of the 17 kD processed form. And in fact, we can measure it by ELISA. So it is secreted, it's in the supernatant. So in the wild type situation, pro-IL-1 beta goes to mature IL-1 beta and it gets secreted. Now let's do the same experiment, but with Gesdermin D null macrophages. When we do that, pro-IL-1 beta is processed to IL-1 beta. You're getting mature IL-1 beta. So that's good. But look at this. It's not getting out of the cell. So from experiments like this, we were compelled to conclude that Gesdermin D is required for the exit of IL-1 beta from the cell. Okay. You still get proteolytic processing. Pro-IL-1 beta goes to IL-1 beta, but you require Gesdermin D to get out. Now here was a surprise, which is that the pro-inflammatory caspases 1, 4, 5, 11. I just show you the data here for 11. Actually cleaved Gesdermin D. And they did so only on inflammasome activation. So let's look at the data for this. This is a Western blot. Here we are blotting for Gesdermin D. So it's there in wild type macrophages. It's there in caspase 11 null macrophages, but it's absent in 2 knockout strains for Gesdermin D. So we are looking at the right band. Now, what we're going to do is we're going to stimulate the inflammasome with LPS. And if you look at the wild type situation, you will see that very quickly there's a diminution in intensity of the wild type band. And concurrent with that is the appearance of a P30 fragment. Now, importantly, this doesn't happen when you stimulate caspase 11 null macrophages with LPS, suggesting that caspase 11 is cleaving Gesdermin D. And so we were able to show this using recombinant proteins. If you take Gesdermin D and incubate it with caspase 11, you see 2 predominant fragments, P30 and P20. And the cleavage is after an aspartic acid residue. As I said, caspases cleave after an aspartic acid residue, in this case, aspartate 276. So what I've told you then is that when the inflammasome gets activated, the pro-inflammatory caspases, and I showed you the data for caspase 11, cleave the single polypeptide, Gesdermin D, that very little is known about or was known about. And it gives you 2 fragments, P30, P20. Yeah, but you could ask, well, it gets cleaved, but is the cleavage important? I mean, what does it matter? So in order to address that question, we made a knock-in mouse where we altered the aspartate 276 to an alanine. So now this knock-in mouse is expressing Gesdermin D, but it can't be cleaved by pro-inflammatory caspases. Okay. And now we isolate macrophages from wild-type mice, and these mutant mice stimulate with LPS and measure the release of IL-1 beta. And when we do that with wild-type mice, they really make a ton of IL-1 beta. When we do that with Gesdermin D null mice, that's how we discovered Gesdermin D, then not surprisingly, IL-1 beta does not get outside of the cell. But look at this. When we do this with the mutant form of Gesdermin D that cannot be cleaved by caspase 11, it also, there's no release of IL-1 beta. So for the release of IL-1 beta, not only do you need Gesdermin D, you need to cleave it. So this made us think that maybe what we've stumbled upon, since it's required for IL-1 beta secretion, you require cleavage to give these 2 fragments, that maybe one of these fragments is the membrane damaging entity. So very simple experiment. We just said, let's overexpress these proteins and see if we permeabilize membranes by looking at the uptake of a nuclear dye. So when we overexpress full-length Gesdermin D, the cells do fine. Overexpress the P20 fragment, the cells do fine. Overexpress P30, whoa. Now all the cells get intensely permeabilized and uptake the dye. So P30 is the fragment that's perforating the membrane. Okay. So now we were wondering, how is this happening? And we postulated that maybe what happens is that once P30 is generated, it self-assembles, oligomerizes to give a pore in the membrane. And we were able to give some credence to this notion by doing a reconstitution experiment. We basically take purified liposomes, we take full-length Gesdermin D and cleave it with caspase 11 in the presence of these full-length liposomes. So we generate this P30 fragment. And lo and behold, what we find is that in short order, when we look by negative stain electron microscopy, you see these pores forming in the liposomes. And we calculated that this was a 24 symmetric protomer, so that's 24 P30s coming together to form this pore, which is a fairly large internal diameter of about 15 nanometers. Now, I should say that we discovered Gesdermin D in 2015, and it was also discovered at the same time by Feng Shao. And we attempted to do its structure, but that work on the structure has really come from Hao Wu at Harvard University. And I'm summarizing her results in this slide because she has achieved a mechanism at the atomic level for what we see in our liposomes. And so essentially, what I just said is that Gesdermin D is a single polypeptide, and in a cartoon representation as P30, P20. And it looks like this on structure. So the P20 is embracing the P30. The protease cleaves between P30 and P20. The P20 is released, and look what happens to the P30. The P30 undergoes a substantial conformational change. So there's this unveiling, unruffling of these beta sheet strands that insert into the membrane and give you this spore-like structure, in her case, about 18 nanometers. So I think the work that we did with the genetics, the discovery of Gesdermin D, and the subsequent structural work all coalesce to suggest that this is the fragment, that the P30 is the fragment generated from Gesdermin D that mediates spores in membranes and allows for the release of leaderless, signal peptide-less cytokines. So this is a summary of that. So that when Gesdermin D is cleaved, you get insertion into the plasma membrane, you get the formation of a pore. The spore is quite large, estimates are 15 to 20 nanometers. This, of course, is going to collapse the electrochemical gradient, but is large enough to allow IL-1 beta and IL-18 through because they're about 4 nanometers. But obviously, when you collapse the electrochemical gradient, you're going to get lysis of the cell that we measure by measuring the release of macromolecules like LDH. All right. So in the last part of my talk, I'm going to talk to you about a discovery that challenges a notion, that challenges dogma that's in our high school books of biology. So what is that? Now remember, when we do these forward genetic screens, we are always stimulating with an inflammasome engager like LPS and measuring IL-1 beta in these 10,000, 12,000 pedigrees. And generally, unless there is a defect in the secretion pathway like Gesdermin D, we see enormous amounts of IL-1 beta being released. And now remember, I just said to release IL-1 beta, you have to make these big pores in cells, 18, 20 nanometer pores in cells that collapses the electrochemical gradient and the cell lysis. And we measure cell lysis with the release of LDH. So just as a sanity check, we say, whenever we do IL-1 beta release, we also measure LDH release. And always they go hand in glove. If you get high IL-1 beta, you're going to get high LDH. That's just, that's just basic cell biology. But then we found this mouse here, high IL-1 beta, but very low LDH. I'm like, how can that be? The cells from this mice, the NINJ1 mice, after treatment with LPS, are dead. So IL-1 beta is small, 17 kDa. So in the NINJ1 mutant mouse, that's getting out. But large molecules that we measure cell lysis by, like LDH, that's not getting out. Now, LDH is a standard measure for cell lysis. It's used in the hospital. It's used in the research lab. Kits are sold for measurement of LDH. This was a shocking finding. This would be like if you get a high school student in your lab and you say, go and kill some cells with cyanide, and then take this kit and measure LDH. And he comes back and says, sir, madam, the cells are dead, but there is no LDH release. You'd be like, that's not possible, because that's how we measure cell lysis, with LDH release. So what's going on here? So we decided to knock out NINJ1 and made a knockout mouse. And we are here comparing macrophages from wild type mice with the NINJ1 knockout mice. The knockouts are in red bars. And we are going to stimulate with LPS. That's how we engage the inflammasome in our case. And we are going to measure IL-1 beta release. And much like our preliminary studies, we saw there was no problem in the release of IL-1 beta with the NINJ1 knockouts. It's the same as wild type. And in fact, we confirmed that in the NINJ1 knockout, Gesdermin D pores are forming. That these pores are forming because there is an antibody that is specific for P30. So we could blot with this antibody and say, okay, P30 is being formed in the wild type and it's being formed in the NINJ1 knockout, and IL-1 beta is getting out of these cells. But now when we measure LDH release, obviously it gets out of the wild type cells, but it's not getting out of NINJ1 null cells. So again, what is NINJ1? So NINJ1 is a plasma membrane protein. It has a very unusual architecture, N-terminus out, C-terminus out, 2 transmembrane domains, and has external amphipathic helix. So how can I summarize in cartoon fashion what I've just said? When you damage membranes, irreparably damage membranes, then we are always taught that cell lysis is a passive osmotic process. But what this data says is no, that you can actually genetically divorce, genetically separate, genetically dissociate membrane damage from membrane lysis, because membrane lysis following membrane damage is not a passive process, but requires NINJ1. So let me just, because this is such a surprising finding, let me just summarize what I've said, that the Gesdermin D pore is large enough for IL-1 beta and IL-18 to get out. You collapse the electrochemical gradient. But what appears is that the subsequent lysis is not a passive event, but is an event that requires this membrane protein, transmembrane protein, a NINJ1, 2TM protein, that allows for the formation of much larger disruptions in the membrane. We refer to it colloquially as megapores. And this allows for the release of high molecular weight substances. We just measure LDH, but we can do proteomics and really confirm that it's high molecular weight proteins. And a lot of these high molecular weight proteins are actually mediators of inflammation, like HMGB in complex with nucleosomes. So this is what I refer to as DAMPs. Okay, so many of the large molecular proteins that are released upon cell lysis, remember, are mediators of inflammation. Okay. All right. All right, okay. So let us, so again, unexpected discovery, ill-characterized protein, small protein, 16 kDa molecular weight, cell surface expression with true transmembrane regions. And it's conserved. It's conserved all the way down to the fly. And Janet Thornton just gave a beautiful talk on AlphaFold. If you use AlphaFold predicted structures and then do more sophisticated searches of the structure database, you see equivalent molecules in all forms of life, NINJ1-like molecules, in archaebacteria, in prokaryotes. So we are scratching the surface here. Very little is known about it. It's basically expressed in all cells, but especially high in myeloid cells. It's induced after, you go back to the literature, there's not much there, but it is induced after injury. When we knock it out, we don't quite understand this, but about a quarter of the mice die of severe hydrocephalus, but once they get past that checkpoint, they seem to do okay. Now, a picture speaks 1,000 words. So this is what you've all seen. We're treating cells with nigericin, a potassium ionophore, a poison. The cells balloon, and then they shrink, right? That's what you're expecting. So that is your typical necrotic death. Now, look what happens if you take NINJ1 null cells and expose them to this poison nigericin, this ionophore. It'll kill the cells, no question about that, and we show that every which way but up. But the cells remain ballooned. They don't collapse. They remain ballooned. And I was talking to a colleague at the VHI, and he said that when your paper came out, we were so disbelieving, we immediately CRISPR knocked out NINJ1 in 3 different cell lines. And guess what? He said, they ballooned and stayed ballooned. Now, but this is a very important point, because I've been talking about inflammation, I've been talking about cytokines, but I want you to remember this, that NINJ1 appears to play a role in plasma membrane rupture globally, even independent of Gesdermin D. So let's look at the data. We're looking at cell rupture here, LDH release. Again, wild type mice are open bars, red bars are NINJ1 null macrophages. And if you look in the absence of any treatment, there's about a basal level of about 18% background cell death. If you freeze thaw the cells, then there's no difference, as you would expect. The nigericin data I just showed you, where the NINJ1 cells are protected from exploding. But look at this, cisplatinum. This is a chemotherapeutic that kills independently of Gesdermin D. And here, the release of LDH, here cell lysis also appears to be dependent on NINJ1. So Gesdermin D independent membrane damage also requires NINJ1 for membrane rupture. So many roads to membrane damage all coalesce on requiring NINJ1 for eventual lysis. So now, this is a post-death event, right? So the cells have sustained irreparable membrane damage. They're essentially dead. And then NINJ1 comes into function to give you these megapores that collapse the balloon and allow you to visualize what you normally see, which is an imploding necrotic cell. Well, one assay that we have, it's a surrogate assay, is that if we overexpress NINJ1, it is fiercely toxic. It's just going to dissolve the plasma membrane. And so, but that becomes a good surrogate assay for us. If you take human or mouse NINJ1, you overexpress it in 293 T cells, you see membrane rupture that you can measure. But here's the fly NINJ1 from Drosophila. You overexpress it in 293T human cells and boom, it dissolves the plasma membrane. Well, lyses the plasma membrane. Now, what do we know about NINJ1? So this is a schematic cartoon going from the N terminus to the C terminus of NINJ1. This is the amphipathic extracellular helix. This is the first transmembrane, second transmembrane. And this is a conserv score, which is a score about conservation of sequence. Lower the score, higher the conservation. And you'll see that this amphipathic helix is highly, highly conserved through evolutionary time. Now, if we do a scanning mutagenesis where we alter 5 amino acids from the N terminus to the C terminus to alanines and assay for their ability to lyse cells, of course, wild type NINJ1 will lyse cells quite prominently, shown here as a killing score. The very N terminus is fairly tolerant of mutations, but as soon as you mess with the amphipathic helix, you lose the ability of NINJ1 to lyse cells. What about if we look at endogenous NINJ1? So when we look at endogenous NINJ1, here are wild type macrophages. We are exposing them to nigericin, which is this ionophore poison. In the absence of any exposure, NINJ1 is monomer, polymer, predominantly monomeric. As soon as we expose to nigericin, at least by 1 hour, 2 hours, you find these very large polymeric species. Now we know we are looking at the right bands because it's absent in the NINJ1 knockout. And when we do immunofluorescence staining, again, this is the NINJ1 knockout that does not stain, so that's our control. We see that when the cells are exposed to nigericin, very quickly NINJ1 goes from the plasma membrane to form these punctate-like structures, large aggregates on the plasma membrane. Now, we were really interested in how is NINJ1 getting activated? Is it undergoing a post-translational modification? Is it undergoing phosphorylation or ubiquitination or some other chain? Turns out that's not the case. That's not the case, and I'll hypothesize why that may be. Regardless, the amphipathic helix seems to be very important. We can show that in reconstitution studies. So if we take macrophages that are null for NINJ1, then they're relatively resistant to a large dye release on exposure to a lethal stimulus, LPS. If you reconstitute with wild type NINJ1, then you reconstitute cellular lysis. But if you reconstitute with alpha breakers like this, alanine to proline mutation, then and you disrupt the amphipathic helix, then you lose the ability to lyse the cells. So what is happening? Now, we really don't know because there's very little literature on this, hardly any. So we think what's happening is that, that the NINJ1 is sensing the health of the plasma membrane. And remember the asymmetry of plasma membranes, the composition of plasma membranes, plasma membrane tension is all dependent on energy expenditure. So when a cell has died, there's no ATP, there's going to be an alteration in plasma membrane composition, dynamics, characteristics. Now we believe NINJ1 senses that, and that triggers an oligomerization because this amphipathic helix inserts itself into the plasma membrane, and this triggers this oligomerization event. But now you're asking yourself, you're saying, hold on, this is a post-death event. Why is it conserved in evolutionary time? Why has evolution conserved this from fly to man, certainly for NINJ1, and NINJ1-like molecules, maybe even further in evolutionary time? Why would you do that? Well, at least for NINJ1, our model is that the very rapid lysis of the cell and the release of these large molecular weight entities, many of which are pro-inflammatory, allows for a more rapid immune response. So if that is the case, then you would say if mice lack NINJ1 and cannot mount this optimal inflammatory response, they should be more susceptible to infections. Conversely, you can say that in sterile inflammation, right, where there's no pathogen, but let's say chemically induced inflammation, sterile inflammation, that in that instance, you should be relatively protected because the release of inflammatory mediators is diminished. And indeed, that's what we see if we take our knockout mouse and expose them to this pathogen Citrobacter rodentium. The wild type mice do relatively well. They survive. This is a survival curve. But the NINJ1 knockout mice struggle, and by about 2 weeks, half of them have died, consistent with our model. Alternatively, if you take a model of sterile inflammation where we intratracheally instill bleomycin, and measure body weight, we see that whereas wild type mice very rapidly lose body weight, the NINJ1 knockouts are somewhat protected. So these animal models are consistent with this hypothesis. More recently, however, we've generated a neutralizing antibody to NINJ1, because remember, it's a plasma membrane protein. And this we believe will be an important tool because it actually protects cells from lysis. So this is the release of a large molecular weight dye as a function of time after exposure to nigericin, and wild type macrophages release the large molecular weight dye because they're undergoing lysis. The knockout does not, which as I've shown you. But if you take wild type cells and treat them with the antibody, then it's just as inhibited in the release of intracellular, large molecular weight intracellular contents as the knockout. But how is all of this working? Is NINJ1 really forming a megapore? So, you know, when Howard Carter was asked, when he peeked into Tutankhamun's tomb, he was asked, what do you see? Can you see anything? He said, yes, wonderful things. And this was our mini Tutankhamun moment. So when we did cryo-EM, we suddenly realized NINJ1 was oligomerizing into these very large filaments, about 6-7 times the diameter of a Gesdermin pore. And when we look at higher resolution, we appreciate that there is this serrated tooth pattern to the filaments, which we think are bundled helices. But it'll require further analysis to get a structural insight into how this molecule oligomerizes. And it'll probably require liposomal reconstitution studies to see how exactly is it being activated, what in the membrane environment triggers its oligomerization. So that's the summary. I've talked about Gesdermin D. And then I've said when membranes get damaged, that subsequent lysis, contrary to what is taught in high school biology books, is not a passive event. It actually requires a protein, requires NINJ1. This accelerates lysis dramatically and leads to the release of higher molecular weight substances. So with that, I'll end. I'll thank my colleagues who've done the work with me. In particular, Nobuhiko Kayagaki, who has been a wonderful colleague for over 20 years. We work together, and his associates who are gifted experimentalists, Irma Stowe, Bettina Lee, and Ofer Konfeld. The forward genetics work, which is a very huge undertaking, doing it in mice, has been a large collaboration with Chris Goodnow and Ed Bertram in Australia. And some of the initial structural work was done with our structural biology group at Genentech. So with that, I'll stop and be happy to take questions.
Thank you, Dr. Dixit, for this wonderful talk. There are a few questions. Avik, can you unmute and ask the question?
Yeah, so I have the question that is, so what can be the reasons that the cells develop a distinct pore forming mechanism for secreting IL-1 beta? I mean, why do the cells not follow the conventional way of leader sequence based secretory pathway to secrete IL-1 beta? Does it provide any advantage to the cells?
Yeah, you know, it's difficult to experimentally verify one's notion. I suspect IL-1 beta and IL-18 only come into play when cells are faced with a lethal challenge. And that's when they sound the inflammatory alarm. So baseline cytokine levels, especially from innate immune cells, especially from macrophages, probably that explosive release of these cytokines is orchestrated with cellular lysis, the lysis being an indication of damage, of cell damage. But it's difficult to ask nature questions like why doesn't IL-6 not have a signal? Why does IL-6 have a signal peptide? It's difficult. I mean, I think we have to, one can only come up with, I can think of experiments to do to address your question, which is to give them signal peptides and see, but I'm not sure where that would lead.
Yeah, so I was actually asking one question just in continuation to it, like what if we put a secretory leader sequence in IL-1 beta in the background of gasdermin D knockout? So can it be like impairing immune functions? So can this sort of experiment answer this question?
Could be, yeah. No, I think it can be done experimentally. I think the question you pose certainly can be addressed experimentally. Absolutely, one could engineer the leaderless cytokines to have signal peptides and then simply ask, how do you alter the immune response?
Okay, yeah, thank you, sir. Thank you.
Prasad, you want to unmute and ask?
Yes, Sandeep, thank you. Great talk, Dr. Dixit. So I was wondering, like, have you looked at any escort masonry recruitment or involvement of escort pathway in this NINJ1 mediated damage?
Not really. I would say that it doesn't come up on CRISPR screens, but the escort pathway components are essential. It doesn't come up on CRISPR either. I'm not sure about the involvement. So I would say we don't have any evidence of involvement. That's not to say it is not involved.
Great, thank you.
Jasminara, you want to unmute and ask?
Hi, Dr. Dixit. I just wanted to understand, like, what prompted you in the first place to look into the cleavage of GSMD, like the GSDMD protein. Like caspases usually cleave IL beta, but what made you think that it might be cleaving GSDMD?
So that's a great question, and that's why we all are doing what we do, because we thought, how is gasdermin D getting activated, right? So the favorite model was maybe it's getting phosphorylated. So we ran a gel and we said, hold on, it's getting cleaved. But then the question was, well, a lot of things get cleaved in dying cells because, there are proteases around. Is this cleavage important?
Yes, yes.
And then we made the mutant and addressed that, right? So just looking at the data and let the data do the speaking. Same with, now with NINJ1, we're in a similar quandary. Like, how is it getting activated? I think it's a more difficult question than gasdermin D, but we're hoping as we continue to, maybe that's why I went to the liposome reconstitution studies, if we can see what the effect is, what lipids influence it, what environment influences it, we may get a better notion. But yeah, that's a complete, just looking at the data. There was no reason to look at the cleavage. There was no literature, right? There wasn't anything that said this family gets cleaved. Yeah.
Thank you. Thank you.
Can I ask you a question, Dr. Dixit? This is Srivatsan from Los Angeles.
Please.
I was wondering, it's quite good in model systems. Did you find any of the, either the cancer model or neurodegeneration model where you have this NINJ1 mutations leading to resistance?
Yeah, no, that's a good question. It's early days. There is a report of a Japanese family that has a polymorphism in NINJ1. that incidentally they noted was associated with very low LDH release. So it's clinical literature, obscure paper, but wow, when we found it, we said, that fits right in. But this applies more probably for Gesdermin D and NINJ1. What is their role in disease? Are there any deficiencies? I think all of those are very pertinent questions that will be addressed in time, and especially also the making of inhibitors. There's a lot of inhibitors available now. I think the Gesdermin D inhibitors that have been published, but they also hit other components of the pathway like caspases. So they're more pleiotropic in their action. But if we could get a highly specific inhibitor of Gesdermin D, would it have therapeutic benefit? So those are all very interesting questions that this work has opened up.
Because at least in cancer, I would have thought you might have seen it, because cisplatin resistance is a common factor, and of course, people go into metastasis.
Yeah, no, we haven't looked at it ourselves. There is a body of literature that suggests that other members of this Gesdermin family are important in killing of tumor cells by granzyme A. So that's not granzyme B, which is the major granzyme. Granzyme A is a minor granzyme, but granzyme A seems to have function in certain contexts by cleaving Gesdermin D family members to engage cell death. But that's not our work.
Dipankar?
Thanks.
Yeah, a fascinating talk, Professor Dixit. I was, in fact, I had a question along the similar lines. Are there polymorphisms associated with NINJ1, and are those associated with any sort of pathological consequences?
Yeah, no, I think that's something we're going to have to look at. I think if there are polymorphisms in these large, like COVID studies, that in particular, if you could see that if you had loss of function, or if you had a hypomorph in Gesdermin D, you may in fact be protected from the cytokine surge that gives you COVID lung, but that'll require pretty detailed analysis.
Yeah,
thank you very much.
It goes back to the Dr. Dixit about the polymorphism being not that important.
Other people are waiting to ask questions. Let's finish them first.
Okay, thank you.
Yeah, so Dr. Dixit, this is Rangarajan, very fascinating talk. So I just have one question. Have you compared the secretomes of wild type, Gesdermin knockout, and the NINJ1 knockout cells? How many missing proteins you may find?
No, it's a good question. We published that. So that's in our paper this year, the Nature paper on NINJ1. We did a full proteome analysis, and it's consistent with the model that I just showed you, that NINJ1 is high molecular weight.
So did you find any other unknown proteins which have not been reported earlier that will require these proteins to come out?
Yeah, I would ask, I look at the database. I don't remember the details. I mean, we focused in on the ones that are known mediators of inflammation, but have a look at the database.
Okay, there are a couple of questions in the chat box. Sharath is asking, could there be any function associated with P20?
I think it's a great question. People have looked long and hard, and nothing to date. No reported function.
So one more question. Ramesh is asking, does NINJ1 has any role in vesicle formation and release?
We can't exclude it. I think it's a very good question. When I begin to wonder, because NINJ1 is very highly expressed in the choroid plexus, maybe the hydrocephalus is related to that. I mean, it may have a day job, or at this juncture, we really don't know. Nothing directly to suggest in all our work, which is sort of preliminary. nothing to suggest vesicle formation, but it's a good question, good point. I think it'll require a lot more work and may require the appropriate context, like as I mentioned, the choroid plexus.
Thank you. Okay, I think this is the end of the session. Let's thank Dr. Dikshit for sharing his exciting work with us. Thank you, Dr. Dikshit.
Thank you.
Over to BICS volunteers.
Thank you, Dr. Vishwa, for the wonderful talk, and Professor Sandeep for chairing this session. At the end of day 2, we thank all the speakers and chairs, including the students and alumni of today's sessions, for the insightful talks and discussions. We also thank the audience for attending today's events. We hope to see you all again tomorrow at 4 p.m. IST, either on Zoom or YouTube. Until then, thank you, everyone. See you soon. Stay tuned and good night.